So Dr. Swami is there, eh?
Yes, sir. Good morning, sir.
Good morning.
I am Dr. Mahadev Swami from US Raichur, sir.
Okay, very good. Good morning.
Yeah, very good morning, sir.
Good morning, sir.
Good morning, Dr. Swami. How is your work going on? How is your diversity park? There is a lot of demand for planting material. Please gear it up, production and distribution.
We are multiplying, sir. We are multiplying.
Because I am diverting all my calls to your number.
Yes, sir,
So that you can give them proper support.
Program schedule.
Many calls, sir.
Sir, good morning, sir.
Good morning, good morning.
Aram, sir.
ಆರಾಮಿದ್ದೀರಿ ನೋಡಿ, ತಾವಾಗಿದ್ದೀರಿ?
ಚೆನ್ನಾಗಿದೆ, sir.
Very good. Denish ಅವರೇ, good morning.
Thank you, sir.
Sir, good morning, sir. Shall I start, sir?
Yeah.
Okay, we are ready.
A very warm good morning to everybody. First of all, I would like to introduce myself. I am Dr. G.K. Halesh, working as Assistant Professor, Department of Biotechnology and Crop Improvement, College of Horticulture, Bengaluru. I am also a co-organizer for this e-conference. Excellencies, distinguished delegates, learned delegates from the corporate and academia and research institutes and industries, it gives me immense pleasure to extend you all warm welcome on behalf of the Department of Biotechnology, Crop Improvement and the College of Horticulture, Bengaluru and the University of Horticultural Sciences, Bagalkot. I welcome you all for this international e-conference on advances and future outlook in biotechnology and crop improvement for sustainable productivity from 24th to 27th November 2020. During these 4 days, we will get plenty of opportunities to discuss, share the knowledge and insights in the field of agricultural sciences, that too in the various aspects of biotechnology and crop improvement to ensure the global food and nutritional security, multidisciplinary efforts using modern scientific and technological advances with special relevance to biotic and abiotic stresses. We are providing a platform to discuss and address the problems in the field of horticulture and agricultural crops. Encouragement, enthusiasm, excellency are the major aspects of the organization, with an object of encouraging the spirit of research and innovations. Department of Biotechnology and Crop Improvement feels proud to host the symposium. This 4-day seminar or e-conference consists of 10 different themes. Each theme is having one keynote address, 4 to 5 invited speakers, following 6 to 7 presentations. For this esteemed functions, our Honorable DDG, Deputy Director General of Horticulture Crop Science, Dr. Tilak Raj Sharma, and the Indian Council of Agricultural Research, New Delhi, has given a consent to become the chief guest of this program. So it will become incomplete if I just don't tell the accomplishments of Dr. Tilak Raj Sharma sir. I would like to introduce Dr. Tilak Raj Sharma sir. After completing his doctoral degree from Himachal Pradesh Agricultural University, he completed his postdoctorate from University of Alberta, Canada, in the area of molecular biology of plants and plant pathogens. Subsequently, he did 2 more PDF trainings in Cold Spring Harbor Laboratory, New York, USA. So he has worked in various capacity, starting from assistant professor at HPKV Palampur, with effect from 1991 to 99. He has joined as senior scientist at NRCPB New Delhi, IRA New Delhi. Okay, after that he had worked as principal scientist from 2007 onwards. He contributed extensively for 16 years of rice crop improvement His significant contributions include mapping, cloning, and functional validation of rice blast resistant gene, PIKH. He has authored more than 95 research papers and students. He has bagged several awards, University Gold Medal during his doctoral program, Young Scientist Award, NAS 1998, ICR Young Scientist Award, Narasimhan Academic Award, and then Umakant Award in Biochemistry, Biophysics and Biotech, Indian Science Congress Award, DBT National Bioscience Award for his career development, NAS Recognition Award during 2008, and several other awards he has taken. So I am very proud to say that Sir has accepted our invitation for this inaugural program. Now, I request, with this brief introduction, may I now request Dr. G.S.K. Swami, Dean, College of Horticulture, Bengaluru, and also Chairman of this e-conference. Okay, I welcome Dr. Swami sir to welcome the honorable delegates. Dr. Swami sir, please.
Thank you, Alesh. Ellargu namaskara. Good morning to all. It is indeed a pleasure to have a gamut of professionals and dignitaries who have congregated virtually for the e-conference on advances and future outlook in biotechnology and crop improvement for sustainable productivity. I wholeheartedly wish that deliberations to be held on these 4 days would meaningfully address the core issues of the conference. At the outset, I welcome Dr. Tilak Raj Sharma, the Honorable Deputy Director General Crop Science, ICR New Delhi, who has gladly agreed not only to inaugurate the conference, but also to enlighten us about this frontier development in this field through this keynote address. Welcome you, sir, on behalf of our university and on behalf of all the organizing team. It is because of the constant support and motivation of our Honorable Vice Chancellor, we are conducting this virtual meet. I delightfully welcome Dr. K.M. Indresh, the Honorable Vice Chancellor of our university and President of this today's inaugural program. Dr. S.P. Dandin, sir, former Vice Chancellor, and Dr. M.R. Dinesh, Director of IHR Bangalore, also gracing this occasion as guest of honor for this inaugural program. I welcome both of them on behalf of the conference team and also university. I also welcome all the key officers, Dr. T.B. Alloli, Registrar, Dr. D.R. Patil, Director of Research, Dr. Y.K. Kotikal, Director of Extension, Dr. M.S. Kulkarni, Dean PGS, Dr. S.I. Athani, Dean Students Welfare, all the members and students of our university for this inaugural program. I welcome Dr. Fakhruddin, Organizing Secretary, and also with this conference and his team for this inaugural program. This program has been possible because of the participants across the globe who have enrolled to present their research findings in this conference. I welcome all of you on this occasion. Once again, I welcome all present in this meet with the wish and hope that the conference would enlighten us on the frontier development of this field. Thank you, Anna.
Ladies and gentlemen, I thank Dr. G.S.K. Swami, Dean, College of Horticulture, Bengaluru, as he has welcomed all the delegates of the e-conference. Now, I would like to request Dr. B. Fakhruddin, Professor and Head, Department of Biotechnology and Crop Improvement, College of Horticulture, Bengaluru, and Organizing Secretary of this e-conference to brief about the conference. Dr. Fakhruddin, sir, please.
Ellargu namaskara. Good morning to everybody. Respected DDG, Tilak Raj Sharma sir, our Vice Chancellor, Galaxy of dignitaries, all the learned delegates across the countries who have registered for this conference. Interestingly, when we started the conference, it began with a few registrations, but right now we have about 1,100 participants participating in this particular conference. We have, as we all know, the biotechnology being an important discipline in agriculture and horticulture. It is making its big impact, a greater impact in many disciplines such as genetics, plant breeding, molecular biology, physiology, seed science, and other related fields. The toolbox of biotechnology and that of the plant breeding is continuously growing. Much recently, we have been seeing the tools that are really very powerful that can do a few things that previously we could not do as biotechnologists and plant breeders. And this has opened up a new opportunity for breeders and biotechnologists to breed new varieties and come up with technologies that are really useful at farm level. Apart from this, the NGS platforms and associated technologies have really become handy and affordable at this moment. And this also has enabled all these disciplines which are involved in crop improvement and generation of new technologies. This has brought the genetic engineering also to a newer height wherein things can be done much better given the backdrop of the regulatory issues. Now, with all this, what we thought during this COVID pandemic time, we should assemble, the experts should come together and maybe share the knowledge so that the researchers, young teachers, researchers, and scholars would get benefited by this. We have in all about 10 themes in this conference, starting from the PGR, plant genetic resources, to data science and bioinformatics, covering range aspects. So, interestingly, all the great agree by the experts across the globe, many good experts who have made their own name in each one of these disciplines have agreed to participate and give the either keynote talk or the lead talks in each one of the theme. These keynote and invited speeches are going to be really a learning experience to most of us. I really thank all the experts from all across India, from USA, Canada, Malaysia, and other countries who really agreed to be part of this particular conference. We hope that this conference, to give you one more information, we received about 680 abstracts covering all the 10 themes that we had. In fact, till last evening, we had abstracts for the experts who were on the panel and encouraged the delegates to register for it. And we have brought out a book, abstract book, which was released, and we will share that to all participants as we go on. So over 11 country participants from 11 different countries are there as part of this conference. Coming maybe 4 weeks are going to be a good learning experience. Looking forward for this feast of learning as we go on. Thank you everybody and welcome again to the conference. Thank you.
Now I would like to thank Dr. B. Fakhruddin, our organizing secretary, for this for elaborating this our program. I once again thank Dr. Fakhruddin. Okay. All the dignitaries, delegates, and participants, may I now request our honorable chief guest, Dr. Tilak Raj Sharma, Deputy Director General of Science, ICR New Delhi, to kindly inaugurate the e-conference just by telling e-conference is open.
Good morning to all. E-conference is open.
Ladies and gentlemen, at the same time, may I now request Dr. K.M. Indaresh, Honorable Vice Chancellor, University of Horticultural Sciences, Bagalkot, to kindly inaugurate the program by releasing the abstract book and the CD of our abstract book. At the same time, I request all the officers of the university, Registrar, Director of Research, Director of Extension, Dean PGS, Dean Students Welfare, to kindly join this inaugural program. Okay. Ladies and gentlemen, this is really overwhelming situation for me. I would like to thank Dr. T.R. Sharma, DDG Crop Science, Dr. K.M. Indresh, Honorable Vice Chancellor, UHS Bagalkot, Dr. S.B. Landin, Founder, Vice Chancellor of UHS Bagalkot, Dr. M.R. Dinesh, Director, IHR Esrigata, Dr. T.B. Alloli, Registrar, UHS Bagalkot, Dr. D.R. Patil, Director of Research, UHS Bagalkot, Dr. Y.K. Kotikal, Director of Extension, Dr. M.S. Kulkarni, Dean Postgraduate Studies, Dr. S. Athani, Dean Students Welfare, Dr. B. Raju, former Director of Extension and also Vice Chancellor, all the deans of our different colleges. Okay, I thank all the officers. Ladies and gentlemen, may I Tilak Raj Sharma, DDG Crop Science, ICR New Delhi, to enlighten us by giving conference keynote address on the topic application of biotechnology in achieving food and nutrition.
Good morning to all. Am I audible? Good morning to all.
Yes, sir, you are audible.
Yes,
sir.
Sir, audible,
Okay.
sir. Good morning, sir.
Good morning. Honorable Vice Chancellor of the University, Dr. K.M. Indresh, former Vice Chancellor and guest of honor, Dr. S.P. Landin, and guest of honor, Dr. M.R. Dinesh, Director, Indian Institute of Horticulture Research, Dr. G.S.K. Swami, Dean College of Horticulture, Dr. B. Fakhruddin, Organizing Secretary, distinguished officers of universities, deans and directors, participants across the globe, students, ladies and gentlemen. It gives me an immense pleasure to be here this morning to participate in this international e-conference on advances and future outlook in biotechnology and crop improvement for sustainable productivity. It's really a new medium of interaction during this COVID-19 situation, and we have seen that such conferences are being organized globally on various themes of biology and agriculture. So I would like to congratulate the organizers, particularly the Vice Chancellor and the organizing secretary and all the faculty members of university to taking this great initiative to have the conference on a very important theme for these 4 days. By looking at the program of conference, I was really amazed to see that almost all aspects related to biotechnology, molecular biology, next generation sequencing, everything is included here. Not only that, the list of speakers, which is really rare to bring together all these distinguished scientists at one platform when we always meet physically, because it is very difficult to come across the globe. But when you see the list of speakers in this whole program, is amazed to see that all who's who of biotechnology are included as speakers, starting from experienced old scientists as well as from young scientists and also mid-career scientists. So I'm sure that this conference will really gives an important opportunity to the students and young faculty of the universities and other universities in the country to learn some new things related to agriculture, biotechnology, and its application in crop improvement. In fact, I was contacted only 4 days back by the Honorable Vice Chancellor to be here this morning. And since this is a topic which is very dear to me, let me tell you that I started working on biotechnology from my PhD days from 1988 onwards. You can Google and see. As a PhD student, I started working on somaclonal variation, plant tissue culture, and interspecific hybridization using ovary and ovule culture. So the topic is very dear to me, and almost all of my students, more than 24 PhD students, they have worked on different aspects of genomics, disease resistance, and bioinformatics. So that's why I thought that it would be an important occasion for me to interact with all the participants sitting here in this conference. And I agreed to the request of the Honorable Vice Chancellor. We had short discussion. I postponed my other meetings to 11 a.m. so that I could join here this morning and interact with all the participants here. As told by the organizers, I am going to speak on a very structured topic, which is a very favorite topic of mine. So an application of biotechnology in achieving food and nutritional security, for which I'm going to share the screen, and I'm sure you can see it. Is it visible? Can anybody tell me, is it visible?
Yes,
visible,
we
sir.
can see it.
Okay, thank you. So this is the topic which I'm going to speak today, and I will try to include all the aspects related to biotechnology here in this topic. And knowingly, I chose this topic so that to set the tone of this whole conference on application of biotechnology in advancing, in achieving food and nutritional security. So you know that India is a very diverse country. We have 1.3 billion people spread across the country, which has more than an area of 3.288 billion square kilometers, and agriculture accounts for 16.5% of India's gross domestic products. And we all know that nearly half the population in the country depends on farm-based income and providing livelihood to the our population. But we see those who are in agriculture field, they know very well the history of Indian agriculture, what was there before independence and what is there after post-independence. We know that before independence, we were just like as a begging ball and surviving on the donation of food grains received from other countries, particularly from the United States of America and other advanced countries. But after post-independence, there was a green revolution because of the introduction of dwarfing gene by Norman E. Borlaug along with Professor Swaminathan at Indian Agriculture Research Institution, New Delhi. And besides that, there was a lot of irrigation facilities which started, improved hybrid seeds were available to the farmers, along with chemical fertilizers and pest management strategies. And above all, there was a political will at the Indian government side, which set the tone of green revolution. And we all know that Norman E. Borlaug got Nobel Peace Prize in 1970 because of this humane work which he did by introducing dwarfing genes in Southeast Asia. And then in India, Professor M.S. Swaminathan, who is known as Indian father of Indian Green Revolution, received World Food Prize in 1987. After independence, between 1951 till date, we see the growth of Indian agriculture. We have tremendous progress in almost all sectors. of agriculture. This is just an example. These figures might have changed now, but food grain production has increased 5 times, milk production 8 times, fish production 11 times, horticulture 6 times, meat 8 times, and eggs 27 times. In spite of the fact that India has only 2% of the world's land and 4% fresh water, what we support 16% of the world population and 10% of the cattle. So it tells us that we are progressing well in as far as Indian agriculture and horticulture is concerned, but there are certain challenges. The major challenge is our rising population. And you know that the population is very swiftly and steadily rising, whereas our food grade production is not following the same trend, as you can see in this particular graph. So there is a yield gap which can be fueled by increasing area under cultivation, which is not possible because we need land for other resources also. Increasing the yield potential of existing crop varieties, either by using conventional method or by using biotechnological tool, is the only resort which one can use for the increasing yield production in the country. Therefore, there are many approaches which we started using from 80s onwards, from plant tissue culture to GM crops, plant genomics, gene mapping, and marker assisted breeding and genome editing. So I would be giving you certain examples in all these areas which we have in biotechnology, and those who are dealing with plant, they know that plants has a very special character, that is each cell has an ability to give rise to a full grown plant which is called totipotency, and this is how we can start from a single cell to have full grown plants. And it's a very interesting industry, and we have very important tissue culture industry in India, because we know that in food crops, 66% of the banana is being multiplied through tissue culture by using clonal propagation of elite varieties. And those who are in horticulture field, particularly this university, they know that the single variety of banana Grand 9, which occupies more than 80% of the area, is 100% multiplied through tissue culture. Then under vegetable, we have around 2% potato, seed potato is produced through tissue culture. In spices, all these medicinal plants and more than 24% ornamental plants are multiplied through tissue culture. Even in plantation crops, sugarcane, around 2% of the sugarcane is multiplied through tissue culture. The tea, coffee, jatropha, all these are very important examples, including forest crops like eucalyptus, teak, and sandalwood. And we see that more than 200 commercial units are functional in India. And if you see the tissue culture in banana, it has become a sort of a cottage industry in India. I personally see many small units in different parts of the country, those who are working in micropropagation of banana and other horticultural crops. Tissue culture not only used for multiplication of elite germplasm and crop species, but using cell culture, one can make use of somaclonal variation, which is the variation arising from the culturing of single cell, and that is called somaclonal variation. By using that particular advantage of cell culture, Pusa Jaiksan, a high-yielding mustard variety, was released by IARI and NRCPV and long back, and it still occupies major area under this mustard cultivation in India. And because of its high yield, 19 to 25 quintal per hectare, and also it matures in 115 to 125 days. Another important aspect is that when we want to bring genes across the species, then it's very difficult to cross those two species, and one has to resort to somatic hybridization. So using somatic hybridization, this is an example again from NRCP, where male sterile system was transferred from Mauricandia, Brassica Mauricandia species to Brassica gentiana for the development of hybrid NRC Sankar Sarson and also Coral 432. And this is a very classical example where somatic hybridization has been used for the transfer of genes from one species to another species. Coming to second topic, which is genetically modified crops, all biologists, particularly agricultural scientists, are very well aware of this particular technology and the advantage which we have because of utilization of this technique, that is transgenic plants, how these are developed by using either agrobacterium tumefacients or by using microprojectile or bombardment gun for the transfer of species, genes across the species and across the genera to create genetically modified plants. And we see the progress of genetically engineered crops in the world. In 1996, when this crop was started, it was hardly 10 million hectare across the globe, but now 119 million hectare area is grown under transgenic crops in 236 countries, and India is among the first 5 countries by adopting only one transgenic crops, that is cotton. So it's a very successful story of the BT cotton in India, wherein single gene which has changed the whole scenario of the country from starting. We were importing cotton before the introduction of this gene because there was great loss because of cotton bollworm, Helicobacter pharmacea insects. Once this gene, which was derived from a bacterium Bacillus thuringiensis, BT gene was transferred in cotton, which is commonly known as BT cotton. There is that whole scenario has changed, and these figures, if you see in this table, that clearly indicates the advantage of this particular technology. When this was introduced, there was only one event and 3 hybrids we were having in 2001 and 2, and now more than 1,000 hybrid cottons, BT cottons, are available in the country. Even this year, only last month, we released more than 50 hybrids of different companies which are the different parts of the country containing BT genes. The most striking example here which one can make out is that pesticide spray, which was more than 5,748 metric ton before the introduction of BT cotton, has come down to 220 metric ton, and it has started increasing the overall income of the farming community. Similarly, BT brinjal is a very successful story, and you know this fruit and shoot borer is also from the same family, Helicobacter pharmacea, and can be effectively controlled by the introduction of BT gene with this fruit and shoot borer, and more than 12 to 15 chemical sprays are being employed by the farming community to control this particular insect. However, for the introduction of BT genes in brinjal, this can be completely controlled, but because of certain government clearances, this particular product could not be released in India. But you know, our neighboring country, Bangladesh, has already started reaping the benefit of this particular crop, and they are growing BT brinjal from 2014 onwards. These are other examples of GM technology which was used for the development of hybrid mustard by Indian scientists, but still it is waiting for the clearance from the government side. So there are very successful examples world over in case of transgenic crops, where we have to think very seriously and convince the stakeholder that this is a technology or a future technology which can be used effectively. The third approach which is used in agricultural biotechnology is plant genomics, where decoding of genome sequence of complete organisms has become possible, particularly with the advances in sequencing technology from 1999 onwards, when automated DNA sequences was developed, and one can see that Sanger's diary of method was the method of choice till 2005, but when virus sequencing was developed in 2005, it has become the method of choice. And now next generation sequencing technology, which is one of the topics in this conference, these are the certain platforms which are used for decoding of genomes of various species. Of all these different platforms, which many of them has become obsolete now, we have PacBio, RS3, and also Iron Torrent and Illumina platform, which is available still in the country, and Oxford Nanobiotechnology for long read sequencing is available, wherein one can give DNA samples to the companies. Various companies are there, and then get sequence of whole organisms. So whatever method you use for decoding of a genome of an organism, at the end of experiment, you are going to get a typical string of adenine, guanine, thymine, cytosine, 4 letters present in typical permutation and combinations, which is called genome sequence. Whether you isolate genome sequence from a microbe, from a plant, an animal, or human being, the composition of sequence is same. That is 4 letters: adenine, guanine, thymine, cytosine. And DNA is a very large molecule. It is very difficult to sequence whole DNA molecule in one go. Therefore, that is fragmented into small pieces and then sequenced. And by using computational analysis, genome assembly is performed to get whole genome sequence. And this is one of the examples wherein Sanger's dideoxy method was used to sequence rice genome, and the paper was published in 2005 in Nature, and wherein we sequenced Nippon ware genome of Nippon ware cultivar of rice having 388 million base pair sequence and predicted 37,000 genes. It was an international effort, and India was partner in this particular effort along with our institute. After that, tomato genome was decoded in 2012 by using a hybrid sequencing approach, Sanger's dideoxy method and the NGS method, wherein we sequenced Heinz 1706 variety of tomato with the genome size of 900 million base pair and predicted around 34,000 genes. And you can see the down below in this particular slide is a typical multi-dimensional tomography of chromosome number 1 of tomato. Herein you can find each and every aspect of tomato, chromosome 1 to 12. Tomato also has 12 chromosomes. This whole paper is published in Nature, and it has 187 pages, and herein you can find each and every aspect of the whole genome. Then third genome which we sequenced is by using exclusively next generation sequencing method and 100% indigenous efforts, wherein draft genome of pigeon pea sequence was decoded in 2011, and this genome we sequenced Asha variety of pigeon pea having 858 million base pair genome size and predicted 47,000 genes. So if you see globally now, more than 317 plant genome have already been sequenced, and the whole database is available in public domain, which can be used in various purposes. And one of the methods of application of genome sequence of any organism is gene mapping. And for mapping a gene, first of all, one has to identify a gene and then clone that gene. For a typical example I am showing here is a fungal pathogen rice blast, for which we clone a gene. So why we chose this particular organism in our lab for identification of gene is that this is a disease which is prevalent throughout the world, and in India from Assam to Gujarat and from Kashmir to Kanyakumari, wherever rice is grown, this disease is prevalent. And we have estimated that around 68 crore rupees are being spent on basmati growing areas in northwestern plain zones for the management of this particular disease. Therefore, the effective management of rice blast is host resistance, for which identification of gene resistance gene is very, very important. And this is the work which we started in 1996, where we started identification of resistance gene against the race flora of northwestern Himalayan region. And we identified a line, rice line TTIP, which is resistant to more than 119 isolates of Magnaporthia oryzae fungal strain. You can see the phenotype of TTIP leaf is completely free from pathogen infection. Whereas when we want to map this gene, we need another host which is highly susceptible. So two contrasting hosts are required, genotypes are required to develop a mapping population by crossing these. And in 1999, we crossed these two lines and then developed a mapping population. And when we phenotype, we showed very clearly here, you can see that 3:1 ratio we got here, which typically tells us that the resistance response controlled in TTIP is because of a single dominant gene. Then by using different DNA markers, this gene was mapped at long arm of chromosome number 11 of rice by using a DNA markers at 2.1 centimorgan distance. But to find map this particular gene, we used different types of approaches. And this is a typical linkage map of gene, which is PI54, which is flanked by two markers, RM206 at 0.7 and RM224 at 10.7 centimorgan distance. So to find this particular gene within the rice genome at chromosome number 11, we need to find map it. For that, we use the genome sequence of rice chromosome number 11 and identified around 40 new DNA markers and use all those DNA markers for genotyping of our mapping population. And then finally, we got only two markers, TRS26 and TRS33 at 0.7 and 0.5 centimorgan distance, which is flanking to the gene of interest. But physically, this distance comes around 142 kilobases. So to identify one gene in 142 kilobase genome is again a very difficult task. Therefore, we used bioinformatics and computational methods to identify how many genes are there in this particular 142 kV region of chromosome number 11. We identified 18 genes and annotated all those 18 genes, and finally we found there was only one candidate gene which was present here, size of 1.5 kilobases within this particular region here. So computationally, sequence analysis was used to identify this particular gene, but to actually clone this particular gene, we designed PCR primers flanking to this particular gene and then amplified this region from both resistant and susceptible line, sequenced that, and we clearly found that this is the gene structure from the TTIP line, which has open reading frame and also a promoter sequence. But when we compared this particular allele with the susceptible allele of Nippon, where you can see that there are three different insertions in the susceptible allele, and we concluded from here that the susceptibility of Nippon bear sequence is because of insertions of this particular region in open reading frame, and gene become disrupted. Then by using computational method, different domains of genes were identified, and a typical horseshoe shaped structure of three-dimensional protein structure was developed, which gives us an idea that this is a typical resistance-like gene. But unless you transfer this gene from in a susceptible line, one cannot validate its function. So to validate its function, this particular gene was cloned with its native promoter in a plant transformation vector here, and a type I309 blast susceptible rice line was used for transformation by using this particular method here. You can see from cell culture to full grown plants, and by using molecular analysis, we showed that these transgenic lines have the gene within these. Also, southern analysis shows that these lines has one or two copies of the gene, and also PCR analysis tells us that the gene is expressing in these transgenic lines. But actual test of this particular gene came when we inoculated all these transgenic lines with virulent strain of Magnaporthe oryzae, fungal strain, rice blast fungal strain. And you can see this is the transgenic line here completely free from rice blast, whereas the susceptible lines has infection. It tells us that the gene which clone is functionally active. Later on, by using RNAi technique, we again further validated this gene and knocked it down in these resistant lines, and these lines became susceptible. Therefore, functionally, this gene was validated and also looked at its phenotype for different strains of the fungus. Starting from Palampur, that is northwestern Himalayan region, it is comparting resistant. Then 2 strains from Basmati growing region, again resistant. And eastern India from Hazaribagh, we collected another strain. And this particular gene was effective to all the strains of Magnaporthe oryzae, which we cloned here. So then what is the use of cloning of this particular gene and developing markers? It can be used in marker-assisted selection, and this is the example I am showing here. Marker-assisted selection, how it is performed. We cannot see the gene, we can only see the markers linked to the gene of interest here. And then when this is a typical scheme here in which we have shown that a resistant line TTF, as I told you, this was identified as a resistant donor, crossed with Basmati 1, Pusa Basmati 1, a susceptible lines, and then using DNA linked markers, TRS26 and TRS33, and also gene linked, gene specific markers here. These markers were used for marker assisted selection. And I would like to tell you simply that when you do PCR in segregating lines, wherever this gene is present, you will find a DNA band. But wherever this gene is absent, you don't find a band when you do PCR of these transgenic, segregating lines where we use marker assisted selection. And after developing these lines were evaluated across the locations to look at its agronomical performance and reaction to rice blast. And then finally, our collaborator at IARI, they released a variety Pusa 1612 mass-derived blast-resistant variety, and it was released by CVRC for cultivation in Punjab, Haryana, Delhi, and Jammu and Kashmir. You can see the phenotype of grains as well as plant phenotype. Similarly, another variety Pusa 1609 resistant to blast, which also contains PI54 and PAC, was released for Uttar Pradesh and National Capital Region of Delhi, Uttarakhand, Punjab, and Haryana in 2016 by our collaborator at IARI New Delhi. Similarly, we released a variety for Himachal Pradesh and Uttarakhand which contains PI54 and PITA, and this variety you can see grown in farmers' field and have high yield. HPR 2880 was released by State Varietal Release Committee by using this particular gene. So if you see here by using marker assisted selection and ICAR, different institutes have released more than 48 varieties, and many of them are in rice, 31 varieties, wheat 5, maize 8, and like that, are different traits like drought, blight and blast resistance, leaf and rust resistance, quality traits, and then downy mildew resistance. So therefore, and all these varieties are being now used for cultivation in the country. The last approach which I'm going to discuss here is genome editing, and it's a recent approach. You know, this year both the developer of this technique, they got Nobel Prize, and it's a very precise technique wherein one can change a typical DNA fragments, a single base pair change can be made to look at its phenotype, and one can it has already been used in many crop varieties. If you see here, started in 2012, and blight resistant rice was developed, then herbicide resistance canola, and you can see that throughout years up to 2022, there are very successful examples, and one of the successful examples which has already been commercialized is non-browning of mushroom, which is a successful example of genome edited crop plants. And I think in future this is going to become a method of choice for modification of different traits of plants. So not only we need food security, we also need nutritional security in the country, because you know that the world over more than 2 billion people lack key micronutrients like vitamin A and iron, and 2 billion adults are either overweight or obese. And situation is very bad in India. If you see, this is the Indian status here, and 40% of our children below the age of 5 years suffers from either protein malnourishment or having stunted growth. Therefore, biofortification is a very important or sustainable way to eliminate malnutrition in the population, because here biofortified crop, they become part of food matrix, highly bioavailable, cost effective and sustainable, and reaches target people in pure form. Particularly if you are developing, say, rice biofortified with zinc and iron, one can easily spread this particular technology through the disadvantages people through our public distribution system. So biofortification for protein, you know that there is a protein revolution during these past few years. In 2016-17, we were net importer of these pulses cross, but after 16-17, there was a complete big jump in pulse production in country, and we are now having more than 23 to 24 million metric ton pulses which we are producing in the country, because of releasing of high yielding varieties, which we have more than 900 varieties which we have released till date. A lot of work is going on its improvement in the country. Similarly, in case of cauliflower, we also developed at IRI New Delhi Pusa Kesari vitamin A rich, what you call this cauliflower, which contains beta carotene gene. By using marker assisted selection, we developed this particular variety IRI. And in India now, ICAR along with various universities, we have released 53 varieties which are rich in iron, zinc, protein, lysine, vitamin A, and anthocyanin, and many other micronutrients. And recently, even besides these 53, 17 more varieties have already been dedicated by our Honorable Prime Minister to the country only this month. And in 2014, if you see this particular graph, there was only one biofortified variety in the country. Now we have more than 52 in 2019 and 71 now in 2020. Therefore, these all biotechnological approaches are being used in the country for the improvement of crop plants. And I would like to sum up here with my presentation by saying that many successful examples have shown that new plant breeding approaches, particularly biotechnological approaches, can play an important role in developing crop varieties for important traits like insect resistance, disease resistance, and nutritional traits, etc., to achieve food and nutritional security in the country. And I'm sure that the basic science which we are going to discuss in this particular conference during these 4 days will definitely tell you all these examples which I have shown you here. And these are the only successful example which I showed during my presentations, but a lot of basic work in terms of development of new techniques, utilization of different methods in basic molecular biology have gone in this particular work. And that's why you can see the products in different crops. So with these words, I would like to thank you all, and best of luck for your conference. And I'm sure that with the deliberation which is listed for these 4 or 5 days would be very fruitful to everyone sitting here, and also particularly to the young student and the scientist who can be benefited from this conference. So thank you very much.
Ladies and gentlemen, I would like to thank Dr. T.R. Sharma, Deputy Director General, Crop Science, ICR New Delhi. Sir has given a very thought-provoking address. Indeed, it has set a platform, perfect platform for our speakers and many of the scientists to present their area of this research. Sir has covered two different aspects, food security and food nutritional security. In food security has covered all aspects starting from this plant tissue culture and its applications in the field of field crops and horticulture crops. He has touched this genetic engineering also and its applications, and next he has covered plant genomics, wherein he has told NGS, DNA sequence analysis, genome decoding, gene mapping, genetic and physical map of PI54 gene, functional validation of that gene, genetic transformation of blast resistant gene, followed by southern analysis confirmation. And later he has transferred those genes into the rice, Pusa basmati rice, using this masker assisted selection breeding. Next, he has touched upon this genome editing also. And nutritional aspects, Sir has covered this biofortification and many other aspects. I would like to thank once again our Honorable Deputy Director General, Dr. T.R. Sharma, sir. Thanks a lot, sir. Thank you very much, sir. Ladies and gentlemen, may I now request Dr. M.R. Dinesh, Director, Indian Institute of Horticultural Research, Hesarghetta, Bengaluru, to share the knowledge of horticulture and biotechnology to our delegates and participants.
Thank you, Dr. Halesh. Am I audible?
Yes, sir.
Yeah, thank you. Thank you very much. Respected Deputy Director General of Crop Science, ICR, Dr. T.R. Sharma ji, ex-Vice Chancellor of UHS Bagalkot, in fact, Founder Vice Chancellor, Dr. Dandin, Dr. Indiresh, my very good friend, ladies and gentlemen. In fact, having been associated with perennial crop breeding, it was very refreshing to hear Dr. T.R. Sharma cover virtually every aspect of it with regard to biotechnology as a tool in crop breeding. I think I have only very few issues I would like to flag it at this point of time. One is, generally it is assumed that perennial crops being highly heterozygous, all through we have been telling that it would take a long time for us to develop varieties. In fact, the perennial crop breeding added to the heterozygosity is the problem of rootstock and scion. Whenever we talk about abiotic stresses, we need to go in for rootstocks, and with regard to scion breeding, we need to have a different approach. These are the two problems that we generally encounter in perennial crop breeding. Having said that, now with the tool of biotechnology, in the very, very recent past, in fact, the marker-assisted selections, which Dr. Sharma ji has very aptly described, I think this is one tool which can go a long way in helping the perennial crop breeders. Why I say so is because at the nursery level, probably if you can screen the progenies and get rid of those ones which are not useful, and then carry forward those progenies which are of importance, yes, we can do a great job. I, at this point of time, also would like to say that there is a glimmer of hope with regards to transgenics in perennial crop breeding. I say so because the rootstocks, which in no way contribute to the edibility of the crop that we are growing, probably can be used through a lot of improvement can be made through transgenics with regard to development of new rootstocks. Although the rootstocks are also need to be propagated clonally because of the heterozygosity, still in few of the crops, in fact, the days are such that even in the what we see in the Southeast nations with regard to vegetables, rootstocks have been now at our institute, we have developed interspecific hybrid rootstocks for tomato to tie over the problem of excess moisture as well as drought. Even we have developed rootstocks for watermelon. We have developed rootstocks for chilies. So rootstocks are a field wherein we can definitely use our transgenics to come out with rootstocks for various abiotic and biotic stresses, soil related problems. This is one thing which we need to flag and see how best we can actually take it up. In fact, I would request Dr. T.R.S. Sharma, if a networking project of rootstock improvement, both in vegetables as well as in other agricultural crops can be, I don't know which are the agricultural crops which can go with the rootstocks. I'm not very sure. With the horticulture, yes, we need rootstocks, and rootstock breeding is of utmost important, which has happened in many of the species in fruit crops like citrus in various, especially in the Hawaii. Nowadays, in fact, even in a crop like papaya, commercial cultivation of papaya is being taken on rootstocks. I think there is a need to do this. When I mention about papaya, there are two problems associated with papaya breeding. One is the papaya ring spot virus, wherein we have the resistant source in the wild species, which is very difficult to cross. We have found some success. However, my emphasis here is I would request many of the scientists working on biotechnology to start with cisgenics. I'm not very sure. I think Dr. Sharmaji should guide me. I'm not very clear as to how many cisgenics projects we have in the country today, and how cisgenics can be definitely a tool in improvement. Already it has been done in potato. I think we should divert our attention to a certain extent to cisgenics and convince the policymakers to say that cisgenics is not transgenics. At least they would agree to that extent. And definitely, I think we can make some amends in this direction, and we can have a lot of projects on cisgenics. Especially, I am thinking of in the institute now to have projects on cisgenics in some of the fruit crops. Sir, you mentioned about CRISPR-Cas. I think this is one of the most important and revolutionary tool. In fact, at the institute, we have formed a group of scientists who are actually working on papaya ring spot virus with the CRISPR-Cas tool, CRISPR-Cas9, in fact. So we have met with some success. Wish and hope that in a year's time, probably we would definitely come out with a genotype which would be resistant to papaya ring spot virus with our in the Arca Prabhat background, which is a gynodioecious variety of papaya which doesn't have male plants. Another thing which comes to my mind is probably in the horticultural crops, somewhere down the line, especially when I say horticultural crops, I'm more concerned about fruit crops, a little bit biased for fruit crops, precisely because that has been my area of interest. Not many association studies have taken place. In fact, if we need to identify the trait associated genes or genomic regions, I personally believe that we should have association studies in many of these fruit crops. I think this is of utmost important. I have asked our scientists who are working on these aspects to start. In fact, we have started in few crops. Let us see how best we can cope up with that. Then another very important, I think Dr. Sharmaji also mentioned, whenever we, to avoid this linkage drag, in fact, in the guava breeding program, we have been trying to develop varieties with pink pulp and soft seeds. These two characters are very tightly linked characters. That is why I said to avoid this linkage drag, probably tools like biotechnology tools like cisgenics may be of some importance to us. Of course, we have raised a lot of progenies, and then we have tried to break this linkage. We are not very, I must say that we are not very successful. Probably this is one debatable issue which we can discuss in this conference. Another very important thing which comes to my theme, which comes to my mind, is that of tissue culture. I will quote only one example to you, sir, to all the delegates. Probably you can ponder over this. In fact, in the papaya ring spot virus resistance breeding program, what has happened is, when we have found some success with the interspecific, now of course it is called intergeneric because this Vescancella is a different genre altogether. And when Visconzella cauliflower, we have crossed it with Carica papaya. It's an intergeneric hybrid. In the F1 generation, we do get hybrids with the use of sucrose on the pollen. We have been able to break the intergeneric barrier to a certain extent. We have not been able to get the number of progenies that we would like to have. However, we have been partially successful. In that direction, in the F1 progenies, we were able to really get good resistance. But when we take up further back crossing or further crossing or further sigmating to purify the line, what happens is that generally we tend to improve the quality but lose the resistance. This is natural for any of the interspecific hybrid crosses. I think any of the breeders would know that this generally happens. And this interspecific hybridization or intergeneric hybridization on many occasions are coupled with lot of sterility in the progenies. So one of the most important aspects which we need to work on is tissue culture. Why I say so is because in the F1 generation in crops which are essentially vegetatively propagated, wherein we are able to fix the characters, if we can really clonally multiply them. In fact, in papaya, cutting propagation has also been quite successful in China and Brazil. But we need to standardize to our conditions, to our varieties. Probably if we can really take up tissue culture cultivation, tissue culture propagation and standardize it, I think this would go a long way in promoting development of disease-resistant varieties in crops like papaya. I think these are the few points I wanted to share with the delegates. I think during the period of another 4 days, I think Dr. Fakhruddin and his colleagues would definitely pay attention to some of these things. And I, on behalf of my institute, we are very much open for any of the collaborative studies in this regard. Thank you very much.
Ladies and gentlemen, I would like to thank Dr. M.R. Dinesh. He has given a very elaborate lecture on horticulture crops. He has covered this perennial, he has told some issues of perennial crops breeding, transgenics in fruit crops, rootstocks improvement, cisgenics, CRISPR-Cas, tissue culture, and this papaya ring spot virus problem also. Definitely, it would be incomplete if I don't tell the contribution of Dr. Dinesh sir. So Dr. Dinesh sir has contributed in the development of the varieties Arka Surya and then Arka Prabhat in papaya. In case of mango, Arka Udaya, and also in case of Gawa, Arka Mrudula, and then pink pulp dual purpose variety of this Arka Kiran. So once again, I would like to thank Dr. M.R. Dinesh sir, IHR Director, Esrugata, Bangalore. Thank you very much, sir. Now, ladies and gentlemen, may I now request Dr. S.P. Dandin sir, Founder, Vice Chancellor of University of Horticultural Sciences, Bagalkot, and former liaison officer and consultant of Biotechnology International, to share the knowledge of biotechnology and horticulture in horticulture crops. Dr. Dandin sir, please.
Thank you, Dr. Halesh. Am I audible to all?
Yes, sir. Yes, sir. Audible, sir.
Yes, sir.
Okay, fine. Good morning to one and all. At the very outset, I thank the organizers for this excellent forum created and a platform to share the new thoughts regarding the avenues, possibilities, and the problem-solving aspects of the biotechnology role in a modern science breeding. Now, Dr. T.R. Sharma, the DDG Horticulture, so the Crop Science, Dr. Dinesh, and the Vice Chancellor, Dr. Indrish, Dr. Fakhruddin, a brain behind the organization. I thank you all once again. And Dr. Sharma has thrown elaborate light on various possibilities, then the milestones achieved, and the way forward for the role of biotechnology in solving the problem of humanity by and large. Friends, as you know, the world is facing three important challenges today. For these, probably the biotechnological tools and precision technologies will be able to help us. The first challenge would be the loss of resources, including the biodiversity, where our land races, crop oil relatives, and large number of plants species are fastly disappearing from the cultivation scene, maybe due to the monoculture or due to the other aspects that one needs to take into account. How best the most important source of genes from crop oil relatives, land races could be restored through gene conservation and DNA conservation. The second aspect, which is more also important, is the climate change challenges. Now it is a reality. Everywhere we are facing the effect of climate change in one or the other corner of the world. It is a day-to-day affair. It may be a flood, it may be a cyclone, it may be a long dry spell, and also the occurrence of newer pests and diseases, which is a threat to the mankind and all the organisms by and large for this purpose. That is another challenge. We have to look into it. And third challenge, perhaps Dr. Sharma has made very clear, that how to address the nutrition and food insecurity, which is becoming a more and more major problem. As a result, non-communicable diseases like diabetes, the cardiac aspects, and so many other things are surfaced in a large scale. And the only way to address this issue is the food security and nutrition security. And thanks for his last aspect of the biotechnology, where the biofortification of food could be an answer. Definitely it is yes. So that a food can be made involving all the important minerals, the vitamins, and all other things into in a one crop, like golden rice or a good carrot or some other crops, so that the one crop can meet majority of the requirements, but it cannot be a panacea, but to some extent it can be a good thing. So that I'm basically for working on a classical plant breeding and genetics for last about 3 decades or even more. I recall the theory of jumping genes of Martin Cook, who has neglected for quite some time, and now it is being considered the capacity of a gene to transfer from one loci to the other loci, or to get itself changed, or to get itself transformed. These are the things. Then the another milestone, Dr. Sharma mentioned, the Norin wheat gene and also the gene Digen Huze. These two genes, one in wheat, one in rice, have revolutionized the whole food and nutrition, food security rather than nutrition, I should, food security of the whole world. So now it's the time, based on these lessons, what we have learned, we would be able to, through the tools and present technology of biotechnology, will it be possible to generate some new things based on our needs and requirements of the society. We have seen the development of biotechnology as one of the most strong and effective tool to bring the change what the society needs in terms of food security, nutrition security, the challenges being posed by the climate change, as well as the new pests and diseases coming by in a large scale, what we should look into. We have a strength, no doubt, and this strength could be properly used for the benefit of. But however, if you see the world scenario of the biotechnological aspects of newer crops, the biosafety aspect, as well as the future line of thinking, the policies vary from country to country, from one group of scientists to another group of scientists. This is perhaps making us many people to start thinking, well, is biotechnologically improved crops or the GMOs could be a solution without any effect. I have one question to ask the entire scientific population and society in large. When we are depending so much on medical biotechnology to solve our health problems, the food problem is more important than medical problems, because it gives us a totally strong, the immune built health, which is through food only. So food has to be made as a medicine rather than for everything depending on medicine. When our biotechnologists are thinking very hard, working very hard to generate a food which is more nutritious, more useful for the health, why we are doubting? This is mainly because of three reasons. The poor understanding of the biotechnological tools and technologies and its effect by the non-biotechnologists which are go on raising the problem. This is number one. Number two, the difference of opinion being expressed by different countries, developed and underdeveloped, and also the technologically advanced and technology poor source. This is another thing. And thirdly, partially we have to be scientists have to be responsible in starting and pushing the things hurriedly rather than making them foolproof to answer all the ifs and buts of the society. Keeping these things, friends, probably is a time for all the technologies, especially the molecular biologists, the geneticists, and the biotechnological scientists to have a common platform to push our products to meet the requirement. Once the question was raised on BT code, Dr. Sharma was very much effective. He concluded that if this being the case, why not others? Friends, finally, as a student, I started my work on apomictic breeding and transfer the gene of apomixis to the cereals. That dream is still remained a dream. I think this is one of the challenges for the biotechnological scientist, whether we can throw more light on transfer of the gene which is responsible for apomixis in some of the wild grasses, some of the forage grasses to the cereals like wheat, maize, and rice. This is one area where many aspects are considered. My other challenge, these are the classical questions being asked time and again, whether the transfer of the gene which is available in pulse to make the crops to some extent nitrogen self-sufficient. It is the nitrogen fixing gene to the non-pulse crops. These are the questions of the very tall order. But yet, how best we can throw some lights from one corner of the all to the other to make this particular gene which is available in pulses could be transferred to the cereals where the hunger of the humanity to some extent could be resolved. The third aspect, again, these are the questions which are being asked again. Whenever I used to meet Dr. Fakhruddin, these three questions I invariably ask to all the scientists. Third one is, we have two types of plants, C3 and C4. Is there any possibility to have a kind of a transformation of the more carbon sequestration efficient gene to other crops? I know these are the questions which are very easy to raise, but it takes perhaps decades, if not centuries, because of the strong tools available with our situation. Friends, we know that dwindling resources, especially the water, the soil, and the biodiversity, all the three which sustain life on Earth, but seldom we have not been able to add more knowledge on the role of microbes. Probably Dr. Krishna Kumar, who will be able to join, later throw some light on disease and resistance. But if we have the microbes, immediate to our mind comes the problem of pests and diseases, which are by and large caused by the microbians. But microbes have another role to play, because if the T plasmid gene has not been transferred to the bacterium, probably a whole of our research will not be there. So if role of microbes in augmenting the soil nutrition aspects and also to antagonize some of the microbes which are disease causing could be another area. Well, Dr. Dinesh has raised one very important point, that how best the perennial crops like horticulture crops, be a papaya, be a mango, be a sapota, and all those things, what are the next steps? We know the perennial crops have their own problems to be solved immediately, but one aspect is rootstock and the scion combination. The other one could be, to me, the biofortified crops coupled with the induction of the vegetative propagation. I will give a classical example. You know, the drumstick is the God-given gift to the humanity. It's a capsule of minerals, vitamins, and for our nutrition insecurity, such crops are one of this one. But unfortunately, so far, I have been requesting scientists, is it possible to develop a protocol and effective management of tissue culture in all these things? Friends, we have a large number of people working on tomatoes, potatoes, rice and maize. Now our diversion of the biotechnology scientists to take up crops which are really holding promise to the next generation in terms of resistance, in terms of food security, in terms of nutrition security should also be considered. Because if these crops are forgotten, we'll forget them forever. So my request for the scientists could be, for example, cluster bean, he was one heard, but now it is coming. For example, large amount of vegetables, which are also the most of the horticultural crops are having one or the other medicinal properties. How best we can explore possibilities of understanding such crops and to be taken a problem associated with either multiplication or improvement or resistant breeding in the horticulture crops is most important. Friends, horticulture, you know, it is now in a driver's seat, which is taking the food and nutrition aspect much ahead of the cereals, pulses and oilseed crops. No doubt, we require protein, we require carbohydrates. They alone cannot subsidize our nutrition plate. It has to be widened with variety of crops, especially the vegetables and fruits, which are the source. So the scientists who are attending and participating and also will be the partners could also diversify their research activities to some of such crops who have a large amount, who holds the promise and which can be the answer. And I'm sure the large gathering today, as of now, it is 364 participants are listening. And Dr. Fakhruddin told about more than 600 have been registered. Probably this, at the end of this conference, we should be able to crystallize our roadmap, which are the crops, what are the problems. Because now this is not an age of ad hocism or omnibus approach. We should be very, as biotechnological tools are very precise and very direct oriented, our approaches to use biotechnology should be also like that. Hope this conference will be able to draw a clear roadmap for the newer crops, newer challenges, so that by and large, the population of tomorrow will be disease-free with a good of health and immunity built to make their own survival. I thank all the organizers for having given me this opportunity and also to hear some of the very important eminent speakers in the next 3-4 days so that we can enrich our knowledge, horizon of thinking, and also new approaches. Thank you one and all. And I thank once again the organizers for this fine opportunity. Thank you. And I wish the conference will be more meaningful, effective, and successful in throwing some more light on newer ideas, newer crops, and newer challenges. Thank you.
Ladies and gentlemen, I would like to thank our former and founder Vice Chancellor, Dr. S.P. Dandin sir, as he has given a holistic approach on horticulture and biotechnology and traditional breeding. His view was biotechnological interventions and precision farming has a great role in the development of horticulture. Still, he has touched upon many areas. Now, friends, now I would like to, ladies and gentlemen, may I now request Honorable Vice Chancellor, Dr. K. M. Indresh sir, to kindly deliver the presidential remarks. Friends, I would like to inform you that our Honorable Vice Chancellor has involved in perennial crop breeding, that is in coconut. He has released one Kalpataru variety, and also he was involved in the development of this potato hybrid JX576. Now I request our Honorable Vice Chancellor to give presidential remarks, sir.
Thank you, Dr. Halesh. Good morning to everybody who are participating in this International League Conference. Honorable Deputy Director General Crop Sciences, Dr. T.R. Sharma-ji, Dr. M.R. Dinesh, my good friend and Director, IIHR, Bangalore, Dr. S.P. Dandin, former Vice Chancellor of, former Founder Vice Chancellor of UHS Bagalkot, and also the former Liaison Officer of the Biodiversity International, my officers, colleagues from the headquarters, and my own deans from the different constant colleges, and the participants of this today's e-conference from both India as well as across the globe, students, especially the postgraduate students who are participating in this e-conference, and the vice chancellor from other agriculture universities from Karnataka and also from other states who are participating in this, and the delegates and the organizing committee of this conference headed by Dr. Jagadish, who is the chairman of this organizing committee, and Dr. B. Patruddin, organizing secretary, and the co-organizing secretaries Dr. Halesh, Dr. Shivpriya, Lakshmi Devamma, and Mohan Kumar, and all my faculty members from the university, from the discipline of biotechnology and crop improvement and other departments, and my own colleagues from the College of Horticulture, GKVK. I am very happy that my colleagues from the College of Horticulture, GKVK, have organized an international conference on advances and future outlook in biotechnology and crop improvement for sustainable productivity. This is the second international conference being organized from College of Horticulture, GKVK. The first was on plant disease management by the Department of Plant Pathology organized a few days back, and this is the second conference is being organized under the leadership of Dr. Patruddin and his team from College of Horticulture, GKVK, and Dr. Tilakraj Sharmaji has already highlighted the role of biotechnological tools. Role of biotechnological tools in the development of new varieties, new hybrids, and also for its role in the enhancing the agricultural production in the country to feed the population, overwhelming population, and also the importance of biological tools in overcoming the biotic and abiotic stresses in some of the crops. Also, Dr. Dinesh has emphasized the role of cisgenics, development of cisgenics in some of the perennial fruit crops like papaya and other perennial fruit crops, and the role of transgenics nowadays. So, looking into all these things, the conventional breeding methods supplemented with these new generation technologies, especially the biomolecular tools, is definitely going to help in overcoming some of the very serious problems faced by the crop breeders. The source of variability, if it is lacking in some of the wild species, can be created by DNA sequencing that can be used in the breeding programs. So, by creating genetic variability and also for the conserving the very valuable genetic resources for the future use is also equally important. We can definitely protect these genes or the germplasm through use of molecular tools, maybe tissue culture or cryogenic storage, long-term storage methods can be. So there is a lot of potential for the biotechnology and crop improvement in days to come, and this international league conference is definitely going to open new tools, new ideas, new innovative ideas for the delegates who are participating in this e-conference. I heard that nearly 680 abstracts have been received and they have been published, and nearly 1,400 participants are participating in this conference. I am happy that they have brought out the compendium of the abstracts in the form of booklet, and it's a very good development, and that's the team from College of Horticulture. They have taken a lot of interest, and university has provided whatever the facilities available to the and the university, from the university side also we have some facilities for work by my faculty members in the university. We have the separate department of biotechnology, and some projects, some of the scientists are operating some projects, especially development of bacterial blight resistant lines in pomegranate at Bagalkot. And in fact, Dr. Prakuddin is also working on development of transgenics in tomatoes, soybean, and also jack. Also, they have made a collections. In fact, the Garcinia species, they have made an excellent collection of germplasm. And recently, the biodiversity park, which was established by the Biodiversity International, by the biodiversity liaison officer Dr. Dandin, who was responsible for this collection of very good collection of underutilized and minor fruit crops, has been made, and the park has been established. Now it has been handed over to our university. This is a very valuable source of genetic resources, especially for the perennial fruit crops. So this is also being maintained by the university at GKVK, and the Dr. Jagadish, Dean, College of Horticulture, and Dr. J.S.K. Swami, Professor in the Department of Food Science, are involved in maintaining this biodiversity park. And definitely the university is going to give more emphasis on research aspect of biotechnological research in horticulture crops. And in today's program, this conference is being structured with 10 thematic areas covering major areas of research in biotechnology and crop improvement. Participants are allowed to submit abstracts and conduct presentations. The conference aims at bridging different spheres through information sharing and networking. It provides a leading opportunity to reach out to the largest assemblages of participants, and this conference helps to bring together scientists, students, industrialists all over the world, and provides a platform to promote and encourage research and education in all branches of biotechnology and crop improvement, and to facilitate the cooperation and knowledge transfer on a global scale. These 4 days of e-conference on advances in biotechnology and crop improvement, organized by the UHS Bagalkot, is going to be very fruitful. I wish new ideas, new priority areas, and new tools of biotechnology will going to emerge as an outcome of this international conference, and hope these 4 days of e-conference is going to be successful. I wish all the best to the organizers of this e-conference and all the delegates to actively participate and interact with each other and exchange the ideas about their experience in their own field of specialization. I wish a grand success of this conference. Thank you all. Dr. Alish?
It's not audible. Is it audible to all people?
You unmute, madam.
It's audible.
Yeah.
Audible?
One connection.
Okay.
Okay. Let us wait for connection.
Ladies and gentlemen, I would like to thank Dr. K.M. Indresh sir, our Honorable Vice Chancellor, as he has given very wonderful and excellent presidential remarks. Now I would like to give vote of thanks. With all its busy schedule, our Honorable DDG Crop Science, Dr. T.R. Sharma, has agreed to become the chief guest of this function. I would like to thank on behalf of COH Bengaluru and UHS Bagalkot, Dr. T.R. Sharma sir. And also I would like to thank Dr. M.R. Dinesh sir, ICR Bengaluru, for his presence on for this webinar. I would like to thank Dr. S.P. Dandin sir, former vice, and also I would like to thank all the officers of the university, Dr. T.B. Alloli, Registrar UHS Bagalkot, Dr. D.R. Patil, Director of Research, UHS Bagalkot, Dr. Y.K. Kotikal, Director of Extension, UHS Bagalkot, Dr. M.S. Kulkarni, Dean PGS, UHS Bagalkot, Dr. S.I. Athani, Dean Students Welfare, Dr. B. Raju, former DOE of UHS Bagalkot, all the deans of different colleges of horticulture, UHS Bagalkot, all the participants, all the delegates, all our UHS Bagalkot staff, teaching and non-teaching staff who have supported for this international webinar. Once again, I would like to thank all the staff, and also I would like to thank rapporteurs Dr. Marti Prasad and Dr. Amrita Bhatt for cooperating this webinar.
Next. ಇಲ್ಲೆಲ್ಲ ಆಫೀಸಸ್ ಇಲ್ಲೇ ಇದ್ದಾರೆ. ಅದು ನಿನ್ನೆ ಒಂದು ನಾಮಿಕ್ಲೇಚರ್ ಬಗ್ಗೆ ಹೇಳ್ತಿದ್ರು. ಅದೇ ಸ್ಟೂಡೆಂಟ್ಸ್ ಕರ್ಸಿದ್ರೆ ವಿ ಕ್ಯಾನ್ ಕಂಟಿನ್ಯೂ ಡಿಸ್ಕಷನ್. ಕರ್ಸ್ಬಿಟ್ರೆ ಮತ್ತೆ ಎಲ್ಲನೂ ಮತ್ತೆ ಇದು ಮಾಡಿದ್ರೆ ಈಗ ನಾವು ಕರ್ಸಿ ಹೇಳಿ ನಾವು ಡಿಸ್ಕಸ್ ಹೇಳಿ.
ಕಳಿಸಿದ್ರ ಇಷ್ಟಾಕಿದ್ರೆ ಕಳಿಸಿಲ್ಲ ಕತ್ತಲಾಜಿದ್ರೆ ಕಳಿಸಿದ್ದೆ. ಯಾಕೆ ಬರಲ್ಲ ಸೌಂಡ್ ಇಲ್ಲಲ್ಲ, ನಿಂತೇನು ಸೌಂಡ್?
A very good morning to one and all present over here.
ಲಕ್ಷ್ಮೀದೇವ ಮಾತಾಡ್ತಾರೆ. ಮೈಕಲ್ಲಿ ಮಾತಾಡ್ದಂಗೆ ಕೇಳಿಸುತ್ತೆ.
ಹೌದು. A very good morning to one and all to present over here on this virtual platform. A warm welcome to all for this international e-conference, which is organized by the Department of Biotechnology and Crop Improvement, College of Horticulture, Bangalore, on advances and future outlook in biotechnology and crop improvement for sustainable productivity. So this conference has been structured with 10 different themes, which facilitate the scientists, students across the world to share their knowledge, their research outcomes, facilitating the entire community. So we'll begin the technical session. A warm welcome to all the dignitary participants for the technical session. So the first technical session is plant genetic resource management and utilization. For this, the chairman of this session is Dr. R.S. Kulkarni sir, former director of extension and former professor and head genetics and plant breeding. And co-chairman of this session is Dr. M.V. Dhananjaya sir, principal scientist, ICR IHR Bangalore. And in this session, we have one keynote address and four invited talks. Keynote address by Dr. Kuldeep Singh sir, Director, NBPGR New Delhi, and we have invited talk by Dr. Vania Azevedo, Head of Gene Bank, ICRISAT, and Dr. N. Krishnakumar, former DDG ICR New Delhi, Dr. P. Rajshekaran Sir, ICR IHR Bangalore, and Dr. Venuprasad Ramaya from Kirri, Philippines. So now I request Dr. R.S. Kulkarni Sir to chair the session, this team session. I request Dr. R.S. Kulkarni Sir. So I would like to introduce Dr. R.S. Kulkarni Sir, our beloved professor, who is professor for most of our faculty here, who is presently working as advisor Ankur C, and he is former director of extension US GKVK Bangalore, and he has served in his various capacities in the university for 39 years, and he was our professor and head for 12 years, and he has guided so far 16 PhD students and 30 MSc students. And he is instrumental in developing 10 varieties in tomato, pigeon pea, rice, and cowpea. And he has handled so many outside and university funded projects, and he has served in various capacities in the university. Sir, I welcome you, sir, to chair the session, and I request to chair the session. Sir, please.
Open it, sound is coming.
ಅಲ್ಲಿ ಬ್ಲೂಟೂತ್ ತೆಗೆದ್ಬಿಟ್ಟು ಓಪನ್ ಇಟ್ಬಿಡಿ. ಬ್ಲೂಟೂತ್ ಇದು ಓಪನ್ ಇಟ್ಬಿಡಿ, ಹಂಗೆ ಲೌಡ್ ಆಗಿ ಬರುತ್ತೆ.
ಲಕ್ಷ್ಮೀ ಜಯಮ್ಮ ಅಂತ ಹೆಸರಿಲ್ಲ, ಲಕ್ಷ್ಮೀಶ ಅಲ್ಲ, ಇಲ್ಲಿ ಬರುತ್ತಲ್ಲ ಹೆಂಗ್ ಮೇಡಮ್ ಇದು ಅಷ್ಟು ನೆಕ್ಸ್ಟ್ ಈಗ ಅದೇ ಬರುತ್ತೆ.
ಇಲ್ಲಿ ಇದ್ರಿ ಸರ್, ನೀವು ಕಾಣಿಸ್ತಾ ಇದ್ರಿ, ನೀವು ಸ್ಟಾರ್ಟ್ ಮಾಡಿ.
सुमेरपुर में Hello, welcome everyone. Can you all hear me? Okay.
We are hearing.
Sir. Hello, we are starting with the next session, that is on the plant genetic resources management and utilization. The main theme for this particular e-conference, and we are already late because we could not have power here. So it was a power generating resources were on line here. So therefore, I apologize on behalf of the power available here also. So I now welcome Dr. Kuldeep Singh, Director NBPGR. Sir, from Bangalore, I am sitting and I am trying to coordinate this particular session. I am very happy to go through your biodata. I, as a professor of genetics, retired 10 years back, and my contemporaries were with NBPGR, and I know about your achievements. Let us not go in for the highlighting all your achievements at this point in time, and it speaks volumes. And without wasting my everyone's time here, and with my co-chairman, Dr. M.V. Dhananjaya also, I welcome all the participants to this session. And now I keep the windows open for Kuldeep Singh, Director NBPGR, to take charge of the session.
Well, thank you. Thank you very much, Dr. Kulkarni. Am I audible?
Yes, sir. Yeah.
Okay, good. Well, thank you very much to the US Bangalore and College of Agriculture for giving me this opportunity to be with you. I see there are 370 participants here. So my time will start, Mr. Chairman, now. So this is 11:10. So I will have up to 30 minutes. That's my time, right? I see there may be a large number of the students as well. So I have planned my presentation in such a way that first 10 minutes I will give some concerns which are global and the national concerns with all of them. And then I will take them for a visit to NBPGR. I will not talk much of science to them. I will really share more information because I see there are large numbers of 3 days conference. There will be a large number of the people talking on different techniques, technologies, approach. But what's the base material, raw material where they want to work is something I will really introduce you to this one. And maybe one last slide I will give you that how is NBPGR working. There's a paradigm shift in its working now with the advancement of the technologies. And I will give 1 or 2 slides to last at the end to see you and invite you to join NBPGR's venture on that one. So I would just like to share my presentation with you. Okay, I hope my presentation is visible.
Yes, sir.
So I chose the title specifically because this is University of Horticultural Sciences, and I said we'll talk on the status and use of germplasm of horticultural crops in India. Why I chose this one? Because if we generally talk with everyone, I think the general impression is that NBPGR stresses more on field crops. But let me make it clear today. The emphasis of horticultural crops is equal, if not more, than the field crops. And hope you will realize all these things when I move my presentation, my slides to you. And the organizers, this presentation could be made available to anyone who really wants to have the details of this one, because this is all the public data which is available at NBPGR, so I'm presenting to that one. And I will tell you how NBPGR is working. NBPGR has aligned itself not only nationally, but globally as well. Of the 17 Sustainable Developmental Goals, we have identified 6 with which NBPGR is working directly. Plant genetic resources or plant improvement. We say, be it SDG number 1, no poverty, zero hunger, good health, response to consumption and production, climate action, life on land. None of these could be achieved without the thorough genetic resources utilization and management. And we are very clear on that one. Although if we see agriculture per se contributes to 11 of the 17 SDGs, but plant genetic resources and plant improvement, crop improvement directly contributes to these 6. In addition, it contributes indirectly to many others as well. This is a global picture. You see that we will be equivalent China in 2024 and maybe surpassing it 2030. And our demographics, they show that our population may stabilize somewhere at 1.66 billion in 2050. But see, along with that one, there's always a general reading it. See, to feed 1.66 billion people, we need to increase the food grain production. I haven't seen anywhere people talking, we need to increase the horticultural crops consumption as well. So this is a challenge for the horticultural students here. I wish that we really need to emphasize, and I will give you some slides there to show you before I actually take you to the visit and the PGR, that how horticulture is becoming more and more important compared to the field crops as well, because we have achieved pretty good when it comes to the field crops, but we still need a lot to do on horticultural crops. Now, why these statements have started coming is primarily because there's a complete shift in the food habits. From somewhere 7,000 species which we were using locally at some stage, let's say hundreds of years ago, now we are constantly dependent on 3 species for 60% of the calories, or to take it 30 crops which provide 90% of the plant calories to us. Right? And that's a global one. If we see it within country, of course, horticulture crops the fruit crops especially have not been considered here, but have a look. Complete shift from these crops which we were using and consuming earlier to these crops. There's a complete decline in the consumption of these crops. And this has led to several problems actually. And one of the problems, of course, I will skip this one here. One of the problems of that is that, and that's a global challenge, which India is becoming a party to it, is unequal distribution. We have 2 billion people who lack micronutrients like iron and zinc, but we have 2 billion people who are obese as well. Either way, we have, 155 million children who are stunted, but we have 41 million children who are overweight as well. So this is because of the unequal distribution, where policymakers have to play more important a role than the scientists per se themselves. But see, this is very important data which we got recently from, this is our National Family Health Survey 4 that has recorded the data from 2006 to 2016. All these ones which are in orange color, the proportion of anemic people has increased. And I will take example of Punjab, for example. From 2006 to 2016, the proportion of anemic people in Punjab has increased by 15%. Punjab is not a poor state. It has the highest productivity in the world when it comes to production of cereals. 12 tons per hectare are the grains only. Then why this has happened? I think the primary reason for that is the monoculture. Culturing only wheat and rice. 80% of the land of Punjab is under wheat-rice rotation. And that's both the food crops people do not eat, or eat to a minimum level. There was a complete shift. If there are some areas under fruits, this is more of a commercial. What was earlier farmers actually growing for their own consumption, with the commercial agriculture, those things are also going out. So monoculture is primarily responsible for this side of change. Some states have improved, others have gone down, but that's important for us. Have a look on this slide. This is very important and a challenge for the horticulture. This is the Lancet data, very important. This appeared only last year. This is India. Now the outer circle is what is the optimum consumption for a easy for a good health. You see, only cereals is something we have achieved. Look at the fruits, look at the vegetables, look at the nuts. We are very poor in that one. Our consumption is very low. And in fact, per se, that's globally as well true. If we see the global level as well here, this is a global, the vegetable, the fruit consumptions are relatively much lower than what's actually required. Now, we really need to answer a question, why this lopsidedness has happened. Is it that we do not have a purchasing power? Is it that we do not realize that vegetables, fruits, nuts are equally important as the cereals are? Or is it that the productivity level is lower in these? We haven't really improved them to the level that they could be more economical to the farmers and the consumers. It's not only farmer, the consumer has to be equally taken into consideration. We still feel, as of today, buying the cereals is relatively cheaper than buying any fruit or any vegetable. We still see our pockets whether it will permit us to purchase some fruits or not. We really need to answer this as the students of horticulture, at the national level as well. And we also need to see that all these things, despite having incurred too much in agriculture, fairly good amount of research in agriculture, we have been successful in improving cereals only, not other crops to that extent. And here's our message. We said to meet, so here I'm not taking only the energy, food only. It's a food and nutrition, in fact, including climate. We are of the firm belief that the germplasm will be key to it. And we'll show you how the germplasm is key to it. So this is, in fact, the motto on which NBPGR is working. We say for sustainable agriculture and diverse food intake, both are equally important for a healthy population. So we need a population that is healthy. And if you see the pictures here on this slide, there's no main crops, all the minor crops, which are very nutritious. I see Dr. Dandini is sitting here. He has done a wonderful work of assembling the minor fruits in US Bangalore. I have been to that facility as well. And of course, then I say the genetic resources are very important. This is a simple definition for the students that, okay, what the genetic resources are. And this is the definition with which NBPGR is working, because we work on the conservation and utilization of seed, vegetative propagules, tissue, cell, pollen, DNA molecules, anything that we feel has a potential today or for the future, and that can be used for crop improvement. And for us, plant genetic resources will include all these things, be land races, release varieties, genetic stocks, farmers varieties, transgenic plants, wild relatives, all of them, they make the plant genetic resources. And for plant genetic resources management, these are the 4 important activities for NBPGR, be it collection, characterization, conservation, and utilization. All the 4 activities are equally important, though they may go with phases, and I will tell you how we have gone this way. Now, having said that there are challenges available to us or we are facing in the 21st century, Mother Nature has gifted India a lot. We are one of the very important gene centers in the world. We are a gene-rich country. We are a center of origin for dozens of crops. We are a center of diversity for several dozens of the crops. We are maintaining a large number of the breeds. We have a huge diversity for fish, for animals, for microbes, for insects. All these things are there. And there are not one, not two, there are dozens of examples, which are the textbook examples where plant genetic resources from India have actually saved the world. When we are teaching the textbooks, we generally come to dwarfing genes. that genes have made a huge contribution to the global world, to global food production. That's true, no doubt in that one. But there are several important examples where genes from India have been identified and made available to the global community for use. And one of the recent examples is the submergence 1 gene from a landrace called as FR13A. And this single gene has really revolutionized the rice production, not only in Eastern India, but in several other countries of the world. And if we see the total money the Monsanto has charged for Cryo1 gene from India, its contribution to the Indian economy is much lower than the contribution of the SAV1 gene to the world at the moment. That's where we really need, we are telling the world, no, India has contributed not only in civilization, not only in education, we have contributed immensely towards plant genetic resources, and we are contributing as well. And if there are some young students who do not know, let them tell that ICR has one of the finest systems of managing its agrobiodiversity with the help of 5 bureaus. There's a Bureau of Plant Genetic Resources, which I'm representing now. There's a Bureau of Animal Genetic Resources, Fish Genetic Resources, Microbial Genetic Resources, and Insect Genetic Resources. Nowhere in the world you will find such a beautiful system. So that's where probably one can realize how India has been serious in managing its genetic resources. This is how NVPGR will just shift this one. What are our activities? Our major activities, in fact, is really collecting the germplasm, both indigenous as well as the global. Have a look on this one. This is a slide from, I think, 1946 to 2008. After cereals, the total number of the genetic resources are the vegetables. So if somebody is saying that NVPGR is stressing only on field crops, not on the horticulture crops, I think that's a myth. I will give you details of all these things later on. And this is just last 1 year's data. That's 2019-30. Here also you see the vegetables are at the top. The pulses, the fruits. Because in initial years, the emphasis was on cereals and food crops, but we realized that a lot of genetic resources of vegetable and fruit crops is still unattended. And that's why you see a lot of emphasis here. And 30% of our collections now are actually concentrated towards the wild species or the crop wild relatives of these ones. Which you may like to see, which have been collected only last year, be it Karonda, which is from Eastern UP, a beautiful plant, or this is a fruit from the Uttarakhand, which is again a minor fruit. That's where we are actually stressing now. Our emphasis, we have all the 10 regional stations whose major emphasis right now is to really ensure collection and conservation of our minor fruits which are ignored at the moment. This is just a glimpses of how our people go to the difficult areas but still continue. This is our team from Trichur going to the really difficult areas. The germplasm which has been collected last year from the Nicobar and other, most of them is horticultural. So this is the collection. Second, our emphasis on the conservation. And we have different mechanisms of conservation of our genetic resources, both for midterm as well as the long-term collections. And here you see the ones which are stored in -80. This is the recent status. As of today, NVPGR is conserving 4.47 lakh samples of seeds in its gene bank, of which vegetables, fruits, spices, condiments, they make a really an important component of this one. We are conserving a lot of genetic resources in our cryobank as well, in vitro as well. And this is the status of the germplasm that is being conserved either in vitro, which are primarily the perennial vegetables and perennial fruit species, and in the cryobank. Now, this is my, I won't say a challenge to the horticulture students. NBPG is stressing a lot on standardizing protocols for conservation of the pollen. Now, this is where our emphasis is really to see to ensure that a lot of germplasm, the pollen of the minor fruit, other perennial species is conserved. But this technology could be used for breeding purposes by the horticulturists, especially who are working on the perennial species where they cannot manage or they cannot ensure nicking of the flowering of the two different accessions which they intend to cross. The pollen could be conserved for years together. It could be conserved for a week or a month. Use it as and when you see. That's an advantage and the PGR, a good protocol standardized for the pollen culture, and we are using it commercially at the moment now as well. Just the glimpses of the different species which are being conserved, the vegetable ones. I have a list of all of them. You have the complete information.
Sir,
Many wild species of each of them. Can the participants who are speaking mute themselves, please? Yeah. This is the status of the medicinal plants detailed. The presentation could be with you. You can have a look on the details of this one. This is right now what we are conserving in gene banks. This is the data of the NDPGR only field gene bank. So we have 59 nations active germplasm sites, which are primarily the ICR institutions and the state agriculture university farms. I'll give you details of those ones as well. But this is a simple glimpses of the perennial species germplasm that is being conserved by our different stations of the NDPGR. This is a gene site at different state agriculture universities. Some of the glimpses of the wild fruits, which I told that we are stressing on. Some of the species, some of the vegetables, which may be still in wild, especially in the Northeast. We are really trying to emphasize on collection of these ones and developing the protocols as well for the management for seed increase, because when you go to the wild, you don't get sufficient seed quantities as well from there. So we're trying all those things with us. And to tell you that all the genetic resources which are available with NBPGR, they are geo-referenced. For example, anyone can click onto this site, go to this one, you are interested in working on rice, you click on any of these spots, it will give you the details of that one. And now we are working on, we're actually superimposing the soil maps onto this one, the climate maps onto this one, which are available for few crops with us right now, so that anyone who's interested in identifying the germplasm for any sort of adverse climatic conditions, adverse soil conditions, the users should have a liberty to choose rather than telling NBPGR, hey, I need just 10 genetic germplasm accessions from you, which could be useful for this one. No. In 5 years time down the line, we will ensure that all our genetic resources are actually geo-referenced along with the climate map and all these things available to you. We are helping you a lot when it comes to exchange of the germplasm or bringing germplasm from other countries. In last 5 years, we have been supportive in procuring 23,000 accessions of horticulture crops. Though if we see our annual is primarily still a major part of this exchange, our introductions are still wheat and rice from the CG institutions, but those are primarily the nurseries. This is the germplasm which we are supporting both public and the private sector as well. The 23,000 is specifically for the horticulture crops. We are receiving several requests from several people introducing the germplasm, but people should know, our young students, young faculty should know that there are always concerns. We have a large number of the quarantine pests, and if the providing institute, providing gene bank does not issue the phytosanitary certificate, we do not discourage you to not to bring it. We support for bringing the germplasm, but we do the post-entry quarantine. That means we really ensure growing them under containment conditions and then releasing you only if we feel they are free from viruses, from diseases and other pests as well. And this is primarily true for vegetable crops, which are having a large number of the viral diseases. So our request to everyone is be patient if you are introducing germplasm, and if we are delaying a release of this one for 1 year, don't worry about that one. We are doing it to ensure no COVID-19 like situation happens with us in the plant sciences or in our crops. That's where NBPGR is ensuring 100% security, a biosecurity. Some of the classical examples where this germplasm from other countries have been introduced by our breeders, others, which NBPGR has been facilitating. The next 3-4 slides are specifically to show you how the plant genetic resources have been used for release of varieties. We are compiling this whole data, and our preliminary data analysis shows that in the vegetable crops, more than 60% of the varieties which have been released are notified for cultivation are direct selections from the genetic resources. Because we have been asked the questions many times by the policy makers, what's the contribution? Where the genetic resources are being used? We are compiling it. In fact, whosoever is providing the real information we have, for example, these are some of the examples, be it the yard-long bean, some of the varieties which have been released by different centers, be it the cluster bean, the coriander, the ridge gourd, the brinjal, teasel gourd, all the varieties which have been released by different centers, notified by central variety release committees, and basically a selection from the genetic resources, or at least one of the parents is as a PGR directly, be it okra, be it greater yams, Chinese potato, all those things. So that was just a glimpses of the varieties those have been released, and we have a list of that one available with us. And NBPG has another responsibility of really conserving your most important genetic stocks through the process of registration. And out of the 1,300 stocks which are registered with NBPGR now, 347 are primarily from the horticultural crops. So this gives you details of this one, be it brinjal, bay, watermelons, onions, cucumber, chilies, some of the vegetables, the floriculture crops as well. Good number of them, all registered by NBPGR by different centers to ensure that no one misuses your genetic resources, or whosoever uses it uses with the routine or with the regular process of the ICR or the Government of India. All different crops, different genetic resources which have been really screened. We are doing a service to the farmers as well. Any farmer who is interested, because we get a good number of requests, farmers themselves want to introduce. This is one example of banana, where a farmer for Punjab was told that one of the mutant line from the Vienna is resistant and good for Punjab. We bought those, multiplied here, and provided to the farmers. We had several requests from the farmers now, where we call them repatriation of the genetic resources. One good example with us is taro. We got requests from the Kerala farmers that they had some old varieties of taro which were very tasty, very nutritious, but they lost them. 39 of them we have taken back from our in vitro gene bank, multiplied them our Thrissur station, and put back to the farmers. The farmers are growing. We are waiting for feedback from them. Because this has become a talk of the day, that we had old things which were very nutritious, very tasty, we have lost them. We are saying them, no, nothing is lost. Hopefully everything is available with MBPGR and with our research agriculture institutions in the country. Everything is available with us. We are also working on several potential crops, which are the vegetables, and Concorda is one of them. We have released 3 or 4 varieties so far, which are notified from Concorda as well. That's NBPGR's direct work in collaboration with its state agriculture universities and other ICR institutions as well. And here's my last slide, last 1 or 2, to tell my students how we are saying that this was an information for you. How these genetic stocks we are now using, evaluating to make them more useful to the breeders. Here we have given one example of the minor pulses. There's a complete paradigm shift in our approach. We are evaluating germplasm not in piecemeals. We are going complete evaluation. For example, here we have 16,000 accessions of 6 different species of the pulses. 4 to 2,000, something like that. I'll give you some details later on. We are characterizing them. We are actually genotyping them. We are generating the whole genome sequence wherever they are not available to us to generate markers. All high throughput genomics, all phenotyping data. Our idea is to really make available this to all the breeders and other researchers who are interested in mapping genes, discovering their traits in a really different, objective different mode. And in fact, thanks to DBT, they have provided us something to the tune of 380 crore grant for working on 13 different crops. That includes 6 pulses, 4 minor oil seeds, wheat, rice, chickpea, and our next crops in the line are some fruit crops. Dr. Dinesh is sitting here. The mango is one among them, with which we are working with IHR Bangalore. We are working on most of these the cucurbits, because that's a very important group of vegetable crops, but much genetic and genomic resources are not available. We are looking on that one. Brinjal is another crop with which we're looking on a larger perspective. Chilies, we have started in different projects looking for the genetic resources. So actually the focus is completely, for example, to give you this one, this is 4,100 accessions of mung bean being evaluated at 2 places, Jodhpur and Badnapur. Completely 4,100 in augmented design. And then all the 4,100 will be genotyped either through GBS or through resequencing or through SNPs. That strategy will be defined based on the genomic resources available in those crops. And we are doing it in next 5 years. We will have this data for almost 20 crops. And in next 10 years, we presume that we should have such data for several dozen crops, which are economically important, but are ignored, because a large number of the crops where you have the global sport, you have the,
5 minutes.
Right. So this is how we are saying we are using the real high throughput genomics along with the phenotypes, and germplasm being central to that, to identify these genes of interest, traits of interest, having the markers associated with them, and providing it to the breeders and researchers for the utilization. That's the strategy. Thank you. Thank you, Mr. Chairman. Thank you very much.
Anyone, please? Anything on medicinal plants?
Any specific question?
medicinal plants need specific suggestion.
For the medicinal
Sir,
plant?
sir, good morning, sir. May I give a question, sir?
Yes.
Sir, Nirmala Chakraborty from Department of Horticulture, Calcutta University, sir. My work is on the tomato and brinjal. And in that case, and my work is disease prevention, that is wilt disease prevention, Fusarium wilt disease prevention. Is any data is as there to that is the disease protection genes? Can we preserve the Fusarium wilt disease protection genes? Some specific genes is there. Can we manipulate these genes to protect or to evaluate or to produce a high Fusarium wilt resistant plants for brinjal and tomatoes, etc.? Or is there any germplasm? Or is there any germplasm is preserved to communicate or to predict the Fusarium wilt disease resistance data?
Good. So, if you send me a mail, I can really ensure you to send whatever data is available with us on the evaluation of these diseases for this particular crop.
Okay, thank you
we
very much, sir.
We have a portal. We have a portal. If you go to NBPGR web page, at the bottom of that, that's a PGR portal. And the PGR portal, if you just spend some time on tutorial, that will give you completely how you can really surf over this one. And you can actually give keyword Fusarium in brinjal. If there are any lines, they will come up straight from that one.
Okay, okay. Sir.
Part that manipulation, I think you will have a good number of the biotechnology people talking to you for next 3 days.
Yes, sir.
They will be giving answers on that.
Yes, sir. Okay. Thank you, sir. Sir, how can you obtain your email, sir, from the website, sir?
Yes, NBPGR web page.
Okay. Thank you very much, sir. Thank you.
Dr. Sam, one question from my end. These days we are using constructs. How NBPGR is managing with these new constructs, which are not specifically plant-based. They can also come from different sources. And somebody has to monitor the collection and reuse of these constructs.
NBPGR is managing some of the constructs as the genomic resources. In fact, that should have been the responsibility of one of the DBT institutions, which I understand NIPGR started at some stage. They had a special center for all the conservation of all the genes, promoters, constructs, but nobody actually contributed to that one. So what we have some of them for the people who have constructed, but NBPGR is stressing more on conserving the DNA samples of all the genetic resources. We may not enter into the individual genes. That may be responsibility of some other institutions.
Nice. Thank you. So we are running behind time. Thank you, Doctor. It was a very pleasant drop. You did give a good overall picture of the NBPGR and its utility, especially to the horticulture people. Thank you once again for your nice presentation. With my co-chair, I profusely thank you. Now can I call upon the next speaker, Dr. Vania Azevedo, who is the head of the Gene Bank at ICRISAT, Hyderabad. Gene Bank, the topic for is Gene Bank crop improvement. So to give a brief background about Dr. Vania Azevedo, I would welcome Vania Azevedo to this particular forum, and she's from ICRISAT, where we had earlier Dr. H.D. Upadhyaya also, one of my close friends when we started our service at Dharwad, and he has made enough contributions at ICRISAT. Now she has been a savior of genetic resources in the Brazilian Agriculture Research Corporation, and Madam is currently involved in the development of mini-core and distribution for effective utilization of the same. So she has a deep knowledge and commitment to the germplasm of crops that sustain low endowed populations of the world. And she has several projects and when reviewed many journals which are reviewed articles. May I now call upon Dr. Ania Vedo. Please, madam.
Thank you. Thank you very much for the introduction. And thank you to all the organizers for the invitation. It's a big honor to be here talking a little bit about the work that we are doing here at ICRIS Gene Bank. I will share my screen. Okay, so is it visible to everyone?
Yes, yes, please.
All right. I'm just trying to, all right. It's just a bit weird to me, but I can manage. Okay, sorry, I moved more than I should. So I'm just going to talk a little bit about Gene Bank, what kind of work that we are doing here, and how we are trying to make our material more available to the breeding programs with more information associated to it, and how we can be using the modern technologies to achieve these objectives. So a brief introduction. So CGIAR, as most of you know, but we have lots of students, so it's good to give a brief introduction also. The CGIAR is an international consortium with the objective to help develop the agriculture and hunger and the world. So in total, we have 15 centers located in strategic places all around the world. And of these 15 centers, 11 centers have gene banks. And each center is focused on specific crops that are important to the region where we are located. So these are the centers that are responsible for conserving genetic resources within the CGIAR system. So we have 2 centers responsible for rice, Africa rice, in Cote d'Ivoire and Erie in the Philippines. Dr. Veno is also here with us, and we'll be talking a little bit about the work with rice. Biodiversity in Belgium is responsible for the banana gene bank. And then we have CIAT in Colombia, CIMIT in Mexico, CIP in Peru. ICARDA has the gene bank divided now after they had to leave Syria. They have half of their gene bank in Morocco and another part in Lebanon due to environmental adaptations. ICRAF, responsible for the trees and fruit trees, is located in Kenya. ICRISAT here in India, where we are keeping the 3 legumes, 3 cereals, and other 5 small millets in our gene bank. IITA in Nigeria, ILRI in Ethiopia, and IRI I already said. In total, we are conserving more than 750,000 accessions of strategic crops. So the objective of the CGIAR is to work with the NARS and complement the work that the countries are doing and help in the development of the national systems. Different from what we just saw from Dr. Kuldeep Singh, NBPGR has the objective to conserve all the crops that are important to the country. It is the national system, and the international gene banks are here to complement the work with some strategic crops and not for all the crops that are being used. So the CGIAR gene banks have a special relationship with the world. We hold the material in trust for the international community. So the gene banks do not belong to ICRISAT, we do not, or to CGIAR, we do not own them. We conserve them for all the users all around the world. And we have to meet, or sometimes we even exceed international standards. This is mentioned there in the treaty, where we signed an agreement with the governing body of the treaty, and we need to follow very strict standards for the genetic resources conservation. So we are subjected to the policy guidance of the international community through the treaty. And our main objective is to provide a facilitated access to the material that we are conserving for free without any cost to any user in the world who wants to develop research and for food and agriculture. Some of the collections that are mainly the clonal collections that have more costs to be maintained and to be distributed, they can charge some administrative costs for sharing material, but most of the gene banks do not charge anything. The material is made available as long as we follow what is stated under the treaty, the SMTA of the treaty. So as I said, here in ICRISAT, we are focused in the drylands. So we have 6 mandate crops. So ICRISAT is also developing research with these crops, which are sorghum, pearl millet, and finger millet. The gene bank is also holding other 5 small millets: kodo millet, proso millet, barnyard millet, foxtail millet, and little millet. So we have all these other 5 small millets. And ICRISAT is developing a global project now called Smart Food with the objective to make these crops popular again. So I can again refer to Dr. Kuldeep Singh's presentation when he clearly showed how the consumption and the production of sorghum and millet have reduced significantly in the past years. And these crops are very, very important because they are highly nutritional. They are very well adapted to the most diverse conditions. They answer well to the conditions of the climate change that we are facing now, like high temperatures, lack of water. And they have many other nutritional characteristics that are important to us. So the ICRISAT Gene Bank is, the headquarters is here in Hyderabad in India. And we have 3 other stations in Africa, one in Niamey, Niger, one in Kenya, and another one in Zimbabwe, in Bulawayo. And then we also interact with the NARS, and we develop gap analysis to identify from where we still didn't collect germplasm so we can fill the gaps in our collection. So we work very closely to the NARS in the regions to help in developing their own genetic resources systems by providing capacity building and by helping in collecting new germplasms to enrich the diversity there. So here we have in here and in Niamey, the 2 main structures that all gene banks should have. The one that we call the medium term, from where we do all our research and from where we distribute material to all around the world, where we keep the material in 4 degrees, the seeds in 4 degrees and 20% of relatively humidity. And we need to guarantee that these seeds have very high viability because we need to distribute seeds that are alive and that can be used. And we also have to guarantee that they are healthy. So we need to do health tests as well. And we have another copy of the collection, which is the long term, where we keep the material in -20 degrees in aluminum foils, completely sealed, as a first safety copy of the collection. So whenever we need to go back to the original material to regenerate, to check, to rescue some material that was lost, we have another copy. For some of the wild relatives, we also have to keep the material in field or in vitro because they do not produce seeds or they will not produce enough seeds or viable seeds. So we need to use a different strategy. So we have some of the wild relatives of sorghum and pearl millet being conserved in the field. And now we are implementing an in vitro copy of these accessions. And down here, we can see a photo of our collection of Arachis glabrata. It's a wild relative of groundnut, and it does not produce seed. So these accessions were planted in this glass house in 1992, and they are being maintained like this since then. So the main activities that we do in a gene bank is to acquire new germplasm. At the beginning, everything that was there, we would collect when the gene banks were being started. Now we are doing this focused in gap analysis. We have to regenerate the material. We need to guarantee that we have enough seeds to be distributed to all the users all around the world. And we need to guarantee that the material is viable and healthy. And we need to characterize, which means to apply the descriptors that will allow us to differentiate one accession from the other. Of course, all the conservation activities, we need to document everything and distribute to the users with good information associated to it. And the evaluation comes as a research part of the genetic resources work, where we generate the data related to the traits of interest for the breeding programs, and where we can help the scientists in selecting the best accessions for their work. So ICRISAT Gene Bank has now more than 128,000 accessions that came from 144 countries, being the ones mentioned here the biggest donors of germplasm to the ICRISAT Gene Bank. The sorghum collection is the biggest one. We have more than 41,000 accessions now. And then per millet, chickpea, groundnut, pigeon pea, finger millet, and the other small millets. We distribute, as I said, to everyone in the world who wants to develop research on food and agriculture with these crops. We already distributed almost 1.5 million samples to 148 different countries, being India the biggest user of ICRISAT Gene Bank. And we have safety duplication in Svalbard, which is in black box. So we deposit the seeds there, and they belong to ICRISAT only. We are the only ones who can withdraw the material from there. And about 90% of our collection is safety duplicated there as well. Another responsibility that we have is to repatriate the germplasm to the countries where the material was collected. So it's very common that whenever a country is reestablishing or recovering, developing a national system of genetic resources conservation, they come to us and they ask us to repatriate whatever was collected there. And we also do that. So as I showed, the collection is very big and we want people to use it. It makes no sense to have such a big gene bank and not be used. So as part of our work, we are all the time looking for research that we can develop, and we can generate more data that can help the scientists to select the best materials to be used in their breeding programs or any kind of research programs related to food and agriculture. So it's important that we know our accessions, that we can estimate the diversity. The gene bank material is very diverse within the accessions, which makes the breeder's life a bit difficult. We can use all this research to optimize the conservation and to develop subsets based on the traits of interest, and many, many technologies that we can use to reach these objectives. So the gene bank is being used. We could clearly track few accessions that were used to develop cultivars. So more than 1,100 cultivars were released based on material that get out of ICRISAT. I believe that much more than that, but it's really difficult to track how the material is being used all around the world. So the gene bank is being used, and a lot based on what was already done to generate good data. So one way that is known is by definition, the first way for us to develop a subset in a gene bank to optimize the use is by developing a core collection. So Dr. Hari Upadhyay, who was my predecessor, he did this great work when he first started developing the core collection for all the crops, to start making good material with very high level of diversity in a smaller sample available to the scientists. So he first developed, based on the collection the way it was, at the size that the collection was at the time, he developed the core collection for the crops. By definition, core collection is 10% of the collection that can hold more than 80% of the diversity. So the accessions that are more different among themselves, in a maximum of 10% of the collection. So this is published in many papers, and he could, the team here led by him could develop these core collections. But then we can look at that and we can see that for example, sorghum and pearl millet, the core collection has more than 2,000 accessions. So is this really useful? The breeders here can tell me if they would start a breeding program with 2,247 accessions? I'm sure that the answer will be no. So how he could, so he started thinking as a breeder also, how he could do more than that. So as I said, they really realized that it was not being so effective for collections that are too big. So then they developed the concept of a mini core, which would be 10% of the core. So what is the 10% more diverse accessions within the core collection, which will represent 1% of the collection? So then he developed that concept, and he developed the mini core for the biggest collections. So the number was reduced in 10 times. Now we have 200, 180 accessions that can be used by the breeders, and with a non-big diversity. So this increased the chance of finding good traits for the breeding programs. And then, since then, the mini core became the subset that is more distributed by us. Most of the people who want to work with start a breeding program, they start with the subsets, the mini cores. So we have data from more than 36 countries now that evaluated the mini cores and generated data on it. So just from the mini core, we could find, for example, 41 accessions that show tolerance to abiotic stresses, and some of them being better than the control, which is the cultivar that is already released for this trait. 31 accessions that are resistant to biotic stresses, and 2 of them better than the control. 2 agronomic traits to both biotic and abiotic stresses, this for chickpea. Multi-trait material, so at the same time, some accessions that are resistant to drought, salinity, and herbicide, or to different kinds of diseases. So these allowed us to start a very deep work with a small subset of very diverse materials. The same for groundnut. We can see that some of them, they show 33% better yield, for example, or agronomic traits higher than the controls. So we started finding very good materials within the gene bank. And as I said, these became the most used materials from ICRISAT gene bank since then. And the same for all the crops. For sorghum, we can see materials that are multi-trait. We could even recommend the best pairs of most diversity accessions to be used in a breeding program as parental lines. But then we are talking about 1% of the collection, and we have the other 99% that can also have some good information, and then how can we generate more data about them? How we can understand the collection better, so we can be sure that we can share the data, the good data, together with the material to help the breeding program to develop even faster. And also these can help us in improving our conservation strategies, because it's very possible that we have some duplicates here in the gene bank. So we do not need to waste so much money and time with materials that are similar. So we have the advantage of having this high throughput phenotyping structure in ICRISAT. So we work with the physiology group in characterizing and phenotyping the plants. And we also apply very many other different kinds of evaluation to estimate nutritional traits, contents of in our crops, in our accessions, so we can identify accessions good for high nutritional content or for better use of the water, how they are using the water better. So this equipment, this LISI scan, it is reading the plants every day, every 2 hours it goes around, and we can estimate the evaporation, the use of the water in different times of the day, so how efficient each accession is. And together with these, of course, we can add molecular data. So this was a short study we did on heterogeneity to understand the diversity of the accessions. So these are quality and quantity traits that we estimated in 50 plants within each of 36 accessions of sorghum. And we could see that for these traits, we found 6 accessions that were heterogeneous, or the plants within the accession were different, and 30 that were homogeneous. For PGNP, we did the same 36 accessions and 15 plants, and we found a very big diversity. Only 4 accessions were homogeneous and 25 were diverse. So these could show us that the diversity within the accessions for phenotypic data is very high for PGNP. And for per millet, it's crazy. So we clearly see that for 25, we did more plants because we were already expecting a big diversity in per millet. Some of the accessions behave like populations. So it is very difficult if we sequence just one plant of per millet and try to associate the data to the trait that it carries because the diversity is very big. So it is our job to improve the use. We also estimate many of nutritional traits. So at the end of the curve, we can see the accessions that are showing very high level of nutritional content. So they can be selected for this use for both sorghum and PGNP. And we also did a genotyping of the same material that I showed the phenotypic data. So for genotypic data, we did. Sorry?
Another 5 minutes.
All right, I'm almost finishing, yes. For the genotypic data, we did dart-seq. We found the opposite. We found much more diversity. So that is why it's important that we really understand how the accession behave and how we can select, how we need to select the individual plants based on both phenotypic and genotypic data, develop the genetic stocks so the material can be much faster used and good traits identified by the breeding program. For PGNP, again, the diversity was very big within the accessions as well, and some of them very diverse. And for per millet, again, we found a very high level of diversity within the SNPs that we genotyped. So this helped us understand how diverse each crop is, how we need to consider the number of plants to regenerate the accessions because we don't want to lose alleles while regenerating. We cannot cause a genetic drift. We need to conserve as many alleles as possible, including the rare alleles, because they may be carrying the trait that we are looking for. And how important it is when you were talking about gene bank, not to immediately start from a single plant that you cannot associate the genotypic data to the phenotypic data. It is mandatory to be able to associate, because if you sequence one accession, one seed, and then you want to go back to that accession, you may not find a seed that is exactly like the one that you identified. So right now we are considering the development of genetic stocks of our materials to improve the use and the link between the phenotypic and the genotypic data. So we can, from the best plant that we see in the field, we can collect seeds from there, develop a genetic stock, genotype that material, and then we can go back to that seed with the genotypic data associated to the seeds generated from that plant, with the genotypic data associated to it. And then the breeding program can start from a genetic stock instead of the whole germplasm, the whole accession with a very high level of diversity. The other work that we are just now being almost uploading in our database is other subsets. So for use in forage in sorghum, for high biomass, multi-trait subsets, high diversity in proso millet and kodo millet, 11 subsets for chickpea related to disease resistance, other subsets focused in abiotic stresses, and subsets related to other traits that are important for the crop. So the breeders can go directly to these subsets, which are also very dynamic. Whenever we generate data for new accessions, we can add new accessions to the subsets. And also for PGNP, we are now concluding the development of 9 subsets for disease resistance, 5 subsets for different abiotic stresses, 2 subsets for multiple traits, and 2 subsets for the traits that are important to the crop. The same for groundnut, and groundnut also considering nutritional content, resistance to disease, multiple traits, and biotic stresses. So the main objective is that we can make good subsets with good data associated to them. So if the interest is to work with groundnut for high oil content, the scientists can directly identify which are the accessions that we already have data for this trait and that they can immediately start using. All the information is available in our website, genebank.ecrisat.org. All the publications are also there, and the data is also available in the international portal Genesis. So these are just the people that we have to thank for financial support, for technical partnership, and everything to be able to develop more and more data so the gene bank can be better used. That's all I had to say. Thank you very much once again for the invitation and for the possibility to be here. Thank you.
Thank you. So we had a wonderful presentation on the side from the leader. Now, may I ask all the participants to directly send their questions, queries, because the time is a constraint for us to interact on several matters here. So kindly cooperate, try to maybe one or two questions I can allow.
Madam, there are few questions in the chat box.
Sorry?
Yeah, madam, there are few questions in the chat box. Can you see, madam?
I can see lots of text here. I need to find the questions. How the germplasm accessible to farmer producer organizations to utilize for commercial production to get higher yield in millets. Yeah, in particular by ICRIZAT. So there are ways to get material from ICRIZAT. The gene bank material is not meant to be used directly for commercial, is not meant to be used for commercial use. The gene bank material is meant to be used for research. But of course, we have land races that we can give them back to the farmers, and then they, of course, can do whatever they want with the land races that came from them. But the ICRIZAT breeding program also develops, they develop elite lines that can be made available to the farmers. It's very simple. You just need to get in contact with ICRIZAT, tell what you want. And if we have lines related to the traits that you are interested, you will just sign the SMTA of the treaty and the material will be made available for free. The elite lines, they are made available for free to national programs and also to the farmers. There is no charge for this material.
Okay, thank you so much. Any other one question I can permit now?
Thank you.
So if there are no more questions and if clarification can be had on me, I would be appreciating that. Thank you.
Thank you.
So now the next speaker for us is Dr. N. Krishna Kumar, our former BDG of horticulture, and he also happens to be my colleague. And for last 40 years I have known. But for the benefit of all the viewers, I would try to put it very in short that he is going to speak today on utilizing crop wild varieties for biotic resistance in horticulture crops as way forward. He himself being an entomologist should be able to put forth several such issues to you. He had his PhD from Hawaii, then later with the US VA, then he came down, he was in IAHR, he served on several tomato leaf viruses, vectors, and currently he was with the Biotechnology for some time. And now I invite him, without much more introduction, to take the window. Thank you.
Thank you very much. It's nice to see Kulkarni and so many of the eminent persons that you are here. Am I audible?
Yeah. Why not?
Okay. So without wasting much time, can I go to, shall I share my screen? Yeah, this particular topic, which is basically utilizing the crop wild relatives for biotic stress and or management of biotic stress in horticulture crops, and how should we be going forward. I would just start my lecture with a small statement which says that I would feel more optimistic about a bright future if we find that if we spend less time that proving that we can outwit nature, but more time if we can taste her sweetness and respecting her seniority, which is very much more applicable to the crop wild relatives, because it's many of them may not be sweet, but in terms of the benefits that can accrue to the farming community, believe me that they are very sweet in that sense. And we should accept that in the evolutionary trends that have taken place in the course of evolution are far superior to a snapshot of our own thinking process, that the evolution has occurred over millions of years, and the innate knowledge that they possess, an organism or a wild species possess through its genes, is something which is very valuable and should be scientifically utilized for the betterment of the croplands, especially horticulture crop species, which in comparison to many of the field crops are more susceptible by very nature of their crop growth and the utilization. Now, I don't want to go, many of you would have already known, some of my predecessors would have said the major centers of crop diversity. But as far as India is concerned, we also have one of the group of islands of Andaman and Nicobar Islands, which somewhere are being hybrid between India and Southeast Asia. And thereby, when we talk about the major centers of crop diversity, it's also we should be really looking into Andaman and Nicobar Islands, a stretch of nearly 4,000 kilometers of islands, which consist of some of the wonderful genetic diversity. In fact, any of the time when I sit in horticulture group and discuss about where from the wild species or other collections are made, it is many of them are from Andaman and Nicobar Islands, and I just wanted to draw this attention to you. India itself is a center of diversity, one of the Wawilonian centers. I don't want to go into how many, but 50 indigenous fruits and their wild relatives, approximately 400 in number, in different agroecological zones. But it should also be appreciated in the spirit of things that no country has got everything, and it is only when we share, it is only that when we import, it is only when we discuss the utilization part, we will be richer and will be able to address many of the problems that are going to arise in the coming future. So this is just a slide which I don't mind any of you can see the number of crops which are of the primary centers of the diversity, as well as India is also many of the secondary centers of diversity. Some of us may be thinking that many of these crops are more Mediterranean in nature, but believe me, some of their wild relatives are also found in the foothills of Himalayas and the Indo-Afghanistan region. Of course, Doctor, I was listening to the director NVPGR, and they have a large collections in addition to some of the ICR centers and the state agriculture universities, horticulture universities having large accessions. So what is really important is we really categorize the numbers basically in terms of the cultivated species, and the crop wild relatives are included in one. But a time has come to say that in each of these crops, how many wild accessions do we have? And among the wild accessions, which of the species, and those kinds of splitting hairs is required now. Because in my opinion, if one has 5,000 accessions, what is more important is how many of them are crop wild relatives. If you go to a private sector, especially in horticulture crops, the first question they ask is, do you have this species? It is not about how many Lycopersicum esculentum you have. It is about how many accessions of Lycopersicum chilense that you have, which are going to be answers for futuristic breeding. Now, what is a crop wild relative? For those of you who would just like to know, it is a wild plant species closely related to the crop, as well it has got the genes of its wild ancestors. You can say that they are our wild cousins. The wild species became early, domestication took place with the evolution in farming, and then you have the modern varieties. And almost all the crops that we have today have evolved from the wild relatives. Many of you may not even know that for 200 years, tomatoes were not eaten in many parts of the world because they were considered as poisonous apples. So they were even afraid to touch the present day tomato, without which no culinary event will take place in our day-to-day activities. So it took quite a long time for the human mind to understand that these things are edible, and then the evolution of plant breeding has made it possible to use the introgress the genes to put it in the right perspective that we are able to harness. Horticulture revolution, if it has taken place today, the unsung hero is the crop wild relatives. Be it tomato, be it a number of other crops. Crop wild relatives are those which have not been given their due place, but in terms of genes, they have contributed tremendously. See, the wild relatives of crop plants that we have in India, of course, these are matters of statistics, but I just wanted to present these slides because tomorrow some of these details will be difficult to come by, and I thought I'll share with regard to how many relatives, crop wild relatives we have in different crops so that we can make use of it. Now, crop wild relatives, why are they important? Crop wild relatives are important because in one stroke, many of them are answers to both your abiotic and biotic stress. Many of them, what we apparently think for biotic stress, very nicely they come. For example, the dogridge rootstock, which was bred for salinity tolerance, is also moisture stress tolerant and is probably tolerant to many of the diseases. So today, the present day maize is a derivative of the teosinte, which is a progenitor of maize, and all the corn that we are talking originally evolved, and also some of the best rice varieties, salinity tolerance, and many others. As the climate change takes place, as the world becomes a global village, as persons begin to import and export in a large number, and travel becomes, it is only a matter of few hours from one part of the globe to others, both biotic stresses are going to increase in leaps and bounds. And more so in India, considering the type of quarantine that we have at this point of time. And when I mean India, it is predominantly across the globe in the developing countries. These diseases have the potential, like the coronavirus, when it is accidentally introduced, can devastate the cultivation of a number of crops. And the answers to many of them really lies in utilization and looking for source of resistance, incorporation of resistance, evaluation, and developing a gene pool with multiple disease resistance. Now, I just want to give an example of, you just look into these crops. We have said major biotic stress. You go to any farmer, and the first question he will ask is, I am able to get a variety that I want. I have somehow I will be able to manage. Even if it is a little unscientific plus and minus, I'll give some fertilizer. But when it comes to pests and diseases, I have no answer. Either it is the wrong diagnosis, or it is the wrong spray, or it is the wrong dosages, and then you have a series of problems. And in addition, for a number of viral diseases, soil pathogens, phytoplasma, you don't have easy answers. In fact, for most of the viral diseases and mycoplasma diseases or phytoplasma diseases, ecologically and economically speaking, host plant resistance offers the best source. And if you want to have host plant resistance or resistant material, where are the genes going to come from? The genes have to be, we have more or less really looked into in a number of major crops within the cultivated species, but we are far away from really looking into the gene and the gene pools that the crop wild relatives can offer. Let me just tell you that we have, in the case of tomato, tomato is one of the largest traded commodities across the world in terms of not cotton, but it is tomato. And why tomato? Because tomato leaf curl virus is important, and the tomato today stays or it stands on top purely because of Lycopersicum hirsutum. Today it is called as not hirsutum, it is called as Habrochetus. Lycopersicum habrochetus, that George Kennedy PI 177 that he donated, is today the background for all the leaf curl resistance across the world. And that too, it was originally done by an Indian, my predecessor, Dr. Kallu, who was first time responsible for crossing Solanum hirsutum with Solanum Lycopersicum esculentum and hirsutum, thereby showing interspecific crosses can work. Now we have tremendously exploited all the hybrids with the hirsutum blood, but when the tospoviruses or tomato tospo came, we do not have answers, and we are looking for Solanum chismai. Solanum chilense is one of those which is resistant to leaf curl virus, and my own PhD thesis, which I did long back, indicated this could be one excellent source even for resistance to tomato tospo. So today we want multiple disease resistance. Similarly, in the case of for increasing cassava protein or water resistance. So for a number of abiotic and biotic stress, we have to look and intensify our research on the crop wild relatives. Now, you just see the economic losses by different virus crops and the percentage yield loss. You will be surprised, 70, 80, 3200, 60, 8200, a series of viruses for which no amount of drenching with chemical pesticide is going to provide you with an answer. The answer lies in host plant resistance. The answer lies in breeding for resistance. The answer lies in introgression of very scientifically introgressing those desirable genes, either through marker assisted selection or through any other molecular approaches that you have. Unless we exploit these kinds of relatives, we have no future with regard to mitigating some of the emerging viral diseases. This is just to show up now crop wild relatives have been used in several crops, but this is mostly restricted to specific traits. But what we are looking is for multiple traits. What in one nutshell this explains is, so far our goalpost has been only in managing disease resistance, but in addition to climate change, which is going to bring in more problems that is there, our management of disease resistance, we have to move the goalpost, accommodating both climate change. Somebody is speaking. Can they switch off? Hello?
I request the participants, please.
Somebody is speaking in the background.
Participants, please mute yourself.
So we have to move the goalpost in our futuristic breeding. Now, crop wild relative breeding is a difficult process. No, for heaven's sake, somebody should not think it is such a simple process, but it is not something which is impossible. There are many undesirable traits in crop wild relatives, but wild plants, I don't know something, 1 second. Just give me. Yeah, wild plants are difficult to manage under domesticated conditions. There are crossing barriers, requires a lot of time, and the protocol has to be very, very critically looked into. And access to germplasm itself becomes difficult, and information on already pre-breeding material is often missing. The taxonomy itself, the crop wild relatives itself is very much sometimes in doubt. But having said that, it is possible to identify the targeted wild species, and based on the genetic and phenotypic evaluation, we can, in different environments, we can make it very useful. The crop wild relatives, I don't want to go into the details, but if somebody is interested, they can make use of it in this one. Now, one of the things when it comes to crop wild relatives, we always talk is about the breeding process, but the exploitation of the pollen of the crop wild relatives in breeding double haploids, in interspecific crosses, in getting into metaxenia effect. Some of them are absolutely, for example, the custard apple that we are seeing, Arka Sahan, is a classical hybrid but also has an external input of metaxenia in it. So pollen cryopreservation and then the aspects fall with respect to embryo rescue. These are all some of the techniques which need to be developed for a number of crops. We have to rapidly advance forward in this. Similarly, it is also important that marker-assisted selection, we need to very closely involve if we have to reduce the breeding
There's no
time.
more meeting, no?
Pardon me? And we have to go rapidly. I'm just rushing because of the things. I will not go into the details of how do you develop these kinds of population, but those of you can just go into this. I'll come to only the concepts. Now, the list of crops and the varieties that have been identified are very many, but believe me, wherever there is a mosaic resistance and many others, you can be sure that in most of the cases, one or the other wild species will be deployed. But what is really surprising is, in a country like India, there is no one center really earmarked for conservation of crop wild relatives. We have to, at least for the sake of horticulture crops, there should be one place. There is a need for ex situ conservation and there is also a need for in situ conservation. And at least in many of the plant species which cannot be conserved in situ, we have to create that kind of an atmosphere where we can at least conserve a whole range of germplasm ex situ so that it is very useful. At Biotechnology International, a small project made a beginning. I hope that it gets forward. It is in the UHS Bagalkot. Now, let us not be under the impression that these things have made tremendous impact only with regard to the horticulture crops, whether it is blight or resistance or blast resistance, tremendously, even with basmati rice, some of the wonderful work that has been carried out, even with monogenic resistance, has been by use of some of the wild relatives. But you can just see that while cultivated tomato occupies only a fraction of the genotype, we have tremendous way to go forward with regard to really looking into the genes, even in a crop which we apparently think we have done a lot of work. Still, we have many miles to go. In the case of tomato, it all started with looking for the breeding for leaf curl virus resistance. Today, if there is a hybrid tomato and the market is there, the leaf curl tomato virus, which is transmitted by whitefly, the market for hybrid tomato is 2 to 3 times or 10 times higher often as compared to the local varieties, simply because it has got the genes introgressed from the wild and is tolerant to tomato leaf curl virus. But most of them, you can see that we have Galapagensis, Chismani, Pimpinellifolium, Chimelswaki, and a number of other species, each one Peruvianum, and some of them can contribute a number of traits which we have. In fact, if the horticulture universities and others have to do, I would say that one of the finest research agenda should be we have to map different wild species for which of the diseases, which of the pests they are resistant. And once you have the data, and then the breeders will very beautifully make use of it in collaboration with pathologists and entomologists in developing very rapidly that breeding process. You have also seen that all the crop wild relatives need not be used for breeding. We are now seeing a revolution in horticulture crops through protected cultivation. And one of the major challenges in protected cultivation is the nematode problem that is associated. And today we have seen, in case of eggplant, in case of cucurbits, and in case of tomato, the grafting the cultivated species on like Solanum, Tarvum, or it is in the Cucumis, some of the species, they are highly resistant. You don't have to breed, you have to just use them as a rootstock. As far as horticulture crops are concerned, the application of crop wild relatives as rootstocks has tremendous advantages. One is it can straight away give abiotic and also partly and greatly biotic stress, especially for soil pathogens and nematodes. Besides, there is no linkage drag associated with introgression from the crop wild relatives. Thus, even then, we have not done a significant scientific profile of the different crop wild relatives for nematode resistance, for soil pathogens, for soil insects, salinity, and water moisture stress Combined resistance, I think we have got miles to go in these particular aspects by utilization. The answer lies is in future, we have to grow wild. This is one of my own work which I did for PhD. On the left, you see is the Lycopersicum hirsutum. On the right, you can see the small glistening structures or the trichomes, glandular trichomes, which have trapped the thrips. These are all happening at a microscopic level, and that is what contributes to resistance. All the plant species are endowed with different types of resistance. Wild species are having more of glandular and non-glandular, but to think that they are merely hairs and conferring mechanical resistance is one of the most stupid things. For example, the chemical that it secretes is 2-tridecanone, which is extremely toxic and traps many of the sucking pests. And this kind of a microscopic look along with chemical analysis is what is really required when we have to profile the crop wild relatives. Now, this is recently we also know that while leaf curl virus we were very much used to it, and tomato tospo virus, reports from Tamil Nadu by Malati and others very clearly indicated the occurrence of tomato spotted wilt virus, which was never there in this country. Now, in addition to the tospo, we have one more tospo, and you have to really profile different crop wild relatives if you have to combine with combined disease resistance. Every one of you knows the classical breeding that has taken place in okra with respect to okra yellow vine mosaic, the symptoms of which you see on the left and transmitted by whiteflies, and the aspects that we are seeing. Of course, this is a sweet potato. I'll come to okra again. The sweet potato weevil similarly has got a sweet potato weevil, and till today with this important crops suffers very heavily due to sweet potato weevil. And we have not really looked into the aspects of the crop wild relatives really conferring resistance to sweet potato weevil, but it makes a great difference to food security if it is utilized. Similarly, in the case of the legumes, mung bean, yellow vine, mung bean was a virus is one of the most important crops, and work carried out IHR, you can see how wonderfully the introgression has taken place and disease resistance has been bred. So this is what contributes to food and nutritional security, not simply dumping the crop with more and more insecticides. Now, okra yellow vine mosaic is a classical case. It took nearly 15 years to introgress Abelmascus manihart with regard to esculentum to come out over 15 years a resistant material, Arca anamica. Arca anamica was for about 10 to 12 years it was resistant, but gradually in the early and mid 90s it began to show breakdown of resistance, and even today we are thinking which of the species to be introgressed. Unfortunately, in the case of okra, there is a variable cytology associated with okra, so individual plant selections after crossing with a wild species is something that has to be clearly looked into. So cytology, interspecific breeding, field evaluation, all of them have to go hand in hand if one has to come out with a durable resistance. You can see that nature has bestowed us with lot of diversity, tremendous diversity, and some of the accessions collected by NBPGR are showing excellent promise. When it comes to eggplant, one of my earliest work showed that the Solanum macrocarpon, which is one of the wild species. Here I would like to say that resistance does not mean 100% resistance. Resistance can be to the tune of 90 to 90. Even the best of the vaccines that we have developed, we are telling 90% efficiency, 92%. So it doesn't mean that just because somebody takes a vaccine, he's going to be immune. No. Nature operates in a way that you can have high level of resistance, but it's still a great advantage when you have nothing. Likewise, Solanum macrocarpon we identified is extremely resistant to fruit borer, but with about 90 to 92%, leafhopper resistance, maybe bacterial mildew resistance, etc. Now, this introgression of the species to cultivated brinjal was often said impossible, but it has been very clearly demonstrated that this introgression is possible, and futuristic work is required. Futuristic work is required in this line before we go further. But look, the other side of the coin. If you are able to introgress the gene successfully, then all the debate about BT brinjal to a great extent. Even if you have a BT brinjal, it is only going to provide you 80 to 90%. And 10%, like in the case of cotton, always there is a resistant population that's going to evolve. And this is, not go into the details, part of the work done at IAHR, I'm sure. Now, why this the interspecific resistance is very, very essential is, there are a species of insects called as gall midges and such others, for which the plant will respond with a gall. As you can see on the left side, the ovary, there is a bulge. Now, when it is a host response, it is the host plant resistance which is going to confer the resistance, not the insecticide. And here we see the scope for the crop oil relatives. This is the type of damage that a gall midge will cause, and this is also a damage in capsicum. And the bacterial wilt resistance is one of the, especially in the coastal regions of the country, when the soil pH is low, the bacterial wilt is very, very significantly damaging. Though there are varieties resistant available, but I would still consider the use of the rootstock, rootstocks of crop oil relatives to be extremely. Look at the variability that is there in the case of chilies. We have Chile is a center of origin is Latin America. One center of domestication is Europe. Another center is in the Indian subcontinent. And most of the answers can be provided. For example, our own work very clearly indicated, if you just look for varieties which do not retain the sepals post-fertilization, they will be free of thrips damage in capsicum. So we have to look for trait-specific traits in the crop oil relatives for us to get that kind of a problem. Now, the management of virus resistance is why I would place it in number one. In fact, when we had a meeting with Dr. Swaminathan and the Allied Group and Indian Council of Agricultural Research, it was identified that the molecular approaches for insect resistance remains the number one priority, because even disease resistance to some extent we can manage, but when it comes to insect resistance, traditional breeding is extremely difficult, and molecular approaches, and more so when it is virus resistance, because the aspect of the vector is also involved. For example, take the example of papaya ring spot virus. All over the world, red lady is cultivated, and we do not have durable resistance. There was for some time the genetically modified papaya. There are legal issues, resistance breakdown, but what is required is traditional breeding. Now, some work was done with regard to the role of wild species, but I do not know to what extent it has been, but still the work should go on. One failure does not mean the end of it. We have to continuously work on these aspects, because nutritionally and economically, this is a very important crop. One of the emerging challenges that we are seeing through the indiscriminate import of the ornamental crops is the spread of the phytoplasma. Today, you go to any crop, whether it is coconut, whether it is cucurbits, whether it is sandalwood, whether it is a number of brinjal, the problems of phytoplasmas are increasing leaps and bounds. Like viruses, they can neither be controlled with insecticide, and it is a manipulation of the genes of the host plant by the pathogen which is responsible for the disease. Now, that gene has to be fought again. How the wild species are able to. Really, we have to understand at a molecular level how the wild species is able to overcome the pathogen and introgress those genes into our cultivated species if we have to manage. This is an example where you can see gerbera, instead of towering into different colors, all of them have become. This is common nowadays in marigold. This is common in gladiolus. Also, it is coming. A number of ornamental crops it is showing. This is just to show that Andaman and Nicobar Islands is the region where we should systematically focus. This is an example to show how a single use of rootstock made tremendous contribution to the hundreds of crores of rupees to this country merely using it as a rootstock in a matter of 15-20 years. But the aspect of diversification of rootstocks, whether it is the rootstock that is required for management of Phytophthora in citrus, or in the case of apple, many other rootstocks have to be really profiled, just to show the profitability of it. Another aspect which is also coming, the use of crop wide relatives in horticulture crops, is the management of excessive moisture stress. Excessive moisture is the one which basically contributes to a number of root pathogens and sometimes also contributes to diseases such as Phytophthora. Now, if you can graft it on the resistant wild stock, absolutely, at least you can provide 50 to 100% more. That means if a plant wilts in 2 days, cultivated plant, those that are grafted on wild species will last long. The same thing is fusarium wilt that has come on banana. There are wonderful breeding wild species that are there. ICR, in collaboration with international organization, is working on this. Now, sometimes the problems of interspecific breeding can be different number of chromosomes. I'll not go into this, but so I'll skip some of them. But what else we have to do along with this is we have to understand go very detailed. Today is a world where molecular biology is advanced leaps and bounds. For a number of viruses are basically very smart genes packed in very tiny organisms which took over the cell machinery or the entire gene sequence, gene functioning of the host. And we have to understand the genomics of both the virus as well as the plant species and the crop wild relatives if we have to manage some of this. Now, if you say important ones, borer resistance, nematode resistance, leafhopper transmitted viruses, bacterial, these are some of the aspects that are required. Parallelly, we have to understand the DNA barcoding and molecular phylogeny of different vectors, the aspects how much RNA interference can contribute, and metabolomics and insect genomics. RNAi or is something which plays a wonderful role, as you can see how the different species of constructs or chimeric RNAi constructs have provided very answer. In fact, one of our own work showed when we had played with the volatile genes, the brinjal shoot and fort borer was not even able to recognize its own host plants. So in addition to this, you can also use through different plants, wild relatives, to engineer, if not for edible purposes, for synthesis of some of the wonderful chemicals or the antioxidants, for example, as in this case, the lycopene enhanced tomato through genetic manipulation. So in future, the world is going to be a place where we are going to use different approaches to introgress what is there in the wild. And I feel a science-led progress with the good participation of plant breeders, molecular biologists, entomologists, pathologists, nematologists, I'm sure will pave the way for a food security, nutritional security, and also environmental security. Thank you very much.
Thank you, Krishna Kumar. It was a
Hello? Not audible. Hello? Hello? Hello?
many things.
I could not hear many of the things.
Thank you, Krishna Kumar. It was a very refreshing talk. I learned after a long time I had an opportunity to hear to you. So also the speakers. So there are an innumerable number of queries coming up. I can see on the screen. Maybe you can also give your email address or a contact where you can clarify some of them, because the time will be definitely a constraint here. So maybe one or two important questions you can take now and try to tell.
I have to go back to what?
Okay.
One
Please,
minute, hold on.
you please communicate your email address so that people will try to contact you on that.
Okay. If there is, I'll go through this. And my email address is kumarihr@yahoo.com.
Okay, thank you so much.
Kumar
Nice of you.
Thank you very much. Thank you all.
Thank you, thank you, Krishna Kumar.
Thank you.
So now the next speaker will on our agenda today is Dr. Raj Shekaran again from IIHR. He is going. His specialization is on biodiversity conservation, in vitro conservation, cryopreservation, IPR, and related issues. So quite a broad spectrum. He has been on the technology has been developed for long-term cryopreservations of nuclear genetic diversity materials and cryopreserved pollen in the form of NGDs of citrus, papaya, grapes, mango, and several other crops. So the pollen cryobank is managed by periodic replenishment of the cryogen and maintaining a constant cryogenic temperature throughout the storage duration. So this pollen cryobank at IIHR Bangalore has found a mention in the Limca Book of Records 2001 also. So without taking much of your time between the audience and yourself, now the window is yours, Dr. Rajshekaran.
Thank you, sir. Respected Dr. Professor Kulkarni and my dear delegates. I'm going to talk on the horticultural genetic resource conservation challenges and prospects. And in this presentation, I will be discussing on the issues which I mentioned in this slide. I'll give a short introduction on the various conservation strategies for the horticulture crops. And mainly, I will be focusing on the conservation of horticultural genetic resources happened at IIHR and the way forward. I will end up my talk with the way forward. And as you know, the diversity in horticulture is tremendous. We have the fruits, vegetable, flowers, spices, and vegetables and plantation crops. And there are so many other wild relative. Dr. Krishna Kumar has already told about this in his presentation. And the share of India in the world horticulture trade is only minuscule, that is only 2%. So there is we should do a lot, and the global issues identified in the horticulture are given here, and in there the genetic resource conservation and development is featuring very much. So the importance of genetic resources, this is the basic material on which we breed and produce new hybrids and others, and so there is a lot of importance is given for the genetic resource conservation and development. Now, coming to the horticulture, why there is, now there is a lot of importance is given for horticulture crops in the present scenario. And the production of horticulture crops have more than the fruit crops now, other crops now. And you can see here that there is a need to increase the value of local horticulture species. And coming to the conservation, the first gene bank was established in 1920 at Leningrad. And there is, of course, there is a need of in situ and ex situ conservation of horticultural crops. And there is tremendous effort worldwide on this. And there are more than 1,750 individual gene banks which are conserving 7.1 million accessions. And there may be duplications, and in these gene banks the number of horticultural crops accessions are very less, and the PGR is a fundamental input for productivity, diversity, health and quality of agriculture and food and feed production and our environment. And now coming to the conservation per se, these are the material by which you can conserve the accessions or the genetic resources. Of course, in situ conservations under the Ministry of Forest, Environment and Climate Change. We are not concerned with that. All other aspects we are concerned, and at IIHR we are conserving all this, using all these resources and techniques. And for any conservation strategy, we should be able to conserve maximum genetic diversity. So we should have a complementary conservation strategy. One strategy may not be enough to conserve the entire genetic diversity of a given species. So the two strategies should not be seen as an alternatives or in opposition or another, but rather as being complementary. And this had led to the adoption of more holistic approach to conservation. The formulation of an overall conservation strategy should apply a combination of different techniques available, as I mentioned, to conserve the maximum genetic size. Sometimes, in the case of wild relative, Dr. Krishna Kumar was mentioning that we should have the in situ and ex situ technique together, where the different methodologies complement each other. And coming to the the hypothetical representation of the proportion of the gene pool, if you use seed storage, how much we can conserve? If you, in the case of root tuber crops, forest species, you can see in this slide, by using different techniques, how much genetic diversity of a given crop or a given group of crops, how much genetic diversity you can conserve. So that again depends on the crop species and again depend on the type of technique you are going to use. Of course, the best way of conservation is seed, but it is not possible for many horticulture crops because they are not producing seed. Even if they are producing seed, most of the cases the seeds are recalcitrant, you cannot conserve in the gene bank. So the available other techniques are the field gene bank and in vitro and cryopreservation. So now coming to the scenario of conservation of Ecrisat lady already presented these aspects, and I'm not going to the details of these gene banks. But I would like to mention that there are 7 lakh samples available, but horticulture crops are very much limited in this CGIAR gene banks, mostly this having the cereals and other related crops. And these are the places of these CGIAR gene banks. And this I would like to show you that these are the accessions available in this gene bank concerning to the horticulture crops. There are very less number of crops and accessions. And maybe due to the lack of seed in the crops and most of the cases, if seed is there, it's recalcitrant and difficult to store. That may be the reason why these CGIAR gene banks are having less number of accessions in the case of horticultural crops. Now coming to the ex situ conservation, aim is to keep the germplasm alive as long as possible and reduce the frequency of regeneration and minimize the genetic erosion. And we have the techniques of seed banks, field gene banks, in vitro, slow growth, cryopreservation, and DNA bank. And for the wild and other species, we have the botanic garden, arboreta, etc. And as I was telling you, I will be giving you details of the work which we have carried out at IHR. So this is the how we can conserve a species given by Maxwell. So we should have the ecogeographical survey, then we should outline the conservation objectives, do the field exploration and collect the accessions, and then we should form the conservation strategies. As it's well known that we should have the ex situ and in situ conservation, and ex situ conservation is in the form of seed, field gene bank, botanical garden, in vitro, pollen and DNA, and in situ collection is in the form of genetic reserves on farm for the cultivated species and on farm and home garden for the cultivated species. And of course, when we achieve the conservation, we have the conservation products, and this should be utilized for the betterment of the society. So these are the well-defined protocols available for both the in situ and ex situ conservation. Of course, in situ conservation, as I mentioned, it is with the Ministry of Environment, Forest and Climate Change. And for the cultivated field species, we should have on-farm conservation. And ex situ conservation we achieve, as I mentioned earlier, through seed bank, in vitro, cryo and DNA. And field gene bank is for the vegetatively propagated and tree species, we should have invariably we should have the field gene bank. But conserving in the field gene bank is very costly because we need a lot of land and other resources to conserve the material in the field gene bank. And coming to the conservation activities at IIHR, we have the exploration.
Hello, Rajshekaran, can you hear me? Hello, Rajshekaran. Can you hear me, please?
There is some network issue from the speaker side.
Yeah. I will be back now. Now I am. But 1 second. I'll.
Okay.
Yeah.
Thank you, sir.
Bro, ഇതൊന്ന് എങ്ങനെയാണ് ചെയ്യാ ഫ്രീ ഇതില് ചെയ്യണം അതെങ്ങനെ സൂം എവിടെ ഇതിലെവിടെ സൂം
I can hear you, Rajshekaran.
Yeah. Just 1 minute. Sorry for the. വരുന്നില്ലല്ലോ സാർ
Rajshekharan ji. Dr. Rajshekharan.
Hello, sir. Sir, it's coming, sir.
Okay, go ahead, please.
Rajshekharan ji.
Yeah, sir. One second, it is coming, sir.
If any issues there, we will take up other presentation.
Over, sir. Now it is coming.
ಸಿಗ್ತಾನಮ್ಮ ಫೋಲ್ಡರ್.
Hello.
Go ahead, doctor.
So these are the activities which are related to the plan. This we did in North Canara. And another aspect is where these land races and wild relatives are available. In a village that the tribes of Siddhi, they are living. These tribes are came along with the Portuguese, and they have their own cultivation, and they are ready to share their resources and information with us. And some of the tribes are not ready to share the resources, and this you can see brinjal variety we collected from Shimoga. This weighs up to 500 to 700 grams, and this is cultivated in a small area. And on the right side, you can see how the farmers conserve the seeds. And in Amarantha, we collected a lot of diversity. In fact, 45 accessions belonging to different species of amaranth. And in Maharashtra, in a small village like Gowal, they are cultivating the traditional vegetables of cowpea and brinjal. Then in Dharwad, we could collect a lot of local varieties. In fact, in brinjal, we have more than 2,000 varieties in India. Many of them are land races, and these are all the local varieties which are preferred in Karnataka. And in fact, we went to Ooty to collect the beans. There we could collect 38 accessions. In fact, it came along with Europeans to India, and in Ooty they are cultivating this. And as Dr. Krishna Kumar was telling, a lot of importance for the wild relatives. This is a wild relative of Momordica, that is Momordica sahyadrica. And in Dakshin Kannada, they are not allowing any seed companies to establish their office in Dakshin Kannada, and they have their own gene bank called Namma Community Gene Bank, where you go with your seed, you can get it exchanged. And coming to the our pollen cryobank, this is one of the biggest pollen cryobank in the world. We have 100 species pollen belonging to 50 families, and for the last 37 years we have been cryopreserving this, and this technology has been transferred to 3 seed companies, including the Indo-American hybrid seeds, and they are using this for the hybrid seed production mainly in solanaceous crops. And maximum period we tested for pollen is 26 years, and technique for the collection, viability and fertility assessment of pollen optimized for 100 species. And as I was mentioning, this was featured in the Limca Book of Records in 2001 for the largest pollen cryobank in the world. And this is a pollen cryobank, the snapshot of the pollen cryobank. And in fact, we have the seed bank. Now we completely refurbished the seed bank. And this is the in vitro gene bank. So we have all the type of conservation technique we can use it and conserve maximum genetic diversity. So I was telling you, we have established a field gene bank for rare and endangered medicinal plants. In fact, we have taken 32 species belonging to these rare medicinal plants and established a field gene bank. The idea is to collect the diversity from the in situ conditions and bring it and establish its field gene bank. And you can see the different views of the field gene bank. And this is maintained completely organically, and we don't use any chemicals, and including the border we are cultivating these plants. And we did the chemical profiling of these plant species which we have collected to know what are the chemicals available in each species. And you can see the chemical profiles of these species in this slide. And to know the genetic diversity, we did the molecular profiling using the molecular markers. And you can see the molecular profiles of Acorus calamus, Elastus paniculatus, Alpinia calcare, and Oroxylum indicum in this slide. And in fact, we were receiving a lot of visitors. On the top, you can see Professor Dr. Pushpanathan, who was a former director of TBGRI and NBRI, and the former director of BSI, Dr. Sanchita. And now I would like to share one small case study on the in vitro conservation of Nothopodytes pneumonia, also known as Mapia foetida, and in Kannada it is called Durvasa nemara. And first we established the plants the in vitro system for this plant. It was very difficult. In fact, it took 6 months to establish the in vitro system for this. And we used all the explants, but only the seed cotyledons were given the response in this. And you can see here, after 3 weeks of incubation, small spherical globular structures were seen in the media. And without the thiodiazurone, there was no response for this species. And you can see here the somatic embryos of this plant, and we could get almost 70 number of somatic embryos per tube. And you can see the fully developed plant below. And we could get the multiple shoot also. So this is two ways we could establish this plant. One is through somatic embryogenesis, and another is through multiple shoots. But in case of multiple shoots, we can get this by using the TDS medium in one week time. But shoot elongation was not possible, and we have to use another the medium for getting that. And you can see here the well-developed shoots, and for rooting you have to give IBA treatment, otherwise the concomitant rooting was not there in this species. And you can see the response of reduced culture condition. We were able to conserve these plants in the in vitro conditions, and we went for the hardening, and we could establish the plants ex vitro. So now the last 5 minutes, I will just tell you about how, see, this is project it was in a network mode, and we were asked to do the conservation in Maduka insignis. It has got a very interesting story. The Maduka insignis was rediscovered after 120 years, and if you see the red data book of IUCN, there it is mentioned as extinct. And these plants First, we ask this question: what is the current status of this species in the nature? You can see in the IUCN red list. If you go to the IUCN website, you can see it is extinct. And after 120 years, it was rediscovered by Dr. Gopalakrishna Bhatt and his team in Shimoga district of Karnataka. And what we did is we did the distribution and prediction map we generated using RTTS. It is a software, and by using this, we generated a distribution map and prediction map of the species. You can see the prediction map on the right side and the distribution map on the left side. And you can see first time with using this technique, we could identify three population of this species in Karnataka. And we did the prediction for 2020, 2040, 2060, and 2080. You can see what is going to happen for this species in years to come, and what the conservation strategy required to conserve the species, because the number of individual plants in the population is very much limited. And the second question we asked: are these collected species are authenticated? Are the collected species are really Maduka insignis? And you can see here, the Maduka insignis is closely related to Maduka nidifolia. So in the seedling stage and even in the other stage, it is difficult to identify this species. And you can see the almost similar, even in the seeds are also looking similar. So what we did is we developed molecular markers, species-specific scar markers for identifying these insignis. And you see here the sequence how we developed the molecular markers. And by using these molecular markers, even at the seedling stage, we can identify this plant. And next question we asked is how we can conserve this species. And we developed the conservation strategy like this. What we did is we isolated a microbial consortium from the soil taken from the root side of this plant and used to germinate the seeds of this species. In fact, seed germination is possible only using this microbial consortium, and we generated the seedlings. And if we use this microbial consortia, the growth was very good, and you could see we developed a lot of seedlings of this species. And another thing what we did is we augmented the population of Maduka insignis. In fact, only 18 plants were available in nature. So what we did, we did this work in collaboration with the Karnataka Forest Department, and in fact they were very cooperative in this aspect, and we re-established these plants in a number of 600 plants we established in the forest ecosystems. And now the forest department is taking care of this, and with this we could increase the number of plants in the wild. And Pilikula Nisarga Dama also we could establish these plants, and even IHR we have a small population of 25 plants. And you can see the status in different places wherein we could locate.
Another 5 minutes?
Not even 5 minutes, 2 minutes I will be over. The status of the plants you can see in the total population here, and there is
Rajshekharan, another 5 minutes?
Yeah, not 5 minutes or 2 minutes. So these are the problems for the this plant species, why it is getting extinct, or why it is getting the number of plants is getting reduced in the wild, is because of it. There is excessive mining, and excessive sand mining is going on, and fragmentation of the ecosystem, then excessive sand mining, as I mentioned, in these places, and the loss of Maduka insignis inflorescence and damage due to the frugivores. And coming to the summary of this work, we did the population studies, and first time we did the isolation characterization and submission of the plant growth promoting fungal consortium, then development of the scar markers and submission of gene sequence to the public domain of NCBA and chemical profiling we did. And even we did the cancer studies using this crude extract, and we reintroduced this plant into its niche habitats and providing roadmap for the future conservation and defining exciting research areas to be explored in the future. And most importantly, taking a small step to help to conserve this critically endangered species. And in fact, we could get a lot of publications from this project. You can see 8 research papers have been published. And now another conservation aspect which I would like to share with you is this UNEP-GEF project undertaken by IHR. There mango and other 3 fruit species were taken. There the custodian farmers were identified, and they were conserving the species. I'm not going to the details because of lack of time. And this is the way by which you can conserve the tree crop species. And now we have come out with 2 compilations with the Springer Nature. This is the first one, conservation of utilization of different chapters by the experts have given, and the latest one is on the conservation utilization of threatened medicine plants. This also with the Springer Nature, and for pollen protocol book is coming in the next year, but also to be published by the Springer Nature. And this is my Google site, this one. And to conclude, see, 75% of the world poor are rural, and most are involved in farming. And the 21st century agriculture remains fundamental for the poverty reduction, economic growth, and environmental sustainability. And our national food security is depending on to conserve all our biological wealth. And I would like to thank the organizers, especially Professor Fakhruddin and Dr. Halish, for inviting me for sharing my experience in the conservation of genetic resource of horticulture crops. Thank you very much.
Thank you, Shekhar. In spite of even all problems in audio and video, you did make it in time. I thank you once again for making it very nice. Now the next speaker on our line is Venuprasad Ramaya. He is now the head of the gene bank at Erie, Philippines. So he happens to be a master's and a PhD student of my department. So I have very much thankful for providing his biodata here, which I had missed for the last 10 years. So he has worked in Nigeria and in Erie as a adjunct postdoc person, later in Nicrisat and Taiwan and Philippines. So it's not much to say, except that he has been able to release some varieties for Nigeria and Madagascar. And besides that, he was our Melinda Gates Foundation grant holder. With these few remarks, I now call upon Venu to take the windows and address the audience in the limited time that is now marked for them is just 10 minutes. Thank you.
Thank you, Chairman, and good afternoon, everybody. I hope I am audible to all of you.
Yeah.
Thank you. And is my screen visible?
Yeah. Right now, no. Yeah. Now the screen is. Now it is visible.
Oops. Sorry. Okay, good afternoon, everybody, and thank you for the organizers for inviting me, and it's a pleasure to be here. And so thank you for the introduction. The topic that I am talking today is about how international and CGR Gene Bank, particularly taking example of IRI, how they are working and contributing to this sphere of conserving and exploiting plant genetic resources. Of course, you have to recognize the rice is no horticulture crop, but I believe some of the examples that we provide will enlighten the group about how the CGR gene banks are contributing. And some of the parts that I am talking is also been covered by my good friend Vania from ICRISAT. So many of the things that she covered also applies to us. So I would be avoiding some of those things in the interest of time. So many of the speakers today have already spoken about the gene banks and their role. So that we have a main role in conserving crop diversity. And in the worldwide, if you see this conserving crop diversity is no small task, and it cannot be done by any single organization or a country. So it needs a network of gene banks and countries worldwide. So there is an estimated more than 1,750 gene banks worldwide conserving more than 7 million accessions. And these gene banks can be grouped as international, regional, and national gene banks. And within the international gene banks, we have CGR and non-CGR. And I and Vania represent the CGR gene bank, but there are others, ICBA in Dubai and AVRDC in Taiwan, also contributing to these topics. Now, this has been covered. So we are based in Philippines. So International Rice Gene Bank is said to be one of the oldest rice gene bank. It was set up in 60s, but officially became an entity in 77. It has a lot of feathers in its cap. It is said to be one of the oldest CGR gene bank. It's also the largest in terms of the size of collection, and the collection is said to be the most diverse compared to any other gene bank. And it has also been said to be one of the best maintained gene bank, best characterized, and most heavily used. Now, so what is that we do in terms of conservation? So we have over 130,000 accessions, most of which are cultivated species of both the Asian and African cultivated species. Then we have a very good number of wild accessions. All the 22 wild species of cultivated species we have in our gene bank, and it also includes 7 related genera. So where do these accessions come from? This picture here tells how we have the collections coming from different rice growing regions in the world. There are, I would like to say that hardly there is any country in the world which is a rice growing country, and we don't have accessions from them. The green dots here represents the amount of collection that we have coming from that particular region. So it's a very worldwide connection that we have. And also to show how these acquisitions have accumulated over time. You know, it's 130,000 acquisitions didn't just accumulate in the first few years, but it has taken almost 45 years to accumulate this amount of collection that we have. So it is most diverse and the most largest and most well documented and characterized collection. So we take safety very crucial and mandatory for us to have 2 duplications, and we have one in Fort Collins in US and one in the Svalbard, and thus we ensure that the important diversity is safely conserved for future generation. So conservation part is somehow, as I said, is taken care, but we also have to make this information and the resources available for people to use them. Otherwise, there is no point in just collecting, conserving them. So we mentioned here some of the web pages that users can access the information that we have, and then also request for germplasm. So as Vania said, we give this germplasm to anybody in the world free of cost. So students, researchers, farmers can access these materials. Then coming to distributions, how to show how our gene bank has been used. We have recently on average we to worldwide. We have sent materials to 116 countries. And when we send materials, we ensure that these are healthy, viable, and by following proper policy guidelines. So we also undertake some of research. These research are related to conservation. So how do we make our conservation better and reduce the cost of operations? This is a main concern. So we are active in this area. And seed longevity is a particular trait of interest. And because of learnings from our research, now our management have been fine-tuned so that we have a very good setup where the longevity is maximized. Some of the accessions we have over 45 years, they have been still since the start of the gene bank, and they are still maintaining high viability. We have identified some of the secondary metabolites which are important in the longevity. So this research is currently ongoing. Now we also undertake research into understanding diversity of the gene bank collection. This is a bird's eye view of our characterization nursery in Nairi in Philippines. This is the wild, the cultivated species grown in the Then we also have diversity in the related species. As I said, we have 24 species, but here for representative, I have indicated 12 species of Oryzandia. As you can see, there is tremendous variations that is there. So we also undertake research into saying how much genetic diversity is actually there in the gene bank, and particularly at the genomic level. So just 2 years ago, we published this work where we have sequenced more than 3,000 accessions of Oryza sativa, and we have published these results. And the very interesting information emerges from this type of sequencing data. Earlier, we knew that we have 5 subgroups within Oryza sativa, but the sequencing information has told that there are actually 12 subgroups, and within one of the main groups, there are several ecological differentiation is also observed. We also have some estimate about the kind of variation at the molecular level. Within the 3,000 samples, we found that over 30 million biallelic SNPs are there. And when you extrapolate this information to the whole collection, we assume that, we estimate that there could be more than 110 million SNPs, which is about 1 SNP for every 4 base pair. This is very, tremendous amount of diversity that we have in the collection. So once we have the diversity, we also have a standard pipeline to mine the gene bank, but we have a native trait discovery and deployment pipeline. So I have indicated here, in the interest of time, I cannot go to them. We are also involved in pre-breeding, especially how to exploit the wild species. And this picture indicates the usefulness of the wild species in the grass. If in the picture, the yellow spot here indicates the cultivated and back and crossed to Oryza rufipogon coming from 4 different countries. And as you can see, for drought, there is plenty of diversity that is coming up, and many of this could be actually exploited in breeding. So that's about what we do, and let's take a deeper dive into how we contribute to this conservation and particularly the utilization topic. I want to give some hardcore examples which tells us how we have actually been contributing. So the main contribution that we have is to developing of improved varieties, to breeding. A lot of people have already touched upon this, and this figure here indicates how many gene bank accessions have been used in varieties developed by IRI and its partners. So this study studied all the varieties that was released by IRI and its partners in Asia. So over 4,000 varieties were studied, and they say 90% of them have at least one accession which can be traced back to the IRI gene bank. So that's the contribution to the breeding. And it says this type of varieties have caused, have been used or developed for various purposes. The Green Revolution is role of gene banking. Green Revolution is very well documented. It's lifted millions of people from out of hunger. Then in grain quality is an important trait for rice, and then the gene bank has contributed heavily for this. If you know a variety called IR64, which has the $1.3 billion per year, is said to be its contribution. And there are a lot of wild cultivated and wild species in this blood, which comes from IRI gene bank. Similarly, our gene bank has contributed to traits such as leafhopper resistance, Sogato. In Latin America, imagine a varieties coming from Asia have contributed to improved leafhopper tolerance in Latin America, and now the 25% of area has been protected because of that in Latin America. Another example, popular example, is bacterial leaf blight, and XA4 gene is very well known, and it has a tremendous impact. Grass-resistant virus, Dr. Kuldeep Singh from NBPGR already mentioned this. This came from a wild species from our collection, and then this has been widely used also. And there are some estimates which says that in Southeast Asia alone, whatever breeding that has happened contributes to about $97 billion. And our gene bank had a lot of contribution to this type of work. Now, these are historically how things have happened, and some of the recent examples is contribution to abiotic stress. Dr. Kuldeep Singh mentioned about the flooding and submergence tolerance. This is a picture of that. So we have a gene called SUB1, which came from an accession that was collected in Orissa in India, and now because of that, the worldwide, this gene is being used, and almost all the mega varieties in Asia today have these genes, and it gives protection against flooding. Now this gene has also been moved into African varieties, and now they are also taking benefit of this. So that's the beauty of this type of work. And about 49 million acres is said to be benefiting from this type of work. And coming to nutrition, we recently released the world's first high zinc rice in Bangladesh, and the source of this high zinc is traceable to ERI gene bank, and it's estimated that around 2 billion people can benefit from this type of varieties. We also have a lot of work ongoing on low GI in rice, which is an important topic, and it's estimated that at least 500 million people can benefit from this type of varieties. So not only that we are contributing to improved varieties, we also have a role in disseminating traditional varieties to farmers. Often farmers give us requests for their own traditional varieties which are no longer in cultivation in their places. So we help them revive them, and then there are plenty of examples of this. We also help in reviving some of the lost materials, and if you can, some of you may know about this famous example of Caronilla gold, which was lost during this modern agriculture, but it has now been revived, and now it is like a heirloom traditional varieties being grown in South Carolina in US. Of course, this is mainly USDA gene bank related, but I take this classical example. And we also have several such examples. For example, Hong Kong, no rice is grown there because of whatever, but rice was grown before, and then we have accessions that came from Hong Kong. So recently there is interest in Hong Kong to grow rice in terrace farming, for example. And then we have sent them back their own varieties, and they're quite happy with this. So we also help in restoring diversity. Sometimes farmers lose their material because of flood or maybe due to civil strife, as is in Cambodia during war. A lot of materials were lost, and because they are safely kept in our gene bank, we help them restore them back to their original state in their countries. We also are involved in supporting community seed banks. Ours is ex situ gene bank. We don't do a lot of field gene bank or in situ type of work, but we support the national programs in conducting some of them. This is one example in Philippines. Then we also have provided a lot of support to the national programs. ERI Gene Bank has helped many countries to set up their national gene banks based on the experiences, lessons learned. And many of the countries in the world keep their safety backups with us, like Sri Lanka, Cambodia, Laos, PTR, and Philippines. All their safety backup is at ERI. Now, restoration to collection. Sometimes what happens is when the gene banks are starting, the national gene banks, there may be several reasons why they lose their collection. Maybe because of war, or as I said, or because there were temperature problems, or because of lack of facilities, or for many reasons they lose their collection. And now, since they keep their materials with us, we give it back to them so they are able to restore it. So I would say that India is one of the major beneficiary of this type of activity, and over 40,000 accessions from the Indian national, I think NBPGR gene banks, we have restored back to them. So this is the value of this type of work. And capacity building is one very important topic for us. And every year we host several, at least 2 or 3 gene banks. And this is one example where Myanmar Gene Bank faculty are being trained at ERI Gene Bank in their routine, and this helps them to improve their operations. So we also very serious about how our resources, our information is being used by users. So we regularly take feedback from the users. And this is 2018, we conducted one major survey, and then we can say that users are quite happy with our ERI Gene Bank with respect to the material, the quality of the material, and the information that is available. And we can say there is 90% satisfaction rate. Looking into this, now recently we got what is called as permanent funding for the ERI Gene Bank by Crop Trust because of the contribution that we have and the way our collections are being maintained. So as long as this is a perpetual grant, so as long as humanity exists, ERI Gene Bank is having funding. So we receive about 10 crores per year for our work, 10 crore rupees for our work. And this is a perpetual grant, and we are thankful for our donors for this type of funding. So this is my last slide. Yes, we are very active in conserving, and at least we can ensure, we can say that the price genetic resources that are in the gene bank are safely conserved for the future use. And it's not enough if we just conserve them, we have to properly use them. And I think this is still a limitation. The wealth of diversity maintained in this in the gene bank, even in ERI Gene Bank, is largely untapped. Some reports says that only 4% of the collection has actually been used, which is very, very meager. So there is a tremendous, and given that the challenges the world is facing now, there is a tremendous need for us to be digging deep to exploit this diversity. And I would like to say the other point is that gene banks are generally gene rich, but they are data poor. So there is a need to increase this data availability on these materials. So then only then the utilization of that will pick up. And there is also challenges in adoption of technology. New technology comes fast and then there keeps changing. But ERI Gene Bank is at the forefront of adoption of technology. A lot of things are automated. We are now ventured into artificial intelligence and machine learning platforms to improve our management of the collection. And also there are a lot of things happening in the policy area which can really impact us. If some of you may be familiar, the area of DSI, the renegotiation of SMTA, these are some of the challenges that can impact the way we function. With that, Chairman, I would like to thank the opportunity for us to share whatever we are doing with the larger audience. And if there are any questions, I would like to take. Thank you.
Thank you, Venu. Dr. Venu has some wonderful work to be done at the ERI. So may I now ask the participants to limit their questions to only 1 or 2 important ones. Any questions? Any questions, please? So if there are no questions, we will now move on to the oral presentation. Thank you again, Venu.
Thank you very much. Have a good day.
Now we move on to the list of poster presentations. The first speaker is Dr. Kumar Swamy, HH. He is a scientist biotechnology of ICAR, Oilseed Research, Hyderabad. And he is going to talk briefly on genetically stable genotypes with contrasting phenotypes. And I now welcome him to take the window and restrict himself just 10 minutes. These are the poster presentations. Thank you.
Hello, am I audible, sir? Hello?
Okay.
Yes.
Am I audible?
Please.
Yes, Dr. Kumar Swamy.
Sir. Okay, sir. Sir, share screen, I am going. Sir, I'm unable to share screen. Okay.
Hello. Brian also.
Can somebody guide me how can I share screen? This is totally different. I am Zoom meeting first time attending.
Yeah, can you see the option share screen below?
Yeah.
Yeah, click on that.
There are one participant share at a time.
No. Just click on the share screen.
Okay.
Or plus Alt 5. Oh, sorry, Alt S. Alt S. You can use the shortcut key Alt S.
Okay. Is it shared now?
No.
Alt S I am pressing.
You select the screen which you want to share.
Now, okay.
You got a pop-up? When you press Alt S, you got a pop-up? Yeah.
Now is it visible, sir? Is it visible now?
No.
When I press, okay, it is popping up, but there
you select your PPT. Yeah, it's starting. Yeah, it's visible.
Now I'll go to my.
You can go to full screen mode, presentation.
Yeah. Is it okay, sir? Now?
Yes, perfect.
Okay. First of all, at the outset, I thank the organizer for organizing a wonderful international conference as well as for giving me this opportunity. So today I'm going to speak to all of you these genetically stable genotypes with contrasting phenotypes that have potentiality for using as a genetic resources for mapping of yield contributing traits. To begin with, let me set a precedent. When maize, rice, and such other crops are traveling by flight, sesame, Niger, like these minor crops are still traveling by bullock cart. That means it has not completely utilized or harnessed the available potentiality of marker-assisted selection. Therefore, we took up this work. Sesame, as we know, it has a very highly edible oil and it's a very good quality, keeping quality. So it's called queen of oil seeds also. And because of industrial utility and pharmaceutical utility of both the oil and the seed, sesame is globally an important oilseed crop. But at the same time, there are still challenges that are left unmet. That is stagnated seed yield level and lack of marker-assisted selection technique, tools, and methodology in sesame. Therefore, however, we have key as an opportunity. That is now genome sequencing has been done and around two varieties from India, including Sweta and Zhongyi 13 from China and other land races have been sequenced. And there's a complete genome sequence which is revealing around 274 megabase of the gene. But SSR and SNP markers are still under the process of development. So along with SSR and SNP, there is a need to convert germplasm resources into genetic resources. Therefore, since genomic assisted breeding has to be introduced into sesame crop improvement, yield rate mapping has to be done. And that needs mapping population. As we were listening in the morning to our Honorable DDG Crop Science as well as Director of NBPGR, there is a need for contrasting parent to start with to exploit any genomic resources for that matter. So this is the material we use. We started with 3,000 lines. They comprises land races, varieties, and breeding lines. And 3 to 5 generations, we advanced them to stabilize genetically. Why 3 to 5 generations? Because even though classically sesame has been known to be self-pollinated crop based on flower biology, in the presence of honeybee, the extent of crossing can go up to 80%, as it is reported in the literature. And after that, we followed the crop management with the standard 45 centimeter row to row spacing and 15 centimeter plant to plant. and recommended package of practice for nutrient supplement, pest and disease management. And we used single row, so augmented block design was used, and the most reliable coefficient of variations was used in our study to decide whether to further advance by selfing to genetically stabilize or to conclude that it is genetically stable. If the CV was more than 5, we undertook selfing and advancing to the next generation. Why we have done all this? Because genomics assisted breeding requires mapping of yield contributing traits, and there is a need for mapping population. So we need for contrasting parents. This is the overall overview of the work that we done. We raised in the field around 3,000 germplasm lines, and then these were analyzed for exhaustive morphological data. Then we analyzed the diversity with respect to yields. This you can see, this is the diversity. And this cluster divided the 3,000 germplasm into 2 groups. This is the high yielding, this is the low yielding. Then from among the high yielding, we focused on the morphological traits that are contributing to the yield. Similarly, among the high yielding, this one. For example, you can see here, variability within the line after genetic stabilization was fixed like this. And similarly, if you take, for example, number of capsule per plant, it varied from this sparsely distributed low number of capsule per plant to highly dense distributed and high number of plants. Now the question arises, when we are focusing on yield contributing traits, whether we ignore variability for other traits. Yes, because suppose if you consider here, I cannot compare the number of capsules per plant between a branching type and non-branching type, because the branching behavior itself is another trait that interferes between the QTL contributing to number of capsules per plant and the phenotype number of capsules per plant. So to avoid that intervening and interfering variability and traits, we focused, for example, with respect to number of capsules per plant, we only compared between non-branching types. You can see here. Whereas if you come to the plant type, for example, you see here plant type. Then when we stabilized it, the variability was within CV5. So overall what we done, within the germplasm variability was divided into 2 groups. For example, with respect to plant type, I am explaining, same thing was followed for all the traits. The contrasting lines were selected. That means the shortest possible plant, the line with shortest possible plant, and the line with the highest possible plant, tallest possible plant. Then after selecting, we selfed and raised next generation, and we looked for variability. If there is a variability in the line having the lowest value, we chosen for next generation the lowest plant, plant having the lowest value within the germplasm having the lowest value. Similarly with the highest value. This was followed for nearly 17, 18 traits. And if next generation also we analyzed CV. If the CV is more, again we self-put. For example, in this case, we self-put lowest one. In the other side, in the line having highest value for plant type, we self-put highest plant within the line. Like that we advanced and it took 3 to 5 generations depending upon the level of heterozygosity to genetically stabilize. And what is the statistical dictum that it is genetically stable? It is CV having less than 5. Here, when we say CV, we did not go for sample variation. We went for population variation because in a line around 150 plants, we took that 150 plants were representing the particular line. So we took entire observation. So we used population mean for arriving at CV, which is more reliable and also confident. So like that, these are the traits we genetically stabilized. Now each one of these, for example, if somebody want to map internodal length variable ET or QTLs contributing to internodal length, one can use this one and this one. This is the actual genotype. and this is the parameter, and these are the values. This set forms a contrasting parents for developing mapping population for tagging internodal length contributing QTL. Similarly, this one for seed weight per plant QTL. Similarly, this one for molecule per capsule QTL. This pair for capsule per axil. This pair for primary branches. Similarly, for secondary branches, basal bearing, capsule length, capsule width, number of capsule per plant. So plant height, days to flowering, fresh weight, dry weight. 1000 seed weight, oil content and harvest index, we did not study from the beginning with 3000 lines because this was not our objective. Our objective was to tag the QTLs contributing to yield contributing traits. Therefore, after selecting for all these, within the set of these studied core selected
Kumar Swami, we are not able to see the slides. The quality of slides are very poor for everyone. Everybody is complaining about that.
Is it okay, sir, now?
No. It's not at all visible to anyone.
I don't know what to do. It is very clear, sir, here.
For the transmission, there is some problem. Kindly excuse.
During transmission, it is a problem, and afterwards, is it when stable, it is visible, sir?
Therefore, I would ask you to only conclude your work.
Okay. Is it now visible, sir?
No. All are complaining. There is some network problem.
Okay, sir. My idea is visible, sir?
Give your audio conclusions on this.
Fine, sir. And whatever we collected, we analyzed for oil content, then harvest index, and also for biotic and abiotic stressors. We got around 7 lines which showed tolerance to fill loading. The incidence was 20 to 30 when we graded it. And also for anti-gaster insect tolerance, we got 13 lines among these 4 sets that we developed. And for physiological efficiency in terms of leaf temperature and SPAD analysis, we got 11 genotypes. Therefore, what we can conclude from my work is that marker-assisted selection was key to break yield barrier in sesame, which otherwise have been stagnated for decades. Then marker-assisted selection requires mapping of yield QTL. These yield QTLs can be mapped if and only if there is a contrasting phenotype for each of the yield contributing traits. So in our study, we developed genetically stable and phenotypically contrasting genotypes for each of the yield contributing traits, and they can be effectively used for tagging or mapping of yield contributing traits in sesame. Thank you, sir. Thank you very much. And
thank you, Kumar Swami.
And
I am telling all these listeners, kindly note his phone number or email address, which he can tell to everybody now.
Yes, sir. Sir, before that, I'll acknowledge ICR, Indian Institute of Oilseed Research, and ICR, and Ministry of Agriculture and Farmers Welfare for funding this work. And I thank Director of the Institute, Admin Finance, and all the staff. And also finally, the team members. I am PA of this project, co-PA, pathologist Santa Lakshmi Prasad, entomologist Dorai Murugan, physiologist Dr. Ratna Kumar, and co-authors of this work, Jawaharlal, Ramya, and ARG Ranganatha sir, who shared their materials in the beginning, the founding lines of 3000 germ project. And my email ID and all I will share, sir. My email ID is hhkphd, hhkphd, H for Haryana, K for Karnataka, P for Punjab, H for Haryana, D for Delhi @gmail.com. My mobile number is 898-567-1234. Same thing I will be writing also in the comment box.
Thank you so much. Thank you.
I don't know what is the problem.
Okay.
My slides could not appear.
Anyway, thank you, Kumar Swami.
Thank you so much, sir.
Now the next speaker we have is Aparna Velluru, Dr. Aparna Velluru, who is a scientist at the Division of Floriculture and Landscape ICR at USA. She is going to speak on the diversity of 21 wild rose species. Now, may I call upon Aparna to kindly take the window.
Hi, sir. Hi, sir. Good afternoon, all.
Yeah.
Go ahead, Dr. Aparna.
Good morning, sir. Respected speaker, chairman of the session, and all participants. The current presentation, I'm giving a small presentation on the population structure and diversity and genetic relationship of 21 wild rose species grown in India based on SSRs and morphological markers. As we know, floriculture is an important segment of agriculture with enormous growth potential. And this worth of floriculture market in India is $130 billion. And this value is increasing rapidly with CAGR growth rate of 20.1%. And as all we know, rose is an important commercial floriculture crop, and this is priced more for its flower. And there are other traits in case of rose other than the flower characteristics. There are perfumery characteristics and as well as nutraceutical compounds. Rose hips are known to have highest vitamin C content, which is having 30 to 300 milligram per 100 gram of hip. And in addition to this vitamin C, there are other vitamins like B1, B2, K, and P, and minerals, carotenoids, tocopherols, and other flavonoids, fruit acids. There are many other biologically important compounds in rose. So the wild species in case of rose is concerned, there are nearly 200 species which are distributed all over the world, mostly in temperate regions of the world. And there are about 25,000 cultivars in case of rose, which are distributed all over the world. And in case of India, when we see there are 15 species which are existing in India, which are native to India, and there are some species we have introduced from other countries, and overall nearly 25 species are grown in India, and there are more than 600 modern rose genotypes also bred by Indian breeders. Actually, according to 15-16 data provided by the Horticulture Department India, we have our area, we are nearly cultivating, our share in the area is nearly 50% in the world, but our production share and export produces less than 1%. So it's all because of our variety types, and which are little inferior than the exotic varieties. And we are because of that we are facing many other difficulties like the royalties and all from the for the export of our produce. So this is the these are the sir? Here there are some wild species so which are having so many different economically important rights. In this study, we are using those economic this sir, the voice is clear. Hello. So in the present study,
yes, madam. Now it is audible. You can go ahead, madam. Some disturbance was there, so.
Okay.
You are audible.
Yes. Okay. The objective of the present study is to study the variability present in the collections which we have done. And the second one is we have to conservation of these wild species for the future breeding uses, and studying the differences among the collected population belongs to different sections. and to identify its economic values. The planting material what we have selected is nearly 21 rose genotypes, which belongs to 7 different sections. And 2 collections we have made. Those are of unknown section, and these are all species are having one or other important economic trait. Like for example, these are the species like Gumalis and Glutinous. Some of them are having in Canada section vitamin C richness is there. In some of them are having bark character, and from Gallica species we can see the fragrance, right. In case of India, fragrance as well as dwarfness traits as well as flower color also we can see flower type. And in case of pink heliconia, yellow color and toneless characters in the Rosa lutea. Likewise, inside there are species suitable for roost tracks and as well as the species with toneless character. And the last 2 unknown classified species are like Kakinada red. This is a species which is predominantly grown in southern peninsular India, which is fragrant cultivar with a pink color character. And so for this study, first 14 morphological traits we have selected to check the morphological diversity. Those characters are like a plant growth habit, height, and shoot anthocyanin coloration, and hue of the coloration, prickle color, predominant shape of the prickle, as well as leaf shape, leaflet shape, and serration of the leaflet, pubescence, and green color, the intensity of the green color. These are the traits we have taken for the characterization of the genotypes, and then followed by the molecular characterization. So genomic DNA we have extracted, and 48 different SSR markers we have used for the characterization based on molecular study. So these selected markers are identified from the Rosa hybrida and Rosa palulida species, and these 48 markers we have used, and then so these are the markers which we have used in the study. And data analysis was done in case of morphological as well as based on molecular markers. And here we can see the dendrogram based on the morphological markers. So this was drawn based on the Euclidean distance values, calculated Euclidean distance values. So we can see here we can see that genetic distances and genetic variation in different species. So there are species from different sections which are having similarity. So it is systematically it is deviating from the conventional classification of the rows in this study. So likewise, in molecular, based on molecular characterization, out of 48 SSRs, we have identified 31 markers with polymorphism, and the remaining 18 were monomorphic, and a total of 97 alleles were detected with the average allelic loci 3.12 with average 3.12 alleles. And the number of alleles identified in each primer range from 2 to 5. The band size is ranging from 96 base pair to 316 base pair in this species. And there are some markers which are highly useful based on this study. It can be used further useful for the identification of rose white germ blossom. So this is the dendrogram drawn based on the molecular data. Here, this clustering we can see. Even here also there are some species, and even from same, they say some of the students, they say they are clones of the Damascene, like for example Jwala and Himrose. They are said to be clones of Damascene, but here we can see the genetic differences in these. Though we see the morphological genetically they are very different, and difficult to combine these two characteristics also, fragrance compounds also. Like here we can see the genetic relationship. This indica major and brunine. These two are very similar, but they are from different sections. So likewise, we can see in all 21 species we have revealed the genetic relationship, and after in order to support the data, we have gone for this Bayesian model approach. Those structure analysis we have done, and in this we have identified 3 subgroups. So here in the 3 subgroups we have seen in the first group there are almost fragrant genotypes, and all are grouped in one subpopulation, and remaining 2 subgroups were identified with a different population, and it is having more of similarity identified with the dendrogramic results obtained with the molecular data. And here we have studied the genetic variability, different genetic variability parameters section wise. And here more of percent polymorphism are obtained with the Gallicinae, Indicae, as well as synstyler species. And a very less polymorphism we have seen as well as citrosagacity in Pimpinellifolia, followed by the Cinnamomea. And these are the species with as out of 21, there are about 18 species with a highly economic importance. So this can be this study can be useful for future breeding or studying the systematics in detail, because here we have seen more of intersectional interspecies intersectional species similarity. So further we need to update the conventional system what we have present. The similar way, this data generated from this study will be helpful for identification, documentation, as well as conservation of genotypes. Thank you, sir. For this study, I acknowledge ICR, NBPGR substation Shimla, as well as ICR, NBPG main station New Delhi for support, for sharing the plant material, as well as for sharing the work. Thank you.
Okay, thank you. Now,
something was shared by somebody, sir. This is some. Something is coming here.
Friends, please don't disturb.
So thank you, Aparna.
Thank you, sir.
Now our conservation of long-term conservation of Ira Vaidya Malhotra on based on the cryopreservation technique in Humulus lupulus. Please go ahead.
Good afternoon, sir. I hope I'm audible and my screens are visible.
Yeah.
Yeah. Good afternoon, respected chairperson, all the respected dignitaries and delegates of the conference. I'm thankful to the organizers for giving me the opportunity to speak on gene bank and a multi-crop in vitro gene bank housing more than 53 genera and 138 species in the in vitro cultures, and in the cryogene bank, we are Asia's largest cryo bank with more than 11,000 accessions being conserved as in different forms as seeds, embryos, embryonic axis, pollen, dormant buds, and meristems. So this is an overview of how we conserve the germplasm in our gene bank. When we get it either by import or by germplasm collection, it is first quarantined and tested for its health. If it has some disease, the disease elimination takes place. Otherwise, if it is a vegetative propagule and it is healthy, it is introduced into the in vitro gene bank after in vitro culture initiation and multiplication protocols being developed. Or if it is a non-orthodox species, its material is conserved directly as in cryopreservation in liquid nitrogen by testing its moisture content and testing its desiccation, response to desiccation and standardizing protocols therefore. And for the vegetatively propagated propagules, also we are developing long-term conservation protocols. All this has a proper database management, is the data and the information is properly and available in databases which are publicly available. And the material is also exchanged as in vitro cultures. Both the in vitro conserved and the cryo conserved material is regularly tested for its genetic stability, because we want to ensure that our developed protocols do not cause any instability in the conserved germplasm. Now, talking of the work that we carried out, that is hops, it is an industrially important crop and it is grown globally in a large number of countries, as can be seen in this graph. The United States, Canada, Australia, even Russia and China being major producers. And it is actually very valuable for the brewing industry, because its female flowers have some essential oils which are responsible for the unique aroma of beer. And it has also been used in the traditional herbal medical systems. India also has been producing hops in the temperate regions, but the cultivation has suddenly dropped. And recently in the year 2019, there was this report in one of the newspapers that hops cultivation is becoming extinct in India. We have lost the germplasm that was there, and hence the need of conserving it now. As you can see, in 2018 alone, India has imported hops worth 6.42 million. So we have lost the germplasm and it is steadily declining. So there is an urgent need to develop protocols for its safe conservation. Now, because it is vegetatively propagated and its conservation is mainly aimed at by using cryopreservation, which is the long-term conservation at ultra-low temperatures of liquid nitrogen. And this developing protocols for this method of conservation requires a species-specific approach because each species has its own physiological and biochemical uniqueness. And so protocols are designed accordingly. And it has to be very precisely done because cryopreservation has a threat of ice nucleation and dehydration, which can lead to injury to the cells, and we may end up damaging the cells rather than conserving them. So the process of optimization of protocols is a very, very precise method, and it takes a lot of effort to devise very accurate and reproducible protocols. Now, when we talk of conservation strategies for hops, globally, hops germplasm has been cryoconserved using the encapsulation dehydration techniques only, and these are conserved at the USDA Gene Bank in Fort Collins. There are no reports on vitrification-based cryopreservation techniques for hops. So keeping this in mind, we designed a hypothesis like we should going for conservation of hops germplasm using the newer vitrification-based methods. So for this work, first we needed to standardize a shoot multiplication medium. Then we needed to optimize the pre-culture duration that those shoot tips will withstand. And after that, we have to optimize how much of a dehydration using the cryoprotectants can these shoot tips withstand. So the first step was the development of a multiplication medium. We tested different multiplication media, and then one of the media with the TDZ gave us very good shoots per explant, 6.5 shoots per explant. So this medium was selected for rapid multiplication. And we were able to root all of these multiplied shoots as well, and we were able to transfer them to our fields also. So once we had optimized a multiplication medium, we were sure that now if we culture our shoot tips on this medium, MS medium supplemented with TDZ, our shoot tips are going to grow vigorously. The next step that we had to take up, which is for any cryopreservation protocol, is to understand how much of an osmotic dehydration the plant will be able to withstand. Because the main aim of a cryopreservation protocol is to remove out extra water from the cells so that when we freeze it, the extra water does not form ice and damage our cells. So the first step here was to standardize the pre-culture duration, and we cultured our shoot tips on a high osmoticum medium of containing 0.3 molar sucrose, and we cultured it for a number of days, and we tested its survival rate of the shoot tips for all of these days. And we found that shoot tips which were grown on pre-culture medium for 1 day, they had good regeneration, that is 100% survival and regeneration. And the regeneration started to decrease as the number of days for pre-culture increased. Whereas when the moisture content of these shoot tips that were grown on this medium were checked, it was found that the moisture dropped to about 75% on day one itself, and it's remained stagnant. So that that means even if we cultured the shoot tips on high sucrose for increasing number of days, the moisture content did not show any change. It was stagnant after day one of pre-culture. It was stagnant at 75 degrees. So based on this, we selected 1 day pre-culture as a starting method for a cryopreservation protocol. Now the next step was to actually devise the dehydration regime using PVS2. And we tested 3 different techniques here. We tested the technique of vitrification. The second was of encapsulation vitrification, and the third was of droplet vitrification. So both of or the principle behind these techniques is more or less similar. But what happens in vitrification is the isolated shoot tips, when they are pre-cultured and then they are loaded with a loading solution containing high osmoticum of sucrose and glycerol. After that, we just cryoprotect them with PVS2 solution, that is the plant vitrification solution. And then we put them into cryo tubes and we directly put the shoot tips into liquid nitrogen. And then when we are to check for our protocol, we rewarm them and put them for regrowth. So this is the basic vitrification procedure. When we stepped on to the encapsulation vitrification, here after isolating our shoot tips, we encapsulated them in alginate beads. And those alginate beads were then actually treated with the loading solution and then with PVS2, and then they were cryopreserved. So the difference between vitrification and encapsulation vitrification was the incorporation of the alginate encapsulation of our shoot tips. While the latest technique that is now being used is droplet vitrification, and here the isolated shoot tips were again treated with loading solution and then cryoprotected with PVS2, and then they were shifted onto small aluminium strips in drops of PVS2, and these aluminium strips are then put into liquid nitrogen, that is directly plunging them into liquid nitrogen. So the difference that this technique offers over the others and the advantage is the ultra high rapid rates of cooling, because the shoot tips are directly immersed into liquid nitrogen on these aluminium strips. The rate of cooling is very, very high. So ultra high cooling rates leads to lesser damage to the shoot tips, and that is why the recovery rates are also higher. So this is what happened when we conserved our shoot tips. With vitrification, our shoot tips were able to regrow when they were put in liquid nitrogen after treating with 30 minutes of PVS2. So yes, the protocol was working. As you can see in these diagrams, these arrows show the growing shoot apices. And the shoot tips that had been conserved had been treated with PVS2 for 30 minutes showed growth. So the protocol was working. We have a protocol, vitrification-based protocol for hops now. When we improved this protocol and we started working on encapsulation vitrification, we saw that the survival percentage increased to 20%. That is when those shoot tips that were treated with PVS2 and immersed in LN, that is liquid nitrogen, 30 minutes of exposure to PVS2, the shoot tips here had 20% viability. So there was an improvement. And this is how they showed growth after this. These are the LN treated shoot tips in this image, and they were growing. But a major improvement was found in droplet vitrification, where there was a 50% survival rate was achieved. That was a very high success rate. Yes, sir.
Yeah. Can you please conclude?
Yes, yes. So droplet vitrification gave us a very high rate of survival, that is 50%. And this now protocol we have tested on 8 different accessions, and we are getting very high survival rates with all of these accessions. So this protocol is actually very, very reproducible. And here is a comparison that showed that the droplet vitrification has given us very high survival rates, more than 50%. And this optimized protocol is now applicable to most of the hops collections that we are having. So as I conclude this work, as I said, there are no reports on droplet vitrification based conservation of hops till date. So this is the first report of conservation of this germplasm using droplet vitrification. And the rate of regeneration is very high, that is higher than 50%. And it is applicable to different accessions. So it can be used for large scale cryobanking of most of the hops germplasms. So that is our major achievement, that we are the first people to report this successful protocol for hops. And I would like to acknowledge the director for providing the research facilities and the team for working together to bring this protocol forward. Thank you.
Thank you, Ira. It was a nice presentation.
Thank you.
Kindly provide your email so
Yes,
that for any interaction, the
yes,
audience will have it. So with that, we'll conclude this presentation. And now we'll call upon the next speaker, that is Dr. Gopi R, who will be speaking on diseases resistance in flowering clones of sugarcane germplasm. Please, Dr. Gopi.
So slides are visible, sir?
Yep. Go ahead.
Respected chairman, co-chairman, reporters, and other dignitaries participating through online. Good afternoon, all of you. Now I am going to present my topic of presentation is diseases and disease resistance of flowering clones of world collection of sugarcane germplasm. I am working at ICAR Sugarcane Breeding Institute Research Center, Kannur. So coming to the introduction, sugarcane is one of the important cash crops belonging to the family Poaceae, cultivated in the tropics and subtropics. There are 6 species: Saccharum officinarum, Saccharum robustum, Saccharum edule, Saccharum barberi, Saccharum spontaneum, Saccharum sinensis are the important species. Sugarcane Breeding Institute Research Center, Kannur was established in 1961. It maintains largest collection of sugarcane germplasm, around 3,373. In that, the 5 species itself includes 1,360 clones. Other than that, we are also conserving Indian hybrids, foreign hybrids, IA clones, and allied genera of Naringa and Erianthus, and also others. So coming to the flowering, flowering is an important physiological phenomena and is very much important for breeding new varieties. However, flowering is not very common in all the sugarcane germplasm clones. If we take out of 974 clones belonging to 4 species, we found only 137 clones were flowering. It comes to around 14.04%. So among them, Saccharum robustum contributes around 40 clones out of 145, and coming next to that, Saccharum barberi, it contributes 26% clones which were found flowering. So objectives are identification of flowering clones in Saccharum germplasm, identification of disease in flowering clones, grouping of Saccharum species based on resistance. Main aim is that identification of disease resistant clones for various diseases. So these are the list of flowering clones. Like 66 clones belong to Saccharum officinarum. Saccharum robustum contained 58 clones, and Saccharum barberi 11 clones, and Saccharum sinensis only 2 clones were found flowering clones. So these are the diseases occurring in germplasm clones at Kannur: smut, wilt, pokaboing, cetrot, fall smut, rust, ring spot, stalk rot, sugarcane bacilliform virus, and yellow leaf disease, and banded sclerotial disease. So these are the major diseases and time of occurrence. Viral diseases we can find entire season of the crop, whereas leaf spots and ring spots and also pokaboing we can find from June to December. Rust, we have seen from July to December. Smut, it appeared in September to December, and stalk rot normally it comes at the end of the growing season. So ring spot, smut, rust, stalk rot, pokaboing were the major diseases. They were studied. All the flowering clones were studied for these diseases, and also some of the diseases are not very common, like red rot and smut were studied under the laboratory condition by artificial inoculation, controlled condition. So coming to the Saccharum officinarum, like 38 clones were found resistant to ring spot disease, and 21 clones were moderately resistant, and only 7 clones were susceptible. And for rust, all the clones were found resistant except IJ76521. For pokaboing and stalk rot, all the clones were resistant. In case of stalk rot, only 2 clones were found susceptible, that is Chapina and 21 NG10. In case of red rot, it is a very important disease in sugarcane. Baragua was found resistant, and 57 NG78 were moderately resistant. All others were susceptible. In case of smut, only 4 clones were found resistant, and 3 clones Azuldi, Casa, Chapina, and 21 NG10 were found moderately resistant, and others were susceptible. The important clone Velli, from that first hybrid core 205 was developed, also found resistant. Next is Saccharum robustum. For 4 important diseases like ring spot, rust, and pocoboing stalk rot, all the clones were found resistant. In case of red rot, all the clones were found susceptible. For smut, the clones belonging to IJ and that is Indo-Java and New Guinea clones, around 10 clones were found resistant. Rest all others were susceptible. In case of Barbary, like ring spot, rust, and pocoboing stalk rot, all the clones were found resistant. In case of red rot, only Padri was moderately resistant, others all were susceptible. For smut, Kansar and Mankia were resistant, and Kevali 14G and Nargori were moderately resistant, others all were susceptible. In Saccharum sinus, only 2 flowering clones were noticed. So all the 2 clones were found resistant to ring spot, rust, and pocoboing and stalk rot. Whereas all the clones were found susceptible to red rot. Only Tekachung Island was found moderately resistant to smut. So among the diseases, actually, because of high humidity and rainfall, ring spot was very prominent in Kannur regions. So this was further studied. So 4 clones like Mongetgam and Naz and IK767 and white transparent were severely infected. Here, actually, this disease was further studied by using both flowering and non-flowering clones. All the clones were subjected to non-parametric test. So in that, we have found that flowering clones were more susceptible and showed a mean rank of 431.67. It indicates that these clones, flowering clones, were more susceptible to the ring spot when compared to the non-flowering plant. Rest all other Saccharum species were found resistant and incidence was very less. So coming to the conclusion, among the diseases, ring spot, though it is a minor disease, all the clones were this was appeared in all the clones. Most of the clones were found resistant to this disease. However, in non-parametric analysis, flowering clones were found more susceptible. Although rust is prevalent in germplasm clones, only 1 clone among the flowering clone was found susceptible, that is IG78521. Pocoboing and stalk rot was not observed. However, only 2 clones were found infected by stalk rot in case of Saccharum officinarum. Out of 197 clones, only Baragua was resistant to red rot, and 57 NG and 78 red, and Patri was moderately resistant to red rot. A total of 23 clones showed either resistant or moderately resistant reaction to smut. Thank you.
Thank you. You have done the job in right time. So
thank you, sir.
Kindly give your email to other participants.
Sure, sir. Sure.
Please. Now we have the last speaker for the day. That is Dr. Ganapati. Now he is a senior scientist at Rajendra Nagar Institute of Millets, and he will be speaking on induced mutagenesis for creation of new variability in finger millet. Now, can I call upon Ganapati?
Yes, sir. Respected chairman, co-chairman, and all the delegates of the conference. Good afternoon to everyone. Sir, I am also very happy to see Kulkarni sir. He is our teacher, and very proud of you, sir. Sir, coming to the topic of my oral talk, induced mutagenesis for creation of new variability in finger millet. Coming to the importance of the crop as such, this finger millet is one of the important millet among the major millets next to bajra and sorghum. And this cultivated finger millet is a tetraploid. Globally, it is cultivated on an area about 2 to 2.5 million hectares, of which 50% of the area is in India, that is 1.2 million hectares with the production of 1.8 tons. And coming to of this 1.2 million hectare, the major area is from Karnataka, from one single state, and the productivity is was 1660 kg per hectare. So one point I would like to mention is that as compared to other fine cereals, in general millets they have lower productivity mainly because they are mostly grown under marginal conditions with less inputs, where other crops cannot be grown profitably. So coming to the unique selling proposition of finger millet, as you know, in general, there are 2 things for which millets are known. That is, one is nutritional superiority, and the other thing is the climate resilience. So in this recent times where there is a lot of talk about global climate change as well as rising lifestyle diseases, these millets have a lot of bargaining power. So coming to finger millet as such, as you can see here in the left-hand side, The finger millet, it has the highest calcium, nutritional, it has the highest calcium, that is 340 mg per 100 gram, which is about 10 to 15 times of that of any cereal. So therefore, this particular finger millet is well known from traditional times, especially as a weaning foods for as well as for mothers, especially for development of bones and teeth. Apart from that, this finger millet, it has numerous nutritional properties, like as you can see here, it has anti-diabetic property, anti-cancerous, and so many other properties. So coming to the actual work done, this particular mutation breeding we have initiated at IMR from during 2016 after the Directorate of Southern Research got upgraded to Indian Institute of Millet Research. So we were looking into other crops as well. So in finger millet, generally, as you know, and it is a case in other small millets as well. They are having very small sized florets and clistogamous nature of anthesis. And as you know, in finger millet, they flower in the early morning, that is in 2 to 3 a.m. Morning, as compared to other fine cereals where they flower around 7 to 10 in the morning. So therefore, recombination breeding has become a major hindrance. So, what we did is that alternately we looked for a mutation breeding as one of the approach for creating new variability in finger millet. So, although there are a good number of varieties released through recombination breeding, but recombination breeding in finger millet has not been exploited to the fullest extent as that in other fine cereals or millets. So, although we are looking into mutation breeding as one of the approach, but we are at IMR, we are continuously working on understanding the ancestral behavior as well as the improving the hybridization techniques. As you can see in the top picture, so we here what we did is that we standardized one technique that is called the time-lapse method of microscopic method, where we made an arrangement for a camera to continuously capture photograph of the ancestors. And then a series of photographs were taken in the early morning, and then we analyzed the photographs, and we could have a better understanding of the ancestors. So one point I would like to mention here is that although finger millet is a clistogamous, has a clistogamous nature of ancestors, but based on the weather conditions, by altering the humidity or temperature, we can also convert into chasmogamous mode of pollination, where we can have more outcrossing and we can have a better recombination procedure. So the bottom picture, it shows the existing method of emasculation method, which is the contact method. But using this method, currently we have success about 2 to 3%, and trying to continuously improve upon the percentage of recombination. So coming to the actual workflow, what we did in mutation breeding, we have selected 4 varieties which are otherwise having very good yield, but they have like it is having
Please switch
like
off your
maturing
modes. In the background, there is a noise. Naneshwar.
Can I continue, sir? Can I continue, sir?
Yes, we can continue.
Please continue.
So coming to genetic materials, we have selected 4 varieties which have specific weaknesses. So generally, as you know, in mutation breeding, we try to improvise some specific weaknesses in these varieties. So here we have selected 4 varieties, GPU 2867 and 1 germplasm line for specific traits. So this is the usual flowchart which we followed in the mutation breeding. It is as usual as in any other crop, but 2 points I would like to mention is that if you are taking up a mutation breeding, main thing is that you need to raise a very large population size. And here in this particular crop, since it is a tetraploid, the release of variability, it extended up to M3 and M4 generation, and thereafter we followed the year 2 row progeny, and then we could stabilize all these lines for different traits. So these are some of the traits which we are targeted for improvising. One important trait is non-lodging, which is an important trait in finger millet. Mainly we are looking for this trait for which is amenable for mechanical harvesting, and we are now trying to promote finger millet in Rabi season as well. So therefore we are looking for temperature and moisture stress tolerance in finger millet. So these are some of the M1 mutants, like as you can see here, these are the chlorophyll mutants, which we could confirm that mutation has taken place. And in M2 and M3 generation, we could see some of the mutants like for high tiller, early flowering, and so on. So coming to currently we have around 62 stabilized mutants for various traits. So this is one mutant which is GPU 67 mutant. The right hand side you can see the original variety, and this original variety, it has to certain extent scattering, and it is susceptible to blast. So we could improvise this variety, and we have currently a variety like a genotype with better finger length, and then with non-lodging. And one important trait is that we have green leaf at maturity, which is an important trait, especially when considering the fodder value. So apart from that, we are having MR6 mutant. The normal variety is maturing is in 130 days. But it has a very high very high yield. We could have now a mutant with at least 8 days early as compared to the MR6 with the same yield levels. And apart from that, we have GPU 28 mutant with more number of fingers as compared to its original variety. And here we can see that we have a mutant with a very high finger length of GPU 28. Like the normal variety, it has 8 to 10 centimeter, but the mutant, it has 12 to 14 centimeter. And this is almost stabilized. You can see on the right hand side the uniform expression of this particular mutant. So apart from that, we also could isolate two mutants, that is a partial male sterile mutant. So this particular mutant is very important for recombination breeding, mainly to improvise the percentage recombination in when we take up hybridization in finger millet. So the mechanism of sterility is not yet studied, but one important point here is that since we have partial male sterility, it is also easy to maintain, and therefore we will also be using it for the recombination breeding in future programs. So we also took up the stability analysis of all these mutants which we developed. Although in mutation breeding, stabilization is one of the concern. So during the last 3 seasons, we have took up evaluation in Kharif season. And here you can see in this AMI biplot, the lines which are very close to this x-axis, they are almost stable, and the lines which are on the right hand side, they are stable, as well as they are having higher grain yield. So this is again the tabular representation of the mutants with better stability as well as higher grain yield. And yeah, so apart from that, we also evaluated these mutants for post rainy, like post flowering moisture stress, and we also could identify some of the mutants which are very promising, and we will be again taking up this experiment during the current season as well. So here, this is one of the novel mutant, that is a Virescence mutant. This is actually developed at PC unit, and now we are utilizing this mutant. A very particular characteristic which I would like to mention here is that in finger millet, as I was telling, that we need a morphological marker to identify true F1s. So that particular marker which we have, that we will be able to identify at the time of flowering. But in this particular mutant, this is a Virescence mutant, as you can see here, the yellow leaves. So when this is a recessive trait, and when we cross with the normal green, we'll be able to identify the F1s at the very early stage, that is at the seedling stage. So this has a major role in hybridization programs. And now we are trying to diversify in different genetic backgrounds. So again, we are also trying to, we are also successful in using this G1 mutant by crossing with the wild species. We could successfully develop one with Illucin jageri, and then using the G1 mutant, we could confirm it. And then presently we have F2 and F3 generations, which are promising for some of the traits which I will not be elaborating in this presentation. So again, we have now 2 different mutants, partial male sterile mutant and G1 mutant. We are trying to combine these two in a single genetic background. And then we'll be, actually we wanted to study a model system for heterosis breeding in finger millet, so that if you could combine in a single genetic background, we could take up heterosis study, and then we could also develop a model system for heterosis in finger millet. So apart from that, as I was telling, that blast is also a major concern in finger millet. We have evaluated all the mutants for the blast disease, and then we could identify some of the lines which are promising for blast disease. And we have one, ICRISAT, they identified some of the SNP markers which they were able to clearly differentiate between the resistant and susceptible lines. So these markers will be genotyping with the mutants, and then we'll be trying to identify whether it is these markers will be able to identify the resistance genes, as well as we'll be able to identify if some new genes are responsible for resistance. So coming to conclusion, just want to tell that mutation breeding is a old technique, but still it is a very good technique, especially when we want to create new variation which is not existing in the germplasm lines. And together in recent times, along with genomic tools, we will be able to identify new genes as well as gene functions. And in this present study, we identified some stable mutants which we'll be testing for direct release as a variety, as well as we'll be registering some of the lines for at NPPGR for specific traits. And we also will be taking up molecular genetic studies, especially for blast resistance. So I conclude my presentation. Thank you, sir.
Thank you, Ganapati. It's a nice presentation. And
thank you very much, sir.
Thank you. Nice. So we have come to the end of the oral presentations from the posters and also the particular session on the plant genetic resources, the key address, and all the speakers. So it was an opportunity for me after all a lapse of almost 8 years to now we are into the not from seminars but webinars. And there is a lot of to imbibe from most of the studies which we have heard today. And I thank each one of the speakers for giving a wonderful presentations. And I thank the University of Horticulture Sciences also for giving me an opportunity to chair this session. When I looked at the brochure, practically half of the participants and speakers were my own students or colleagues. This what made me to accept their invitation. And needless to say, Fakhruddin did not allow me to escape. So I thank one and all, including the Vice Chancellor, Indiresh, who also happens to be my student. Thank you once again.
Thank you, sir. Thank you, sir, for chairing the session and for the smooth conduct of the first session, where we had a very informative lecture facilitating all of us. I would like to thank profusely the chairman of the session, Dr. R.S. Kulkarni, sir, and co-chairman of the session, Dr. Dhananjaya, sir. We are very privileged, sir. Our professor is here. Thank you. Thank you very much, sir. Hello, sir. Dhananjay, sir. Hello, Dhananjay, sir.
I will not be given a chance to speak.
No, sir. Please, concluding remarks, sir.
Okay, sir. First, I thank my teacher, Dr. R.S. Kulkarni, sir, who chaired this session very efficiently, managed the session very efficiently. And definitely because I involved as a co-chair and after listening to Dr. Or NBPJ Director Kuldeep Singh, sir, and especially Venu from IRI, and that ICRISAT Gene Bank head also. What I found is, it's my observation, there are international agencies exclusively working on plant genetic resources, and at every nation level also there are agencies, premier agencies involved in PGR. And there are some agencies, CJR, they also do, it's not exclusive for PGR, but they do also PGR activity, even that repatriation of collected local lines to the farmers. What I found is some of the responsibilities are overlapping, and what are the legal binding on non-PGR institution in sharing the germplasm acquisition or its commercial exploitation. I found that there is some overlapping of responsibilities between these two kinds of institutions. And the second thing is from Indian perspective. There are quite a few legislations, particularly the Biological Diversity Act 2002, the PPFR Act, the Geological Indication of Goods Act 1999, and also the Seed Act. There are some practical issues at the, what I can say, at the users level when it comes to accessing the germplasm, the commercial exploitation of the germplasm, and also benefit sharing among different stakeholders. So we, especially in horticulture crops, we have come across Some of the lines we use from CJR institutions, but when there is a problem of when we go for commercials, there are problem of legislative difficulties. So there is a need to work out these overlapping of responsibilities and clarity on some of these issues what I raised will take a long way in addressing the issues that the workers working at different agencies and institutions. With these few remarks, I thank the organizers for giving me an opportunity to co-share this session with my teacher. I really owe a lot to him, Dr. R.S. Kulkarni sir, and also all the delegates for their wonderful presentations. Thank you, one and all.
Thank you, Dhananjay.
Thank you, sir. Thank you for your concluding remarks. Very good message we got. Thank you very much. So I would like to thank the chairman and co-chairman as well as the speakers of the first session, Dr. Kuldeep Singh sir, Dr. Vania madam, Dr. N. Krishna Kumar sir, Dr. P. Rajasekaran sir, and Dr. Venu Prasad sir. So we would like to thank you on behalf of our organizing committee. Thank you very much. Thank you, sir.
Thank you, Arnanda.
So the next. So we'll move on to the next session, the second session of the conference, that is advances in horticulture and field crop improvement. So the chairman of the session is Dr. N. Basuraj sir, former Director of Research, University of Horticultural Sciences, Bagalkot, and co-chairman of the session is Dr. Ramesh S., professor from Department of Genetics and Plant Breeding, University of Agricultural Sciences, Bangalore. So Dr. N. Basuraj, a distinguished personality, our former Director of Research of our university, who has served in various capacities for 36 years in University of Agricultural Sciences and Bagalkot. He has served as a member for various councils and committees, awarded with several recognitions and awards like UNDF Fellowship, NCF Fellowship, and is also responsible for the award of Chaudhary Devilal Group Award for Best ACRP and Potato, and is also involved in the development of 11 varieties in various horticultural crops, including coriander, fenugreek, potato, chili, and French bean. And our co-chairman, Dr. N. Ramesh sir, is a very distinguished teacher in Department of Genetics and Plant Breeding, who has been awarded with ICR Best Teacher Award during 2017, and he has also received a Best Researcher Supervisor in 2020, and who has many publications in peer-reviewed journals to his credit, and he has also served as visiting scientist at ICRISAT, Hyderabad.
Hello.
Dr. N. Basuraj sir, can you hear me? Hello. N. Basuraj sir. The speakers of this session are Dr. I.S. Katgeri sir, editor from University of Agricultural Sciences, Dharwad. Next is Dr. A. Mohan Rao sir, University of Agricultural Sciences, Bangalore. Dr. J. Dinakar Radhika and Dr. Vagesh Babu S. Hanur.
Hello. Madam, am I audible? Hello.
Basuraj, sir, Basuraj sir.
Hello, Basuraj sir. Kindly unmute.
Hello.
Am I audible?
Yes, sir. Some disturbance is there, sir.
So if you have your mobile devices or anything, kindly keep on this.
Okay. Good afternoon, everybody. Good afternoon, everybody.
Good afternoon, sir.
Good afternoon. Good afternoon, everybody. Now, I very much compliment and congratulate the team led by Dr. Fakhruddin for making a very good international e-conference on advances and future outlook in biotechnology and crop improvement for sustainable productivity. The topic is very relevant for this day, and also there are already morning one session. Many people have spoken about the importance of this topic. However, coming to our second theme two, that is advances in horticulture and field crop improvement. I'm very much thankful now itself for giving me the opportunity to share this session along with Dr. Ramesh, who is a professor in US Bangalore, who is co-chairing with me. And this there are 4 invited talks are there, and 7 oral presentations are there. The keynote address is to be given by Dr. Major Singh. However, due to urgent session, he is not able to present. However, we will go to the invited talk IS category, that is who is going to speak on fiber genomics, way forward to designing a desired fiber quality in cotton, Gossypium species. In this regard, I'll take a very few seconds to introduce Dr. I.S. Ketgeri, who is also my colleague in US Dharwad. He is presently working as professor of genetics and plant breeding and editor in publications at US Dharwad. He has had a PhD in genetics and plant breeding in US Dharwad, postdoctorate in Nottingham University, UK, higher training in Texas A&M University, and he has many professional careers. 1987, he started his career. He also head the Department of Biotechnology, Associate Director of Research, RS Vijaypur, and also he has worked as a Dean, College of Forestry, Sirsi at US Dharwad, and presently working out at US Dharwad. 2 is many credentials, honors are there. That is gold medal for securing highest marks during the PhD, ICR award on outstanding interdisciplinary team research, incentive award for US Dharwad for mobilizing fund, certificate of honor from Islamic Republic of Iran, Ramnath Iyer National Award for Cotton Fiber Quality, Fellow of Indian Society of Genetics and Plant Breeding, Fellow of Society of Cotton, and many more. He's also guided many MSc and PhD students. Now I welcome Dr. I.S. Ketgeri to speak on the topic that is fiber genomics, a way forward, designing desired fiber quality in cotton, Gossypium species. Dr. I.S. Ketgeri, please. I.S. Ketgeri, please.
Thank you, sir. Thank you, sir. Thank you very much. And thank you, Dr. Fakhruddin. And also, I thank very much profusely all organizers of the seminar. Am I audible now?
Yes, sir.
Yeah, audible. Go ahead, Dr. Ketgeri. Go ahead. Poor quality. Books. You are audible. Go ahead.
Next slide. So can you see my slide now?
Yeah, we can see, sir. Please.
Okay. Good afternoon to all of you. This is a very simple but a very complicated topic. I don't know how exactly, because most of the scientists right now joined, maybe working on most of the horticulture crops, but Fakhruddin included even field crops, other crops as well. And that's how he asked me to give in my talk on even field crops as well. That's how since I started working in cotton, probably whole of my service, I thought probably this is one of the important, very interesting topic. So I would like to share some of the thoughts on fiber genomics, way forward for designing a very desired fiber quality cotton in future. See, all of you know, because fiber is most important fiber crop. Can you see the second slide? Okay. Okay.
Sir, kindly go to slide mode, sir. Presentation mode. Kindly
sure. Okay.
Okay, sir. Thank you.
Fine. So this is why it is a commercial crop. Basically, why do we call it as a commercial crop? Because it gives almost 5% to the GDP in the form of textile industry, earns around $9.85 billion export, and it has several other, apart from only fiber and textile industry, it has several alternative uses like it's in hospitals for wound dressing, pads, swabs, sanitary pads, linters, used in paper industry. Even in it is used in synthetic fiber also. It's in all this cellulosic material. And most important is it produces nearly 1.2 million ton oil, and oil is used in paint and cosmetics, also as edible oil, useful in like animal feed in the form of seed cake. Interestingly, nearly 10 million farmers are involved in producing cotton. Altogether, 40 million people involved in right from cotton growing to complete industry, provides a lot of employment opportunity. That's how it is considered as one of the important crop. See, what we have seen, what right now all of us know is, if you see the cotton fiber, cotton, how you can see the cotton plant. It's a raw cotton produced in the form of raw cotton in a cotton boll. And once it is harvested, see, it is separated as a seed and lint. If you see the proportionate, almost 2/3 is seed and 1/3 is lint. So generally, the productivity, what you can see in literature, it is in the form of lint. For example, now the present lint yielding ability of our Indian of our country is 580 kg per hectare. Means it's a lint. It's not the cotton. All the literature, all the papers, wherever you see the productivity, it is the lint, because ultimately this is raw material going for directly to the textile industry. And what you can see is a final product in the form of yarn from this lint. So this is what the process, entire process, what we see. But what we really want to know, is what is this fiber individually? You can't say individual fiber here. What is this? This is many fibers are together and made a long yarn. That's all. But otherwise, what is exactly this fiber? So if you see the surface of the cell, this is the electron microscopic picture. This is the surface of the ovule, seed, surface of the ovule. From this ovule, it's an outgrowth from the individual epidermal cells. But what is the size? What is the size of these individual cells growing and elongating? It ranges between 6 to 40 millimeter. So basically, initially it just about 10 micrometer, from 10 micrometer to 6 to 40 millimeter, and now we can see even up to 45, 50 millimeter lines, producing lines are also identified. So it means 100, thousands of fold increase in the length from original length. So that much of single cell is elongating. It's a single cell elongating. So that's how many such cells are really elongating from the surface of the cell. It is just 16,000, on an average 16,000 burst initials are coming. So if you, in terms of percentage, if you work out, just 15 to 20% of the cells, this kind of cells are converted into fiber cells. So what is exactly this individual cell? If you see this individual cell and a fiber, so it is consisting of cuticle layer, and then the inside layer is a primary wall, and then primary wall is made up of completely cellulose. You can see in the next slide what exactly it is, and secondary cell, and then you can see the inside, finally inner side. This is the vertical section of the fiber. You can see the lumen. It's completely a pith-like lumen structure. You can see these are individual fibers. This is an electron microscopic picture. These are individual fiber, what you have seen here, after elongation. See, this is the cuticle. So it is made up of waxes. And the primary cell wall is basically amorphous cellulose, esterified and non-esterified pectins, hemicellulose, proteins, and ions are present in the primary cell wall. And individual, there are fibril-like structures. They are actually wound around each other and make a very strong structure of a wall. That's what we call it as a primary wall. You can see the structure. I don't know whether it's clear or not for you people. It is woven-like structure, It's a very strong one. And then, secondary wall is completely made up of crystalline cellulose. It's completely filled with complete cellulose inside the primary wall, and it gives a strength to the fiber. Higher the cellulose filled content in the secondary, in the form of secondary wall, the stronger the fiber. This is what exactly chemistry of the individual fiber. Oh, what is the process? The process of this fiber cell elongation and then thickening is completely involves a lot of physiological processes, and basically there are involved potassium transporters are involved, sucrose, and sometimes the plasmapherescence is opened and sometimes it is closed. So that's opening and closing of this plasmapherescence. You can see here it's open now, and sometime it is closed. Again, it is open. So this kind of plasmapherescence open and transport of the sucrose ions and then potassium ions, all is responsible for creating a kind of turgidity, in the cell, and create a kind of turgor pressure, turgor built-up or a turgor pressure built-up. It determines the loosening of the primary wall, and then it is going to elongate as the accumulation starts. And there's many proteins are involved, especially expansion type of proteins are very much involved in making this cell to elongate like this. So how exactly this whole process in terms of maturity, right from fiber cell initiation to complete fiber development, how many days it takes, what are those stages? If you see, you can see in this slide very clearly. You can see here that the A and B, C and D, I have 4 different structures. A and C indicates wild type bole and fiber development. This is a wild type genotype, a normal and control type, and it's electronic microscopic pictures of different stages. And similarly, B and D, this is a wild type, mutant type. Mutant means it is not producing any fiber cells any fiber cells at all. For example, you can see here, only seeds are there. There is no any fiber at all. See the normal one, a huge quantity of fiber is present on surface of the seeds. That's how you can see the complete locule. These are called as individual locule, and it's called as bole. You can see the complete fiber in the bole. Here there is no fiber at all. It's only seeds are there, and that you can very well see with the help of electron microscope. So on the normal, and before that, what I want to say is there are clear-cut 4 stages of fiber development are there. So there's a clear-cut one is these cells to convert into fiber should elongate, and then once it is wall has to be filled up with the cellulose. That's what exactly the fiber should become completely strong because of cellulose accumulation. After that, whatever water content is there in the fiber, it has to dehydrate and makes the fiber completely dry. That process is a maturation. That's how initiation, elongation, clear-cut stages in fiber. When it really starts, before anthesis, you can see the bud size. 3 days before anthesis, actually the cells start initiating, cells start initiating, which really wants to convert into fiber. They start becoming, you can see, it's not clear, but you can see here, some of the cells which are face of the epidermal cells. Here is completely smooth, not a single cell you can see on the surface of this, right from 3 days before anthesis to 0 days, 1 day. But you can see in normal, that is you can see how the cells started, initiating from the surface of the cell. So this initiation is generally up to 1 day after pollination, although it is clearly started before 3 days pollination, but clear-cut visibility one can see is on the day of pollination or 1 day after pollination. And that's how once they started initiating, then they start elongating. Elongation is up to 10 dpa. Once after 1 day after pollination to 10th day after pollination, all these cells which are now initiated, they elongate. After that, totally nearly 30 days after pollination, this is 10 to 20 days, 29 days, and this is 30 to 45 days. This complete accumulation will take another 20 days. So by 30 days after pollination to 45 days up to pollination, the complete cellulose synthesis takes place and a lot of cellulose is accumulated to complete the secondary wall biosynthesis. So after 45 days, then the cellulose synthesis stops, accumulation stops, and then dehydration starts. So dehydration takes in another 5 days or 10 days, depending upon the variety. Generally, it is up to 60 days in some cases it is 50 days. So that's how from, if you consider from the day of anthesis to complete maturation, nearly 50 days or 60 days, in the sense 2 months are required for complete fiber development in cotton. This is what the entire process, that's how it is taking place now. So this is what the basic information, but what is exactly, since we're talking on the genomics of this fiber development, the basic information, what is another information required at this stage to understand the processes, in undergraduate courses, because most of us probably have done undergraduation in BSc Agri, generally it is taught, polymerization. In polymerization chapter, cotton is one of the standard example always quoted as an example of polyploidy. So there, probably at least some of you can remember A genome into D genome, and then AADD is a tetraploid. That's how, but at least you can remember, I hope you remember. That you can see here, in 5 to 10 million years back from a common ancestor. In the course of evolution, nearly 8 different groups, the species are evolved and grouped in different genomes, like A to K genome, A to K genome. And all these A genomes are only cultivated types. A1 and A2 are cultivated types, means these are also we call old world cotton or desi cotton. What do you mean by cultivated? Means only these two species, A1 and A2 species produce cotton fiber, and rest of these, D genome, F genome, B genome, E genome, C genome, G genome, K genome, all these species are not producing any fiber at all. They are lintless cotton, completely lintless. Of course, they contribute a lot for for different trades. They can still act as a wonderful source for different trades, including cotton fiber. For example, you can see in 1 to 2 million years back itself, this D genome, one of the D genome species, G. raimondae, is crossed naturally with herbaceum, and then it has become allotetraploid. So this one allotetraploid in 1 to 2 billion years, the original one, again in the process of it's a process of domestication. In the process of domestication, and also in the process of kind of evolution, also there are 5 different allotetraploids are developed and differentiated, and out of these 5, G. hirsutum and G. barbarissia are under cultivation. Means out of all these together 45 species, only 4 species, this and these two tetraploids and 2 diploids are cultivated. Here what I wanted to stress here is you can see the genome size. I don't know whether is it clear or not. 1,667 to 1,746 million base pairs. In case of diploids, in case of another set of diploid D genome, it is 841 to 934 million base pairs. And you can see the tetraploid 2,347 to 2,489 million base pairs, million bases. But you can see one of the diploid K genome, its genome size is as good as tetraploid genome size, 2,450, million base pairs. So this is the kind of genome size variation. But with all these wild types and these wild types, although have so much of genome, in fact, genome size is even equivalent to tetraploid, but they absolutely not contribute anything to the fiber development. So you can see the raimondae, D species, why no fiber at all. These are only linters, and this is what fiber, and then how the domestication resulted in different types, and this is what exactly cotton. And today in India, all over the world, not only in India, all over the world, what species cultivated is G. hirsutum species. In nearly 95% of the world cotton area is covered with this species, only, with this species. And very interestingly, all this BT cotton, what we see now, commercial cultivation is belong to this species only. And very interestingly, one more point just for your information, before 1940 in India, our 99% of the area is only with this species and other diploid species, arboreum species. In 1945, I'm talking in 1945, by 2000, this ratio, this is completely reversed. So this has become 90% and this has become 10%. So only advantage is their yield levels, higher yield levels, and good fiber properties. And of course, now because of the BTC, now it has become still the area of this particular cotton is more in India now. So even in fiber cotton improvement, because fiber is always, improvement of fiber quality is always a challenging for cotton builders. Why? Because see, the industries, the what you call modernization is textile industry is very fast. All these ginning machines and weaving machines, evolve, and a lot of improvements happen to meet the demands of all these, fiber quality standards and trades. Timely, the standards of raw cotton quality of raw cotton is going to be changed. It has happened, within my own service of nearly 25 years. The standards are revised 2 times in terms of length and in terms of strength. So it's always a challenging for cotton bidders because we have to be so fast, we have to release and develop the varieties which are suitable to textile industry. Otherwise, it's very difficult to meet the requirement of the textile industry. That's how, systematic breeding although started in 1992 and transgenics come. And in all these, all these whatever different techniques are there, all these techniques are now utilized in improvement of fiber quality in cotton. So that's how the targeted breeding, what we call, with the help of all this, probably will be helping breeders or any scientist working on cotton improvement, especially in fiber, will help. And what are those important techniques really helped a lot in understanding the fiber development processes. Transcriptomic studies, many transcriptomic studies, comparative genomics, functional genomics, mapping, and then now because of next generation sequencing and resequencing, a lot of information knowledge is generated. I would like to just to see a few of them because it's not possible to review all those papers. Of course, in this, we have also contributed some of 2-3 different papers on different aspects. I'll try to touch upon 1 or 2 papers of our own work as well. So in transcriptome, of course, I told you a huge number of transcriptome studies have been done. And this is our own study, just I want to share with you. In 2012, we published this. So here we try to understand key genes and pathways which are involved in regulation of all these different fiber development in cotton, what you have seen previously at different stages. So to understand all that process, we use 2 important genotypes. I mean, genotype point of view, it's MC5, only one cotton genotype. Many of you must have heard the name of the MC5, very famous cotton variety especially in South India. And it's isogenic mutant line, which is not producing any lint at all. So use these 2, and then you can see, you can't see any difference between these 2, mutant and wild type. They are absolutely perfectly similar in all respect. But the great difference, a major difference is the seed. See, this is the normal one. A lot of fiber is there, and seed also fudge is there. And after delinting, you can see, with the help of acids, when we delint, you can see seed like this. But you can see the mutant type. It's completely no fiber. You can see only a very little small amount of fiber at cellulose end. Otherwise, absolutely no fiber at all. So this is a fiberless mutant. We use these 2, and then try to understand. But I'm not going into detail. You can see the, at completely developed whole stage also, you can see the gap, no fiber at all. So here, completely filled. So this is what electron microscopic picture of the same genotype, mutant and then normal one. And similarly, we have got one mutant in diploid cotton also. That is, the previous one was tetraploid cotton, and this is the diploid cotton. So we use this diploid, at least you have a fiber, lints are there, linters, what you call fudge is there. In previous case, even fudge was also not there. So here you can see the fudging, but no fiber at all, no fiber. But otherwise, both are one and the same. But ultimately, with transcriptome studies, and then the qPCR studies also, with help of qPCR, we validated some of these studies, but this is only one figure I can show you. So what is this is the genes which are upregulated and downregulated at different stages, at 0 dPa, 5 dPa, 10 dPa, 15 dPa, 20 dPa. So what are the genes? So nearly in all these stages, in wild type, these genes are these are downregulated. So many genes are downregulated in mutant, but few genes are upregulated in mutant. But these upregulated genes are were really not checked their functionality, but what we checked was downregulated one. But these downregulated and upregulated in the normal are absolutely responsible for different functions which are involved in fiber development, especially cellulose synthesis, sucrose synthesis, accumulation of sucroses, and then formation of complete wall with different pectins and protein binding. Some of the proteins which are involved in binding. All different processes, biological functions are involved with all these different genes. And in this is very interestingly the maps. Now, so far by 2020 to recent, we have collected and reviewed nearly 180 different 88 maps are developed in cotton, and these are different SNP, these many markers, TCAPs, only 3 markers so far. So many markers are identified with so many different mapping populations. But ultimately, what is the end product? You can see here, QTLs associated with all these markers are identified. I have taken here important traits, especially fiber, since we're interested in fiber at this stage. See, so many QTLs are identified, and I'm sorry, and they're all mapped on different chromosomes. This is a number of QTLs on each chromosome, and all these chromosomes. This is for fiber length, this is for fiber strength, and these are age. All these traits are quantitatively controlled, and QTLs are well identified. Only dissatisfaction at this stage is, although so many QTLs are identified, but their utilization in breeding with the help of marker racial breeding is not yet possible because of their distance from the associated markers. They are still far away from the marker. I think still there is a need to saturate these markers to utilize these QTLs in combining and improving the fiber quality. And these are the different reference genomes right now in hirsutum, tetraploid cotton, barbadense tetraploid cotton, arboreum desi cotton, herbaceum desi cotton. So all these reference genomes data is available. So this data and all those previously mentioned, all those QTLs, probably in a couple of years will able to map, will understand actual genes and involved for all those QTLs. And probably once we know the sequences of these genes involved for all these in the form of all these QTLs, probably that will be more useful that we expect near future very soon. These are even complete wild species. Some of these wild species are also now complete sequencing is completed, and now assemblies are available for understanding the sequences of these in comparison with the tetraploid cottons, and then what is their role in improving fiber quality and other traits as well. And this is one complete transcriptome profiling data available, and this genotype is widely used in generating transcriptome profiling for different environments. See, for cold, hot, salt stress, for different traits as well. It has been well developed, and huge data is available, especially for fiber development. And now, in 2017, because genome-wide selection, when association studies when started, One interesting paper is nearly 352 diverse lines, including wild types and wild cultivated types, were completely resequenced and clustered into major three groups by PC analysis. Can you see here? These are wild type made one group, and ABI. These are the cultivated types from America, Brazil, and India. So they made one group, but there are very few genotypes taken here. But rest of them, because this is a Chinese study, and they included all their germplasm lines, and they made one complete group. So what one can visualize from this is, and the germplasms from China are absolutely not utilized in other countries. So not even a single line of germplasm is involved in these groups. Of course, wilds are expected, they made a complete group. So this kind of studies, world over, all these, whatever we can call them as a core germplasm from different countries should be studied, and that word germplasm pool can be utilized for cotton improvement. But in other crops like maize, wheat, and rice, fortunately, international institutes are there. All of them are collaboratively working, and a lot of germplasm exchange is right now available. At least it's already happened. But in cotton, there is no any such international institute where common platform is available for this kind of study. I hope in near future, if it's going to happen, and probably word germplasm can be resequenced, especially core germplasm, what you call, and that can be studied in this fashion and understand the diversity which can be utilized in cotton improvement, especially for fiber quality. From this study itself, they try to integrate some of the genome-wide association map study and then whole genome sequencing study. I just want to show you one trait here, a microneer. So this is a one trait which is now in this study, they completely identified on D3 genome where the hotspot of this complete hotspot of this particular trait is available. This is on D3. You try to recall if in a D genome is absolutely not contributing, not producing any fiber at all, not producing any fiber at all, but you can see it is contributing, although it is not producing fiber, but it is contributing to improvement of fiber quality. In D3, you can see here the fiber length. Of course, here the peak is not there. A hotspot we cannot see, but still we can see a fiber length also. There is a QTLs associated, and at this stage, and probably huge genetic variability, this is contributing for fiber length. So what I mean to say is this kind of association, genome-wide association studies will help to identify, and there when such studies are in comparison with the whole genome sequences are made, and then hotspot areas on individual chromosome can be detected to know the sequences of those genes. And but very interestingly, transgenics are less complicated. But only thing is, if you know complete gene and its involvement in some pathways of this fiber protein. So I told you a lot of transcriptome studies, and characterization, validation helped us understanding some of the important genes involved in some of the pathways. For example, one such study by Bowley et al. In 2015. So try to overexpress the uridine diphosphate glucose pyrophosphorylase enzyme. There is one enzyme, simple one enzyme, which is involved in cellulose biosynthesis and cellulose accumulation. And this gene is overexpressed in cotton. And you can see here, this is the wild type cotton, and this is the transgenic. All these three are transgenic, and this is the one transgenic having highest fiber length as compared to the wild type. So just one gene makes a lot of difference. You can see here what happens here because of this one gene, this enzyme overexpressed. What happened really? So you can see here accumulation of cellulose, the production and accumulation of cellulose. So this is wild type.
Dr. Katagiri,
Sir?
is it your last slide?
Another 3 slides probably.
Sir, you are taking more time, sir.
Another 3 slides, I will complete. Okay.
Be quick, sir.
Okay.
Be quick.
So you can see here because of overexpression, the cellulose accumulation is more. That's how it is very possible. You can see here the length of that 28 millimeter and 33 millimeter length. So almost 3 to 4 millimeter fiber length is increased, just one gene overexpression. Another gene probably this is not overexpression, it is suppressing the one particular gene, especially phytochrome gene, phytochrome A gene. When it is suppressed with RNA, what happened? You can see here because of suppression of phytochrome A, you can see this is the control non-transgenic and this is a transgenic. How the fiber length is increased by suppressing the phytochrome A, not only the fiber length, but you can see the roots, roots are also more roots and root length is also increased because this is associated trait and naturally it is you can see the increase in root length which is responsible for, later it can become one of the important source for drought resistant. So I think this is my last slide. So phytochrome A, when it is suppressed, you can see the overexpression increasing the phytochrome 2 in a transgenic plant as compared to control plant. See, phytochrome B overexpressed, phytochrome C overexpressed, phytochrome E overexpressed only because of this suppression of this. So because of increase in phytochrome A, B, C, and E, the fiber length is increased and roots are increased. Likewise, several studies are there. What I want to say is all these techniques are helping in understanding complete molecular basis of fiber development, and in turn, as I said, the textile industry demands changing the norms of the fiber quality requirement. So in such cases, this kind of techniques and this kind of knowledge will be very useful in improving the fiber quality as and when required. Thank you.
Thank you, Dr. I.S. Katagiri, for giving a wonderful topic on fiber genomics and including A1, A2, and also lintless cotton, and also what are the usefulness and other things, and with very excellent slide. I thank you for giving a wonderful topic. And the questions will be given by in the box. The speakers will put into a box. We can answer it later. Now, I call upon Dr. A. Mohan Rao, US Bangalore. Dr. A. who is going to give a pre-breeding, a prelude to chili pepper improvement. Dr. A. Mohan Rao, please.
Yes, sir.
And I will take this opportunity to introduce Dr. A. Mohan Rao. He is developed one minute. Dr. Mohan Rao, he is working as professor and head department of genetics and plant breeding, US Bangalore, eminent chili breeder and tilde. He has various positions at US Bangalore. ICR Best Teacher Award he obtained during 2013. He is a recipient of Netherlands Fellowship for Postdoctoral Training at Wageningen University, Netherlands. His several publications in peer-reviewed journals guided many students both for MSc and PhD. Sir, over to you, sir, please.
Thank
Mohan
you,
Rao, please.
Sir, thank you very much. I will request the organizers to enable me to share the slides.
Sir, you can share, sir. You can share, sir.
Okay, just a minute.
Sir, you are audible. You can share.
Okay. Yes, sir. I'll.
Okay.
Yes, sir, it is
the
sharing.
slides visible, sir?
Yeah, visible.
Okay, sir.
Yeah, chili with, okay, I'm visible.
Yes, sir.
Okay, go ahead, sir, go ahead.
Good afternoon, everyone. And I thank the chairperson, the co-chair. I thank the chairperson, co-chairperson, and the rapporteurs. And with their permission, I would like to begin my talk on pre-breeding, a prelude to chili pepper improvement. I'm also thankful to the organizers for providing me an opportunity, especially Dr. Fakhruddin, who insisted me to talk on pepper improvement. And thanks to Dr. Kattigeri, who kickstarted this session with another commercial crop, cotton. Of course, he dealt with the advanced breeding tools. I would be talking on pre-breeding tools. And I'll be dealing with two important diseases. One is anthracnose, the other is leaf curl virus. To begin with, the crop as such.
Sir, kindly put on slideshow, sir. Presentation mode.
Yeah, is it okay?
Sir, now it's okay. Thank you, sir.
Okay, okay. Well, chili is an indispensable item in the kitchen, every kitchen, at least in India and other parts of the world. It's a rich source of vitamin A and C, a rich source of capsaicin, which is pungency, and the color. It has varied uses. It finds varied uses as culinary, pharmacy, and cosmetics. And India is considered as the secondary center of origin. If you look at the importance of this crop at the global level, India ranks first in area and production as far as dry chili is concerned.
Proceed, sir. Proceed.
Yeah, and it ranks 30th among the countries in the world as far as green chili production and productivity is concerned. Now, the major biotic constraints that chili encounters is damping of bacterial wilt, chili anthracnose, leaf curl virus, and chili venal mottle virus. And from among them, we are now trying to address two issues. One is the chili anthracnose and the other is chili leaf curl virus. Anthracnose in chili is a serious pre and post harvest disease causing losses up to 50%, both in terms of yield as well as quality. Pepper anthracnose is caused by Cototrichum species. There are a number of species. World over, we have Cototrichum acutatum and Cototrichum gliospordis widely distributed and also destructive. Whereas in India, Cototrichum capsaicii and Cototrichum gliospordis are the two predominant species, which causes, again, losses up to 50%. Capsicum annuum, the most widely cultivated species, is grown across the world. It suffers from lack of resistant sources against anthracnose. And from among the different surveys conducted, Capsicum baccatum is the only available source of resistance to CC and CG. That is Cototrichum capsaicii and Cototrichum gliospordis, which is prevalent in India. Of course, there are a few reports on C. chinensis that serve as a very good source of resistance, specifically to Cototrichum acutatum, and a specific line PBC 932 has been used extensively both at AVRDC or the World Vegetable Center and other chili breeding centers to breed chili varieties resistant to Cototrichum acutatum. Due to poor sources of hybridization, success of hybridization with cyanum, we have not been able to use Capsicum baccatum or chinense. Okay, and because of this reason, the only source of resistance is C. baccatum. However, Capsicum baccatum does not cross or hybridize well with
Capsicum annum. And with this constraint, we started our work with 3 different approaches. Initially, I'll be talking with intra-baccatum cross, followed by intra-annum cross, and then interspecies. Now, we surveyed for availability of resistant sources from among the annum lines. We did not identify any except for Lampang local and Gauri Bidnur local that were moderately resistant. Well, Susan Joy, again, a variety from World Vegetable Center, was identified another line that contrasted for responses to anthracnose infection. It's highly susceptible. Susan Joy is highly susceptible. These were moderately resistant. I'll talk about this advanced breeding lines, that is LG25. These were advanced breeding lines that we developed, and we have advanced breeding lines with lesion sizes of less than 10 millimeters. Okay. Susan Joy, our efforts again to identify contrasting parents for responses to anthracnose infection. Susan Joy is highly susceptible. PBC80 is a highly resistant line. In fact, it is immune. It gives a very, very small lesion with infection by Cortotrichum capsicum, while with Cortotrichum gliosporides, it's highly immune with lesion size of 0 millimeters. So, using these resistant sources, PBC80 is a resistant source. This belongs to Capsicum baccatum. We try to involve PBC80 in hybridization program with the highly susceptible lines that we identified from among our collections. We effected the crosses involving PBC80 as female and CA14 or Ritkalava as male. You'll observe that when PBC80 or Capsicum baccatum was used as the female, there is no embryo formation at all. There is no seed set at all. But when we reverse the cross, when we used Capsicum annum as female and baccatum as male, we do find seeds or ovules that are formed, but at a later stage, there is abortion of the embryos. So there are reciprocal cross differences for interspecies hybrid seed set. And this work suggests that Capsicum annum should invariably be used as female and Capsicum baccatum should be used as male. And probably after the embryos have set or the seeds have set, we could probably follow an embryo rescue to get the F1 hybrids. Well, keeping this in view, because we could not, despite our sincere efforts, we could not rescue the embryos, we went ahead and identified 2 contrasting parental lines from among Capsicum baccatum. So this is the first approach involving an F2 population derived from a cross between 2 Capsicum baccatum lines. We derived the F2. This is the response of the parental lines to infection by Cortotrichum capsicum and Cortotrichum gliosporides. And this is the spectrum of variability for the F2. And this is the method that we use to categorize the responses. No lesion was categorized as immune. Lesion size of 1 to 5 mm highly resistant, 5 to 10 is moderately resistant, and so on. And this was the specialized scale that we used, provided by Hartmann and Wang. The method that we followed was the Hamilton microsyringe method to induce infection, and detached red dry fruits were infected with a spore load of 500 microspores per inoculation. And this is the response of the F2 individuals. And the overall lesion size ranged from 1 millimeter to 26 millimeters. And this is the distribution of the F2 lesion size. And we do find some lines that are highly resistant. Well, apart from this, we also use some SSR markers to identify any association between a marker and genomic regions controlling anthracnose resistance, we identified SR marker E081, putatively linked to overall lesion size. And that was about intra-bacatum. The second approach is involving a intra-C-annum population. We used RILs here. In the initial slides, I did indicate that Lampang local short and Gauri Bidnur were identified as tolerant lines. These tolerant lines were used to synthesize an F1 hybrid, and then we derived the F2. In the F2, we did identify some segregants that had lesion sizes less than the parental lines. This prompted us to carry forward this population, and we went ahead till F6. We developed the recombinant inbred lines, and the recombinant inbred lines were phenotyped. And we followed a single seed descent approach. This population is at F11 now, but we carried studies in the F6 itself. And this is the method that we followed. We identified 13 moderately resistant lines, 131 moderately susceptible, and 16 highly susceptible lines. And the lesion size ranged from 8.33 millimeters to, of course, 30 millimeters. And this is how it is. It ranged from 8 millimeters to 30 millimeters. We did identify some superior recombinant inbred lines that had low lesion size. And these lines, lesion size of 8.98, 9.17, 9.93, they also incidentally had higher fruit yields. Okay, so we did identify recombinant inbred lines with resistance as well as high dry fruit yield. And here we identified a marker HPMS E047 to be putatively linked to genomic regions controlling anthracnose resistance. Well, that's about intra-annum. And now this is the interspecies, this is the pre-breeding work. In the initial slides, again, I did indicate that Susan Joy is a highly susceptible genotype. This genotype was used as one of the parents. Apart from that, we also used Tiwari, which is also highly susceptible. We synthesized the F1s, and F1s could be easily identified using morphological markers because Bacatum has a very peculiar pigmentation on the petals. This character is dominant. This character could be effectively utilized in pre-breeding work to identify promising true F1s. And these F1s were selfed. Unfortunately, the F1 plants did not set any fruits upon selfing. So we could not generate the F2. The F1s were backcrossed to derive the backcross plants. We are at BC2 F1 plants now, and these are being now forwarded to BC3. In another cross, we use Susan Joy, PBC80. We have developed the backcross generations here again. You would observe here, the fruit shape is very, very bad. Though we could get anthracnose resistance into Susan Joy, the shape of the fruit is not acceptable. So this is how the plant looks. Not horticulturally accepted, but expresses high level of resistance to anthracnose. These could serve as donor lines for developing anthracnose resistant varieties in future. Now this is, again, you don't have a spec after inoculating these fruits with a spore load of 500 per prick. The next aspect I told was chili suffers from leaf curl virus, and yield losses are up to 100%. Whenever the plants are infected, it causes culling of the leaves. It's also accompanied by yellowing, stunting of the plants. Leaves become very small. Fruits are also very small, and farmers do report complete losses. And in several regions of the country, farmers have abandoned their chili crops due to leaf curl virus. Genetic options are considered as most effective and eco-friendly, and identification of stable source of resistance is very, very important. Bush jalokia was identified as a resistant source. It was reported to be a resistant source. We also confirmed the resistance behavior of this genotype, and we involved them in developing a population. So here we used Badgi Kaddi very, very promising genotype in Karnataka, known for its rich color or a high color, high oleoresin, highly susceptible to leaf curl virus. Now, Bush jalokia is highly resistant to leaf curl virus. Incidentally, these two parental lines also contrast for color and pungency. Bush jalokia is yellow fruited or orange fruited. Badgi kaddi is high oleoresin genotype. It is also very, very poor in pungency or low pungency, whereas Bush jalokia is known to be or referred to as the most pungent genotype in India. So they contrast for 2-3 characters. So that is how we started with this cross Badgi kaddi into Bush jalokia. We derived the F1. The F1 was found to be highly susceptible. The F2 expressed resistance and susceptibility, and the F2 segregated in the ratio of 15 susceptible to 1 resistance. BC1, P1 plants were all susceptible and crossed back to Badgi kaddi. All the BC1 plants were susceptible. BC1, P2 plants exhibited 3:1 segregation. So this is testing the goodness of it, and it implies that the F2 segregated in the ray 15:1, and chi-square suggests that it is a good test for the F2 fitted the ratio of 15:1, suggesting that the character is under the influence of duplicate dominant epistasis. And we gave symbols to the parental lines. This is Badgi kaddi, this is Bush jalokia, this is Badgi kaddi. This is susceptible. F1s are highly susceptible, and these are 15 susceptible. And the epistatic interaction involved is duplicate dominant epistasis. And 1 out of 16 were found to be resistant. And this one resistant plant will breed true, and because the resistant plants breed true, they can directly be used self to stabilize other genetic loci. We don't have to select for resistance in subsequent segregating generations. We need to select the resistant plants to stabilize other genetic loci for horticulture traits. Now, as already indicated, the two parents also contrast for capsaicin and oleoresin. And this is the cross again, Badgi kaddi cross to Bush jalokia, the spectrum of variability in the F2. And this is the variability in the back crosses and the F2. So 2 different species, Annam and Chinese, could be effectively crossed to generate all the 6 generations. There is no barrier as far as hybridization is concerned. And these 6 basic generations were used to arrive at the genetics of the 2 traits, oleoresin and capsaicin. And we did identify transgressive segregants for oleoresin, for capsaicin, as well as for dry fruit yield per plant. So suggesting that interspecies cross between Annam and Chinese is workable, and you can improve both capsaicin and oleoresin contents along with yield. This is another example. We have capsicum Annam genotypes in the form of Aparna, Gauri Bidnor, and another maintainer line of a CMS line. Now, one example we have shown, Aparna is yellow fruited. Carolina Reaper is a capsicum Chinese line. Deep red color, we can improve the color of our lines. We did develop the F1. This is the spectrum of variability in the F2, suggesting again that capsicum Annam into Chinese can be used, interspecies cross can be used for improvement of color. Well, apart from that, we have also affected bridge crosses. We wanted to combine anthracnose resistance and leaf curl virus resistance. So we chose 7 cultivars: Aparna, Badgi kaddi, Badgi dabbi, Pansivan, Tiwari, Pusa Sadabahar. And these were now crossed to Bush jalokia or Carolina Reaper. These are Chinenses, these are Annam lines. And the F1s derived were crossed to PBC 81 the Baccatum line and PBC 80. So we now have developed bridge crosses involving Capsicum annum, Capsicum chinense, and Capsicum baccatum. So the main aim is to introgress CHLCV and anthracnose resistance, CHLCV resistance chinensis, anthracnose resistance from baccatum. Okay. And we are now forwarding these bridge cross derived progenies to this next level. These are some of our publications.
Yes, sir. Good.
And this is the team that has worked on pre-breeding in Chile. I thank the organizers for giving me an opportunity, and I hope I am well on time.
Oh, thank you, Dr. Mohan Rao, for adhering to the time, and also you have given a wonderful work on chili breeding, particularly bridging of these different species, and also making a color and also pungency by taking the Bujolokia and Capsicum baccatum for your tolerance to anthracnose, and also particularly in Karnataka, because of this, earlier in Dharwad district, we have got a 2 lakh hectares of chili. Now it has come down to less than 500 hectares because of this chili leaf curl and also other diseases. With this, your work will be more useful in the future, and I wish you will continue. Ramesh, Dr. Ramesh, do you want to say some words? Ramesh, our co-chairman, want to speak some words? If there are no, I thank on behalf of the organizers, Dr. Mohan Rao, for giving a wonderful presentation on chili breeding.
Thank you very much, sir. Thank you very much, sir.
I'll call upon Dr. Dinkar Adiga. Dinkar Adiga, who is working in ICR, DCR Puttur. He is going to give a talk on breeding for novel traits in cashew. Cashew is now the crop of the season. Dinkar Adiga, I will give a brief introduction about the Dr. Dinkar Adiga. Dinkar Adiga, are you there in the?
Yeah, I am there.
Yeah, he's also my good friend.
Yes, sir. Yes, sir. Thank you, sir.
Principal Scientist Horticulture Food Science at ICR DCR Puttur, worked as Assistant Horticulturist at Central Coffee Research Institute, Karnataka, and involved in genetic improvement and propagation of coffee. He has also worked as Assistant Professor at KRCC Charabavi, Professor of Fruit Science at College of Horticulture, Bangalore, that is under UHS Bagalkot. Now guided more than 15 students as chairman or member of the advisory committee for postgraduate students. He has got worked as a senior scientist horticulture at ICR Directorate of Cashew and involved in projects like genetic improvement of cashew, rooftop studies. He has organized international conference on green technology for health and environmental policies and issues. So far, he has handled more than 10 research projects on various aspects of horticulture and published more than 50 research.
Hello, sir. Hello.
Yes, sir, you are audible, sir.
Okay. My slides are visible?
Yes, sir.
Okay.
Visible.
Thank you, Basraj sir, for a nice introduction. So I'll be talking about breeding for novel traits in cashew. So the breeding for yield is very common in tree crop improvement. But whereas cashew being a crop of importance for both nut and apple, so we have also carried out some efforts at ICR Directorate of Cashew Research for improving some of the novel traits in cashew. Hello?
Matadi, you please carry on.
Yes. Whether my slides are moving, sir?
Sir, its first slide is projected, sir.
Okay.
Now please move, sir.
Yeah.
No, sir, it's not moving.
It's not moving. 1 minute. Yeah. Could you see this slide?
Yes, it's moving.
Yes.
Yes, yes, yes. So cashew nuts, as all of you know, is valued for high value nuts. Especially we are getting around 8,000 crores of revenue from exporting cashew nuts. So it's a rich source of healthy fats, and India produces around 8 lakh tons of cashew annually, and whereas our processing needs are 17 lakh tons. But unfortunately, our productivity cannot cater to the needs of processing industries, which has put a lot of burden on exchequer while importing of raw nuts. So being a crop of the origin from Brazil, the diversity what is present in India is very less, and which makes the task of breeders a very challenging one. So the efforts done at ICR Directorate of Cashew Research on adding some value to the existing varieties, such as improving the nut size, then reducing the tree height, tolerance to biotic stresses, and improving the quality of apples. These were the lines where the works were taken up recently, and such works are taken up at ICR Directorate of Cashew Research. So coming to the first, the value-added aspect, that is for jumbo nuts. Why we need jumbo nuts? So as we can see from farmers' perspective as well as from processors' perspective, it's very important. From farmers' perspective, it fetches better price. A kg of nut gets extra rupees 15 to 20 rupees extra per kilo if the nuts weigh more than 10 grams. Then for processors, also the nuts fall under the kernels rather fall under the category of jumbo nut. That's W180 is the highest grade. So what we have developed falls into still higher grade. Let's say W150, W130. So they can fetch premium price by exporting these kernels. Next aspect is ease of harvesting. So harvesting contributes a lot for cost of cultivation in cashew. When the nut size is small, the time taken for picking is also more. So when the nut size is large, a same worker can collect 80 nuts to constitute 1 kg, whereas in existing varieties, one has to pick 150 nuts to constitute 1 kg of a cashew nut. So it also saves labor in factories, be it a process of shelling, process of peeling, process of grading, everything will be easy and it can save the labor too. So there are many challenges associated with breeding jumbo nut hybrids. Wherever the nut size is more, the yield will be less. So most of the germplasm, whatever available in our germplasm repository, though the nut size is bigger, the yield is very low. Second one, shelling percentage. Shelling percentage is the one which is very much important for processors. An ideal variety should have more than 28% shelling percentage. So when the nut becomes bigger, usually the nuts have a thicker shell weight and kernel weight goes down. Then cluster bearing, always the jumbo nuts varieties have low number of nuts in a cluster. Say 1 nut or 2 nut, at the most 3 nuts. This is another barrier to be broken while developing jumbo nut hybrids. Uniformity in nut size is another concern. So most of whenever the nut size is jumbo, around 30% of the nuts will be of smaller size and 70 to 80% will be of bigger size. This will again make the task of grading a Herculean one. Then regular bearing. As seen by me as well as the workers in Australia, the jumbo nut varieties, usually they behave like alternate bearers or irregular bearers. So a large-scale hybridization was done at our directorate under ICR network project. Around 1,200 hybrid seeds were generated through massive hybridization program. Among them, hardly a few, and among few, one hybrid is identified as the most promising one for cultivation by farmers. That is hybrid H126, derived from a cross combination of NRCC selection 2 and Bedasi. Selection 2 is a variety released earlier. It's a medium nut size variety, medium to bold nut, where Bedasi is a slightly bigger nut size, it's say around 9 to 10 gram. So this hybrid has recorded nearly 92% higher nut weight and 79 to 80% higher kernel weight compared to popular medium nut size variety Bhaskara. As you can see here, Bhaskara is a popular cultivar released in 2007, adopted by most of the farmers. And here you can see compare the nut size of Bhaskara with 126, the apple size of Bhaskara with 126. That's a huge difference. Then as compared to its mother, NRCC selection 2, it recorded nearly 46% higher nut weight and 36% higher kernel weight. So over both the local checks, it recorded higher yield. So this is a significant outcome of a hybridization program to derive jumbo nut hybrids in our directorate, ICR Directorate of Cashew. As you can see the bunches here, almost all the nuts are of uniform size, you can see. The difference in the nut size is almost nil. So when upon harvesting and drying, I could see that 95% of the nuts fell under uniform category of 12 grams. And you can also see the cluster, number of nuts in a cluster, it is reasonably well. You can see 6 to 8 nuts, whereas you can hardly see 2 to 3 nuts whenever the nut size is bigger. So this is a general view, and this tree is located on a barren land, which is full of infertile soil and with highly acidic pH. Even under such adversity, the tree has profusely produced the flowers. It is full of flowers. You can see the tree canopy is full of flowers, and nut set is really good here. And this is the kernel of H126. Very big kernels, very bold. Whereas normal kernels will weigh around 2 grams. This is weighing 3.6 grams each kernel. And this was also offered to the governing body members of ICR in the GB meeting. And all the members expressed that this kernel is unique and its taste is unique, and this is more tastier compared to the normal Kaju available in the market. So 80% higher kernel weight than popular variety Bhaskara. This can easily be value added for chocolate coating, and that is an exportable produce from cashew kernel. There are many setbacks while developing jumbo nut hybrids. The first one is nut size will not be uniform. As you can see here, one will be small, other one will be big. This is mainly because cashew is associated with one phenomena that is called as primegenic dominance. The first set nut will suppress the growth of the later set nuts. So that leads to variability in nut size. This is a typical example of another jumbo nut hybrid, H130, which is susceptible to this phenomena of uneven nut size, nut drying, apple drying, shrinking, and nut size initially will be high, and at the end you can get hardly 3 to 4 nuts of good size. The rest all will be dry. And then evaluation process, you should always look for yield every year. So it should be almost uniform. If the yield fluctuates, like if it produces a tendency of alternate bearing, then you should not select such hybrids. As we have seen in many of the jumbo nut hybrids, alternate bearing was observed, and such hybrid should be deleted during the further processing. And another thing is that even in Australia also they have observed the alternate bearing in jumbo nut cashew germplasm. So here also we have observed. So we have taken 20 years to release this hybrid. That is the extent of evaluation which has gone through. One is under unreplicated augmented design, then under replicated trial. Though it produces Well, under unreplicated trial, it is not guaranteed that it will produce under replicated trial. So we have exposed them to 2 rounds of trial. Then coming to the next feature, breeding for reduced stature. So the simple question will be why we should breed dwarf plants when tall plants yield higher. But whereas in cashew, we can hardly accommodate 200 plants per hectare under normal density because most of the cashew varieties are vigorous in nature. That is why, so since productivity is an issue here, we need to accommodate more plants per unit area. When it comes to rootstocks, the multiplication of rootstocks by clonal means is a challenge in cashew. Though we have 2 dwarf rootstocks, multiplication by vegetative means is a challenge in cashew, especially the rootstock multiplication. So breeding is an approach wherein you can utilize the available germplasm, but available germplasm cannot be released as such because their nut size will be smaller, their yield will be smaller. So by resorting to the development of dwarf hybrids, we can ensure higher productivity, and canopy management also becomes easy because cashew involves annual pruning and also application of pesticides, 3 rounds of pesticides. So canopy volume makes a lot of difference. So this is one germplasm from Brazil we have utilized for a breeding program. See, tree is dwarf, internodes are very short, but the apple and nut size is very small. This is Taliparamba one. You can see the dwarfest one. It is almost like a crawling habit. Here also nuts are shrunken, nut size is small, flowering is profuse, but nut filling is less. So we have used these 2 as donors in breeding program, and utilizing our popular cultivars, we have made crosses, and from the different cross combinations, we have generated many hybrids, and out of those hybrids, we could identify one hybrid as very promising, that is tree number 626, which fell under semi-dwarf category, where Bhaskara was a female and Brazil dwarf was a male parent. And the plant height was only 2.9 meters, which fell under semi-dwarf category. And whereas nut size was remarkably improved with the 9-gram nut size, whereas Bhaskara has only 7 gram, 492 has only 5 to 6 grams, so nut size is improved and yield has also improved. So these 4 dwarf types were shortlisted, semi-dwarf types, and they are under replicated trial. So you can see at 626 is less than 3 meter in height, and yield was also very good. Nut size was bold. So we did both direct and reciprocal crosses utilizing both the Taliparamba 1 and NRC 492. And we took 4 popular varieties in another set of cross, Vengurla 4, Priyanka, Dana, and Madakatara 2. Dana and Madakatara and Priyanka 3 are from Kerala, and Vengurla 4 is a national variety. And we generated around 800 hybrid seedlings from 15 direct and reciprocal crosses, and were planted for evaluation in way back in 2013. And from that population, we could identify 14 promising trees. With respect to height and spread, the tree number 8 was identified as the best performing one from a cross of variety Dhana and Brazilian dwarf, which was found semi-tall and with the spread less than 6 meter and recorded a nut yield of 1.33 and nut yield of 6.3 over 5 harvests, and it possessed bold nuts 8.3 gram. This is the promising hybrid. So tree height is in a semi-dwarf category, and it can easily be managed. Canopy can be managed easily. It can be accommodated under high density planting. And backcross breeding was another approach which was followed to generate dwarf types in cashew. The dwarfest progeny of the cross combination Ullal 3 and Brazilian dwarf was backcrossed with Ullal 3 to improve the nut size and quality of apple. And progeny of Bhaskara and NRC 492, another dwarfest progeny, was backcrossed with Bhaskara. And we have generated around 472 backcross progenies. They have been field planted, and 17 promising trees were selected during 2018 and 19, and the progenies exhibited semitall features and are under further evaluation. And we are hopeful that in a couple of years' time, we will come up with a semi-dwarf hybrid in cashew, possessing high yield and better nut size. Now coming to breeding for tolerance to tea mosquito bug. You know, tea mosquito bug is a devastating pest which can cause up to 80% loss in cashew yield. And last year, we lost nearly 50% of the crop, 50 to 60% of the crop, almost throughout the coastal Karnataka because of the single pest, tea mosquito bug. So we don't have any resistant or tolerant varieties available among the cultivated species of Anacardium occidental. So we resorted to crossing with wild species. We selected our popular cultivars, 5 popular cultivars, Ullal 1 and 3, then Bhaskara and V4. And we made crosses in both ways. First set we took male parents as Microcarpum and Orthonianum. Both are the wild species of cashew. And in the second set, we took Microcarpum as female parent and the popular cultivars as male parents, and we effected crosses. And among the many interspecific crosses generated, a progeny of a cross combination Ullal 1 as a female parent with Anacardium microcarpum as male has been observed for 2 years, and it has been found to be almost free of TMB damage. The location where the tree is planted is a hotspot for TMB occurrence. Every year we get TMB devastating TMB damage in the location. So the tree is located in that location, and it has shown tolerance to TMB attack. So progeny, it seems to be tolerant to TMB, has recorded an average damage score of just 0.06 compared to Ullal 3 and Vengurle, which recorded a damage score of 1.22 and 1.38 respectively. So further, this tolerant tree as male was crossed with Vengurla 4. That is highly susceptible to TMB and is a commercial variety, to generate 79 progenies, and Ullal 3, it is also crossed with Ullal 3 to generate around 17 progenies. Here, the cross combination progeny number varied because the success in hybridization varies between the cross combinations, especially when it comes to the wild species, crossing with wild species. So evaluation work is under progress. So this is the interspecific hybrid, Ullal 1 crossed with the Anacardium microcarpum progeny. And you can see that the leaves are coppery, new leaves are coppery, and slightly older leaves also. There is no infestation of TMB. You cannot see any puncture marks here, no drying of leaves. So we have been monitoring regularly. And this is a closer view. It has profusely flowered without any impact of TMB attack. So TMB could not attack this tree, this interspecific hybrid. Then coming to the mutation breeding approach. So recently, 113 seedlings and grafts obtained from gamma-treated seeds and scion sticks of cashew of two popular varieties, Bhaskara and Ullal 3, are being maintained in our field. And the next generation seedlings, M2 from all these trees, will be screened for TMB tolerance. So for TMB tolerance, we have resorted to 2 approaches. One is inter-specific hybridization. Another one is mutation breeding approach. So interspecific hybridization has shown promise. Mutation breeding, it's only time will answer. So some of the variants we got pluckering in leaf, pluckered leaf morphology, we could see in mutated progenies. And you could see early flowering also in the seeds treated with mutants. Now coming to the breeding for reduce tannins in cashew apple. So one will ask why you are so bothered about cashew apple, because cashew apples are very tempting to eat, but after eating effects are not pleasing, and you get itching in the throat. And similarly, while processing also, the cashew apple juice, cashew apple syrup, everything, the tannin comes in the way. So unless you remove the tannins, so it will not be a palatable one. So cashew apples are going wasted. See, a cashew produces nearly 8 to 10 times of apple compared to the nut. 1 kg nut produces nearly 8 to 10 kg of cashew apples. So they are really richer than oranges in vitamin C. 1 kg of cashew apple is equal to 4 kg of oranges. They are very rich in antioxidant properties, and also they are very good for some of the stomach ailments. Sources, we dug the sources of low tannin for breeding program. So we found that Anacardium microcarpum, a wild species, recorded low tannin content of just 0.14%, whereas cultivated species had nearly double to 5 times that of Anacardium microcarpum. So microcarpum, which is available in our gene bank, was used earlier in hybridization program for developing TMB tolerance, and the same germplasm, same hybrids are now being evaluated for tannin content, and 125FN hybrids along with their parents, Anacardium microcarpum, Ullal 1, Bhaskara, and other popular cultivars like Ullal 3 and Vengurla 4 were analyzed for tannin content. And tree numbers 820, 834, tree number 829, tree number 822, tree number 830 of the cross combination Bhaskara into Anacardium microcarpum were found to be the top 5 for lowest tannin content. And compared to the released varieties, they are much, much lower in tannin content. And that is why these interspecific hybrids hold promise in generating hybrids, generating varieties which are low in tannin but rich in vitamin C and antioxidants. So we have also identified another big apple hybrid. It's from another cross combination. So VTH 174 is the tastiest of apples we have found in our germplasm. And this was generated as a process of routine hybridization for higher nut yield and higher nut weight. But luckily, we could find one hybrid. This is H186, a cross combination of 2 germplasm available in our repository. The apples were really big, 100-gram weight, and they are really sweet. With you cannot imagine, you won't get any itching sensation in the throat. And pulp can be easily eaten without any fiber matter. So there will not be any adhering fiber matter. You can eat it like a dough, and it easily melts in your mouth when you chew on the apples of this hybrid. So this hybrid was also a high yielder with 4.6 kg nut yield just in the 5th year of 5th harvest. And apples are really good, and apples are very tasty and really
Dr.
big.
Dinakar, is it your last slide?
Yes, sir. Yes, sir. Yes, sir. Yes, sir.
Dr. Adiga?
Yes, sir. Yeah, this is my last slide.
Dr. Adiga?
Yes, sir. Thank you, sir. Hello? Yeah, thank you, sir. Hello? Sir, thank you.
Hello.
Hello? Sir, it's over. Over.
I think Adiga has completed, sir.
Yes, sir.
I think there appears to be some network issues from chairperson, sir.
Yes, yes, yes.
Yeah, yes. Yeah. Adiga, nice to see you after a long time.
Yes, sir. Same here, sir.
So,
It's 2 years back.
Yes, yes.
Yeah, at Puttur.
Very nice and very focused presentation. Very good progress.
Thank you, sir.
I would like to see big apples in the market.
Yes, sir. Yes, sir.
They should become good competitors to the regular apples.
Yes, yes, sir.
In
Yes,
terms
sir.
of nutrients, I believe cashew apples are much better than the regular apples.
Yes, yes.
Okay.
Yes.
Yeah. Sir, chairperson, sir.
Ramesh sir, there is a problem with network of Basraj sir. Please continue, sir.
Okay, okay. So, very good presentation, Adiga.
Thank you, sir.
In fact, last year, last year, I think I visited when you were not there in the institute.
Yes, sir.
For SRC meeting.
Yes.
A good work is going on. Dr. Mohan GS and then Irdasappa. Irdasappa, of course, was in our campus doing his PhD. Mohan, I'm regularly in contact with Mohan. Is doing excellent work, and after you joined, the team has the strengthened, and I could see the impact from your presentation. Very good, sir. Congratulations.
Thank you. Thank you,
And
sir.
working in perennial crops like cashew is a challenging. You're doing good, sir. Thank you. Thank you.
Hello.
Yes, sir.
Am I
Basraj
audible?
sir. Yes, sir. Audible now. Continue, sir. Basraj sir.
Sir.
Sir, Ramesh sir.
Yes. Okay.
Yes,
No,
yes,
I thought Basuraj sir will respond.
Sir, Ramesh sir.
Yes.
Sir, you can call, sir. You can call next speaker.
Okay, okay, okay. So there appears to be some network issues from Chairman
Yes, sir.
sir end.
Yes, sir.
Now I would like to call upon
Adiga.
Dr. Wagish Bob. Okay,
Thank you, sir.
right online. Sir.
Okay, thank you, sir. Thank you.
Yeah.
Yeah, yeah.
Sir.
No, wait, Ramesh, wait. I have to give, Ramesh, I have to give some suggestions.
Yes, sir. Yes, sir.
Ramesh, please.
Yes, sir. Please.
Dr. Adiga. Thank you for giving an insight from breeding from jumbo net size to reduce the tannins. But one more challenge is you have to develop a variety suitable for flat soils. Now you may be knowing the cashew is coming over to the non-traditional area in a bigger way.
Yes, sir.
People are removing the mango and they are planting. So this is the challenge even in during your AGM meet recently held in Bangalore. I have suggested this to your directorate. Please take care of that.
Yes, sir.
Okay, with this, thank you. And now I request Dr. Wagish Babu HS for giving a topic on accelerated breeding by tilling, a non-GE district tree in biotechnology. Just I'll give a brief introduction about Wagish Babu. Sir, are you there, sir? Wagish Babu?
Yes, sir. I'm very well there here. No problem, sir.
He has developed a BT transgenic eggplant and tomato for genetic resistance to the important pests using BT genes and gene pyramiding, characterization of phylloplane and rhizosphere microfile, plant tissue culture, transformation and agrobacterium plant interactions, gene tapping and functional genomics in tomato, development of moss rearing technology for brinjal shoot and fruit borer, Lysinodus arbonalis, and tomato fruit borer Helicoverpa armigera. He has moss rearing of brinjal shoot and fruit borer Lysinodus arbonalis, and he is a member of Indian Science Congress Association, course coordinator for winter school in biotechnology and horticulture, PG teacher in field of biotechnology at NRC in plant biotechnology, IRI, New Delhi, US Bangalore, Coimbatore University, US Horticulture, etc. He has conducted the first national level contained limited open field trials and many things he has got his credit, and presently he is working member instead of Biosafety Committee and the Department of Biotechnology, New Delhi. Sir, over to you, sir. Wagish Babu, yes.
Okay, sir. Sir, I think I am audible very much for everybody.
Yeah, audible, sir, please.
Okay, sir. So, thank you, the host, for inviting me, and providing me an opportunity to share my experience and thoughts with all of the participants and other people. And I'm currently principal scientist in Division of Basic Sciences and Biotechnology, IHR, Bangalore. I'm a biotechnologist. Today, I would like to share with my fellow researchers my topic, accelerated breeding by tilling, a non-G dexterity in biotechnology. Sir, friends, actually, we have seen a lot of improvements in crop improvement breeding programs and everything. And so far, a lot of slides were there, and some of these slides also touched upon biotechnology. My research area is more of biotechnology, application of biotechnological latest advances in biotechnological tools and technologies. And my lesser role is as a breeder in horticulture crops breeder. Currently, I am working on molecular improvement, molecular speed breeding of papaya. And I have also earlier I had worked on transgenic crops, insect resistant BT crops. Okay, and I would like to tell some of the pros and cons of all the technologies, and then I will come to the importance of my work, which I would like to share. See, the conventional, we should not call exactly as conventional, but conventional breeding is a little bit slow in many crops, annual crops, and my friend and predecessors, Dr. Adiga, informed that in the matter of tree species, the breeding is still very slow. If you want to take up 2 generations of studies, you have to wait for 20 years or so, like that. And multi-location centers, everything goes very awfully slow. So that is one disadvantage, of some of the slower disadvantage of conventional breeding. And second part is, we have the selection efficiency. The index of selection is very low, and it is very low efficiency, because, for example, if you want to identify a disease resistant material, a very good trait, then what happens? We need pathologists, we need agronomists, we need all sorts of people. We need to inoculate field crops with virulent pathogens. We have to maintain pathogens, we have to maintain insects. All these things are there. And then if the environment is not favorable, then that entire season is not useful. And then it's like pollinate and prey type of mechanism we are observing. And with respect to pathogens and insects, we observe choice or no choice. For example, if in a breeding cycle, if insects are not appearing, then we cannot select for resistant and susceptible plants. We cannot differentiate. This is the another problem. There are a lot of problems with the selection efficiency and all these things. And less novelty. For example, if there in the germplasm, if there is no novel features, or the features which we are looking into, which we are interested, for example, disease resistance or new traits of commercial importance, if they are not there in the germplasm, in the world germplasm collection, then no, we have to create by mutations. And mutation breeding is altogether different type. We may or we may not get. These are some of the cons, I may tell, not exactly disadvantage truly. And some of the so to overcome all these things, we have we have to integrate biotechnology with plant breeding, very potent mechanism. But in terms of biotechnology, the last 20 years we have seen whatever has happened. For example, in case of GE food crops, especially BT brinjal, we have seen moratorium, blanket moratorium on BT brinjal, because we developed advanced stages of BT brinjal at IHR, and then the policy and all public perception and public awareness, acceptance, they all mattered. Okay, Therefore, then policy was taken to do a blanket moratorium on food crops, food crops, not with respect to BT cotton. BT cotton has really revolutionized the Indian and world agriculture. One BT, single BT gene has really revolutionized world agriculture. Okay, but with respect to food crops, the lot of food crops waiting after brinjal, BT brinjal, BT tomato, and enhanced tomatoes, rice, golden rice, and a slew of improved transgenic GM crops are waiting for release. But right from the beginning, BT brinjal stopped moratorium, and we don't know what is the future. And there are also biosafety and concerns, acceptance issues. Public, if all goes well also public, is difficult to see that their acceptance. So with these all things, we have used up as a biotechnologist. I know when we were searching for new avenues, new opportunities, we came upon tilling, on latest technology, where it does not involve genetic engineering. It's a non-GE method of crop improvement, and it's a purely breeding approach, but it is not a forward breeding and forward genetics approach. It is a reverse breeding, reverse genetic approach, tilling. And then we thought that it can very well easily integrate into our current breeding programs. We have to strengthen our breeders. We have to become molecular breeders ourselves, and increase the level of selection efficiency, and improve the speed. We call it speed breeding or rapid breeding. So that sort of themes, this tilling technology could very well integrate. So instead of going for latest RNAi or gene editing, we took upon this tilling project, and then we are for last 4 to 5 years we are following tilling, and so we are doing in important horticulture crop, that is papaya. World over, very few labs are doing tilling, mainly in Arabidopsis, in rice and maize, etc. But in India, only one lab, throughout India, only one lab is working on tilling, that too in tomato. Okay, so we thought, why should not we do? Because improvement of perennial crops like fruit crops, people, breeders, biotechnologists, they hesitate because of longer shelf life. But we wanted to prove our others wrong, and we wanted to take up this challenge to attempt this novel technology, biotechnological tool, to start with in fruit crops. Why not in fruit crops? And we took up papaya. Papaya was a very good seed propagated fruit crop, and a very good candidate. And we took up papaya, and we integrated this latest technology, tilling technology, and then that's why we took upon this interesting, challenging, and very enticing scheme. Tilling is basically a economy targeted induced local lesions in genomes. Okay, so it is a reverse genetics approach, I will tell. So it was invented by a postdoc graduate from University of Washington, McCallum, Stephanie McCallum, and Luca Comai's lab. She's Luca Comai, and Green and Stefan Enikoff. They did it in 2000. The paper came out in Nature in 2000, exactly 20 years before this technology was there. But surprisingly, many biotechnologists are reluctant to take up this project. It is a very tough project. It involves a lot of technical input. So we did not hesitate, and we took up this challenge. It's actually a best of both worlds. It's a marriage between conventional mutagenesis and non-GE biotechnology. And as I told, it's basically reverse genetics, reverse breeding approach, where in forward genetics and forward breeding, we don't know anything about gene. We just do mutagenesis, large scale mutagenesis, and in advanced generation we do mutations, and then we try to screen, and then go for which gene has been mutated, what is the genetic basis, like that. That is a forward genetic approach. But in this reverse genetics method, as a biotechnologist, we already know what genes are responsible for what trait. So we would like to mutate the gene, do a mutation, do a functional genome. It's a basically functional genomics approach. We do a mutations in the gene, respect to genes, whatever gene of interest, and then look out for the phenotype of our interest. So this is exactly the reverse genetic approach. It's a very novel approach. And no, the another beauty of this tilling technology is that we don't need a large acreage and large number of plants, facilities, and pathologists, entomologists, and all. It is not a season bound. It is not influenced by environment, etc. You can screen thousands and lakhs and possibly millions of mutant populations in vitro, in lab only, in a matter of 10 by 10 square feet, and that is the technology. And then we can select the best mutant, and then we can concurrently we can identify the mutant plant seeds we have, and then we don't need to grow large number of populations, advanced populations. We just need to isolate and do genetic analysis in the lab only using genetic analyzers, advanced analyzers, and instruments. Then we can easily identify mutants of our interest, and then you can correspond to whatever plant lines accessions we have, and then we can grow those. Instead of growing 10,000 plants or 1 lakh or 1 million plants and screening for our interest phenotype, just do it in lab, and then we can identify one plant among 1 million plants in the lab only, and then we can take up that one plant we can evaluate. See, this is the best speed breeding, most rapidest speed breeding method, because we don't need hundreds of acres, and we don't need all those technologies. So this is the advantage of tilling. Very high selection efficiencies and selection indices we can realize. And it's an in vitro screening method of targeted mutations. Mutations here are random, but we are selecting targeted mutations. So it's a very good method of selecting our whatever wanted the mutation that we are interested. So we don't need all other mutations which we are not interested. Then this is type of SNP system research. We will analyze, we will develop, we'll produce, and we will analyze SNPs and indels. And then through different methods, and we have our very excellent lab setup in IHR. We have a real-time PCR based quantitative real-time PCR we are going to do, and then high resolution melt curve analysis. These are the techniques we commonly, routinely, often we use in our labs. Of course, they look very standard, but this is the system we have optimized in our institute. And there is no GE or no biosafety issues concerned, because it's a completely for the breeding approach. There is no transgenes involved. We are targetedly we are mutagenizing, and we are identifying the best plants and lines. And so we are attempting, as I told previously, we are doing the unthinkable. First attempt in fruit crops, papaya. World, nobody has done papaya tilling for shelf life and other traits, and we are trying to do it. So we have taken it up as a challenge. Okay, in our own variety, Arka Prabhat, very beautiful variety, high yielding and beautiful flesh color, tasty. It's a hybrid variety developed in Sunrise and Solo, developed by our current director, Dr. Dinesh, 15 years back. And we have got it's a very progressive variety, variety in very demand, and only important variety in public sector, from public sector, apart from Taiwan Red Lady. So, and our targets are shelf life and PRSP tolerance. PRSP, papaya ring spot virus, spotty virus, the menace throughout the world. There were efforts to develop transgenic papaya, and they have succeeded. People have succeeded in Hawaii, and in India also there was an attempt to bring that PRSP tolerant transgenic papaya, but it did not succeed because of the breakdown of resistance, PRS resistance. Therefore, we have to develop our own indigenous make in India, varieties and hybrids for PRSP tolerance, if not resistance. And shelf life is second most important, because we are losing nearly 25 to 30% yield loss during transportation of papaya, because papaya fruit is very susceptible to even smallest bruises. They are grown in and around peri-urban and villages side, and they are transported to metros during this transportation and storage, a lot of papaya is being wasted, and nearly 30% papaya. So we would like, if you stop that 30% loss is akin, it's equivalent to improving 30%, increasing 30% yield in papaya. So these are the two critical idiotype characters that we are interested in, and we are developing, addressing this by tilling technology. And by this, we have already developed advanced generations. We all know M1, M2, M3 generations. Myself and my students have advanced these generations. We have evaluated, and full SIB evaluation is undergoing, because in papaya, it's a typical non-characteristic plant, because we have both hermaphrodite plants as well as female plants, gynodicious in nature. So selection is little bit difficult. So we have to identify hermaphrodite plants and then do selfing, or if there are very good promising female plants, then we have to do full SIB or half SIB crosses, and then we have to go to next 2 generations ahead. So these are the complications which we are facing, but still we are doing these things. And we also have robust in-house lab facilities, research facilities in biotechnology and basic sciences at our institute in doing this novel advanced genetic technology, biotechnological method tilling by using RT-PCR and real-time PCR, quantitative PCR, as well as high-resolution melt curve analysis, etc. See, this is briefly the methodology. We do EMS plant seeds, and M1, M2, M3 we will develop. Up to this point, we are similar to conventional breeding. Okay, from M2 and M3 onwards, what we are doing is, instead of screening in fields, instead of wasting a lot of resources, what we are doing, we are extracting DNA from each plant, M2 and this segregating population. Then we are going to identify these genes. DNA, we are total DNA will analyze. Then we are doing SNP detection and indel detection by molecular techniques. Okay, that we can do in the lab, in vitro. As I told, tens, hundreds of thousands of samples we can do it in the lab only. We don't need big waste of resources. No resources are needed. Everything only in a small lab. We can do all these things. The selection is very extreme. So this same replicates. We do different PCR, basically PCR method, and later we use either DHPLC. Currently, we are not using this DHPLC method. So we are using HRM method, and HRM is very advanced and very sensitive method compared to DHPLC method. And we are identifying mutations and SNPs and indels in the population. Then we'll do plant analysis. So in aerosol melt curve analysis, what happens basically? See, if there's a mutation in a gene, for example, so wherever the plant, in the plants, out of 100,000 plant DNA we have, see, if one plant is having mutations in ACC synthase, for example, or some virus session gene or some virus susceptible gene, then in that plant DNA, when we put it for real-time quantitative real-time PCR and HRM analysis, that plant DNA, as you can see, will separately do segregate, in terms of the melt curve analysis in the quantitative data, and it will show up. Then we can identify those plants. We can also select a homozygous and heterozygous plant. That is the beauty of this tilling. In M2 and M3, we can. It is difficult in conventional method unless you do segregation analysis. But in tilling, in M2 and M3 only, we can identify homozygous and heterozygous alleles. Allele mining and allele segregation we can do, and we can identify plant homozygous and heterozygous plants also. This is useful for in papaya breeding also, because as I told, papaya is gynodicious, and selection for selfing is difficult. Only it's easy possible in hermaphrodite population, and if the females are very nice, then we have to do 2 times half SIB population or full SIB, and then we have to second generation we have to see. But when we put all these things into HRM analysis and QRT-PCR, then we can easily identify homozygous and heterozygous alleles in a particular sample out of hundreds of thousands of population. We have selected a lot of genes, sir, fellow researchers, in terms of shelf life. it's a textbook method of genetic analysis. We have, as we all know, ACC synthase and ACC oxidase, ACC deaminase, and ethylene sensitive response receptor proteins. All these genes are involved, and the downstream genes like polygalactronase, pectinase, pectin esterase, semicellulase, all these genes we are looking at, and we have cloned around 15 genes and pathway genes in this shelf life analysis. So galactronase, etc. And many of these we have cloned for the first time. There's no report in gene bank, and now we are submitting these things data in the gene bank. And in terms of PRSP virus resistance, I will tell you one small beautiful story. Actually, this papaya ring spot virus is very difficult to manage because it's a very tricky virus, potivirus. So, see, there is one protein in host called translation eukaryotic initiation factor 4. See, this is responsible for translation of host genes, mRNAs. Okay, so this is a host protein. So it is present in bacteria to human beings, across kingdom. This protein is available. This is a translational regulator gene, eukaryotic initiation factor 4, EIF4E actually. So host uses this for translation of all the important genes. This virus, papaya ring spot virus, what it does is it goes. There's a specific gene, a protein called VPG, papaya VPG, PRSP VPG protein is there. It goes and hitchhikes. It sits on the top of this EAF4, this host protein, and instead of allowing host protein, this EAF4 protein translation regulator protein to do translation, it will divert it to the expression of its own genes. A very tricky virus evolved over millions of years. See, then host proteins synthesis gets affected, whereas the PRSP viral proteins get multiplied. Therefore, virus replicates, uncontrollably, and then it will spread throughout the plant. That's why this PRSP is, yeah.
Sir, one request, sir, little bit for sir time.
Yes, sir. Another 5-10 minutes. That's all, sir. Mine. No
Still
problem.
7 speakers are there. Still
No
7
problem.
speakers.
Yeah, I know. I'm a last batsman. I'll fast. Yes, enough. No problem.
After that, oral presentation is this. Huh?
Yes, sir. Okay, no problem. So,
okay.
This gene also we are addressing, EAF4 gene. If you mutate this gene so that no function is affected in host, but virus cannot bind to this protein, then we have developed a PRSP resistant papaya variety. So these are the list of genes which we are addressing. So the blue one is the eukaryotic translation initiation factor. This is the gene which is responsible for providing resistance to PRSV. So as usual, routinely we have put it in BLAST and NCBI website gene analysis, and we have for various genes. I'm showing only for few genes, synthase, oxidase genes we have done, and we have identified critical regions which we undertake mutations. So these are some of the initial generation M1, M2 mutants we have got. It's a beautiful provitamin rich yellow pulp papaya we have got. See, the orange one is the control, but whereas we have got mutant yellow and different shades of mutants. And these are the different gamma irradiation we have done, gamma and as well as EMS induced mutations in the beginning, and we are doing it. This another Vasconcelia. Vasconcelia is wild type papaya, and the right side one, what we are seeing is a big leaved Vasconcelia, very promising. It may be tolerant to PRSP. We are evaluating, and it has a very high photosynthetic index efficiency. This is the close-up of Vasconcelia mutant, leaf type mutants that we have got, and these are different fruits of different segregations, and yellow pulp this again. And these are different M1, M2 analysis. This is the bottom sequence. How do we select important traits and the mutant particular variant, and then that we do by sequence and analysis and either melt curve analysis. We have found, for example, TG, that variations that nuclear SNPs we have detected, and then we are correlating. This is the HRM curves that we normally get like this in the HRM analysis. This is another female plant, where we have got observed. This is one beautiful high yielding variant we have got. This is the close of that high yielding variant plant we have got, and we have found out what mutation causes this variation. We have identified. This is the R4P16, very good high yielder, average fruit yield 1.7 kg. We have got up to 32 bold fruits, and we also have found out enhanced shelf life, 8.5 days compared to normal 5 days shelf life period observed in papaya from breaker stage to ripening stage. There we have also got some of the dwarf, ultra dwarf mutants like Pusa Nanha type. So we have got in our population also, which are very good candidate lines for pre-breeding lines. We have also done metabolomics data after genomics and proteomics also. We have done metabolomics data, and we have found out respiration rate, ethylene, and average fruit height in terms of fruit shelf life analysis, and we have identified promised lines. So these are different lines. You can see top right corner, the plants are already, fruits are already rotten, but some of the plants, even after 10 days of after harvest breaker stage, nothing has happened. It's green stage only. So this is the curves, HRM curve. What we done. Sir, that's all what I wanted to speak out. What we are doing in our lab, a beautiful, challenging technology. And I must thank at this stage our co-collaborators and co-workers, scientists, Dr. Manamohan, Dr. Vasuki, Dr. Sudhakar Rao. And I must be thankful to our director, Dr. Dinesh. Himself is a papaya breeder. He developed Arka Prabhat, and he encouraged me to take up this challenging task, and he guided us. And I must thank at this stage all the organizers of this international e-conference for inviting me so that I can share some of our lab works, and all the students of my lab. I must be thankful. Thank you so much for the patient listening, sir. Thank you.
Thank you, Dr. Vagish Babu, for giving a insight in reverse genetic method of breeding, that is which is having a high selection efficiency, that is stilling, and particularly is the only one lab in India working on this method. You must be congratulated, you and your team. Thank you for giving a brief insight with the good photographs. With this, I thank organizers also. Now we will move on to our oral presentation, starting with Dr. Roshni Sharma, who is going to give expression of bunch architecture related triads in the F1 progeny of Silvio Villard into Thompson seedless, that is grapes.
Yeah.
Dr. Roshni.
Yes, sir, I am here. I think first presentation needs to be unshared.
Your presentation has to be shared, madam, and you have to restrict to the timing, the 10 minutes.
Yeah, so it is there, but I think previous presentation needs to be unshared. I'm not able to share this one.
I don't know.
Yeah.
I am.
Yes, madam. Yes, madam. We'll facilitate. Yes.
Okay.
Lakshmi Devamma, please.
Yes, sir. Yes, sir.
Now, whether she is going to present or
yes.
Yeah, please go ahead.
Sir, Vagish Babu, sir.
Yeah, I'm here.
Yeah, sir. You please undo sharing, sir.
No, mine is over. So if I want to give second time, I can give, no problem.
No, sir. Madam, you can share.
Yeah, it is there. It is visible.
Yeah, it is there.
Yeah.
Yeah, you are visible.
Yeah, okay. Okay. Namaskar, everyone. Respected chairs, organizers, and audience. The topic of my presentation is expression of grape bunch architecture related traits in the F1 progeny of Cervilla and Thompson seedless. Looking towards the scope of the study, when we see the grape production in India, it is 2,920,000 tons, and 80% of this production goes towards the table purpose. When I say table purpose, it means which is consumed fresh, and the consumer prefer bunches which is having very good appearance, and for that bunch architecture plays important role. And here in picture you can see one is a compact bunch and another is well developed bunches. In order to develop such kind of well developed bunches, growers usually do chemical intervention as well as physical interventions. Chemical interventions, it includes the application of gibberellic acid. Around 5 to 8 application of gibberellic acid are done, and it affects, it's very stage specific. And It helps in bunch elongation as well as in the berry sizing. And the second type of intervention, that is, it requires the manpower, that is physical intervention for bunch and berry thinning. It allows the space, or it develops the space for development of berries within bunch, because usually consumer likes uniform berries within bunch. Looking toward the costs of these intervention, it takes nearly 30,000 to 50,000 rupees per acre when we use such intervention. And besides that, excessive use of gibberellic acids may also lead to decreasing or deteriorates the quality of the product. And further, the untimely or no availability of laborers for doing such operations, vertical operations such as bunch or anything, that is also problem. Therefore, growers' demands for those varieties having naturally loose bunches and bold berries in this bunch architecture play important role. Therefore, we initiate our study study to see the bunch compactness or bunch architecture in F1 population of Servuland and Thomson seedless, with the two objectives: to study the segregation pattern of bunch compactness, and the second, which are the major traits which are contributing in compactness. For this, we utilize 113 F1 population, which has been developed after the 10 years of breeding efforts. And targetedly, we only focused on the 6 parameters which we found the potential to be contributing in bunch architecture. And it seems to be only a few characters, but each characters, they are quantitative inheritance. Therefore, breeding program, it is very difficult to improve on these characters. Therefore, we need the assistance of molecular approach. Based on the data available with us on bunch and berry character, we did analysis of various variants using the SAS program 9.3. In that, all the parameters, they showed significant difference and wide range of variability we could able to record in our existing population. Here we can see that variability for all the 6 parameters, and maximum we could able to record in bunch length 22.35. But in addition to that also, more than 10% we could able to record in all other traits. Then to find out the variability existing in our population, we carried out principal component analysis. Total 6 components were produced, and among these, first 3 itself able to showcase the 80%, near about 80% of variability, and the first principal component, it expressed around 41% of variability. It can be also easily depicted from the graphical representation presented over here. Then we move towards to find out which are the traits to be focused and responsible for contributing variability, and which can be further utilized for the breeding program. So in first component, we can find out here the berry diameter and the length was the major contributors. Then followed by, when you see the second component, bunch length and the pedicel length. Here in this figure also, it can be easily seen that berry diameter and berry length, they are the major parameters. Then we find out, we have done the correlation analysis, particularly with the bunch compactness along with the other 5 traits. In here also, it was confirmed that berry diameter and berry length was significantly and negatively related with the bunch compactness, and that was the need of our grape growers also. So, and based what are the data available with us on the bunch compactness, we try to find out this segregation of our progeny in different categories. We divided bench compactness, we have indicated in terms of number of berries per centimeter. We have categorized into 8 categories and try to find out the distribution of our population. They have shown the normal distribution pattern in our indicating their additive gene effect. This is what up till now what I have presented, that is the phenotypic representation or phenotyping of our population. But what's next? How this data is going to help us in our breeding program or our actual application? Then coming towards, along with whatever the data we have generated, that we are going to utilize to combine with our molecular study. This molecular study is still not completed, but what we are going to do, we have already completed the first step, that is next generation sequencing of the hybrids. All the 113 hybrids which were phenotyped, that were also we have given for sequencing. And here grape is having one advantage, that the reference genome is available, but the reference genome is available, but the annotation, gene annotation is poorly understood. This is because the progeny development itself is a challenge in grape breeding program. Therefore, if for any kind of gene annotation program, we need the large progeny. I think the progeny what I am presenting over here, this is the largest progeny of the India of the grape of particular kind of crossing. So this we are going to utilize in identify the certain genomic region of trait of interest. Here we are targeting the bunch architecture. And here within traits also we are concentrating on berry length, berry size, berry bunch length, and other characters. So next generation sequencing has been completed. Now what we are going to find out the SNPs using the genotype by hybridization method using the microarray chip. And after retrieving the SNPs data and processing, we will be able to develop SNP markers. And after developing these markers, we will do trait association analysis for SNPs. Coming towards the conclusion, the data which has been presented, this util information which have generated till now, phenotypic and this genotypic information will able to identify the genomic regions influence the bunch architecture, and based on that we will be able to identify the closely linked SNP markers which are going to be utilized in our marker assisted breeding program. And definitely this is going to be very useful for the breeder, particularly for developing the varieties with the loose bunches and bold berries, and ultimately it is going to reduce the cost of production of grapes. Thank you.
Thank you. Thank you, Roshini.
Thank you, sir. Thank you.
For giving a brief highlights of the grape breeding. But
thank you, sir.
Hello. Which center? From where you are speaking, I was not knowing.
Sir, I'm from ICR NRC Grapes, Pune.
NRC Grape? Okay, yes. Okay, I am from Balkot. Okay, thank you. Next, I will call Dr. Himabindu. Himabindu, please. Yes. Yeah, please. You are from States, huh? You are come back to India or in States?
No, sir, I've come back. I joined back.
Okay, welcome back, madam. Welcome back.
Thank you, sir. Thank you so much, sir. I visible. Hello.
Yes, madam. It's visible and audible, madam.
Yeah. Good evening, everybody. Respected chairman, co-chairman, and fellow delegates. I'm here to present before you a little bit of work what my student, as a part of his PhD work, he has done. Unfortunately, he was not able to present it physically today, so I'm filling up. This is the work what we have done in beetle vine about the ploidy determination and 2C nuclear DNA contents in sexually dimorphic clones of beetle vine. This we have done it through flow cytometry. Just to give you a brief introduction, beetle vine, as you know, it's a perennial dioecious climber, and it is also an important heritage crop of our India. And we know that it is very intimately connected with all the customs and religious ceremonies of our country. And it is also a very important commercial crop, almost occupying around 55,000 hectares throughout India. And it is also an important cash crop, giving 20 million people livelihood, and it considerably 30 to 40 million foreign exchequer returns. And it is also a native crop of our India, and it's cultivated in addition to India and some of the Southeast Asian countries. And the leaves are also have medicinal properties. And basically, there is, though it is a dioecious crop, there are not much studies on how the gender discrimination or distinction can be done at an early stage. Since at IAHR, we are a national active gene bank site for this crop, and also we do a breeding work to develop the hybrids. So we found this area to be an important area of research. And as a part of his PhD work, Mr. Manjesh has taken up this work, and we have done, in addition to this, we have done a detailed metabolic profiling of these clones. So the basic objectives are like to study the variation in 2C nuclear content among the male and female clones. As I mentioned before, it is a dioecious crop, and we need to know which are those clones which are males and females at the early stage. And even when you are doing crossing, some crosses, they don't, many of those crosses, I should say, they don't give us a very viable and good seedlings. So to understand how this gender discrimination, and also is there any ploidy variation, since it's already known to be a polyploid, but exact ploidy status is always a different, very different reports are there. So we try to look into this aspect of estimating the 2C nuclear content and understand what is the ploidy variation among male and female clones. So this he has done basically taking 10 male clones and 10 female clones from the germplasm maintained at our National Active Gene Bank site at IHR. And we have done 2C nuclear content and ploidy estimation through flow cytometry. And the Pisum sativum CV citrate, which he has procured it from abroad as an internal standard for the known genome size, and Piper beetle clones Simroli Babna as an internal reference for the ploidy status. It is already published, it's a tetraploid. So we have taken this clone to estimate the ploidy status. So these are the clones which we have used in our study. We can see that there are 10 female clones and 10 male clones collected from all over the India. And as you know that these are the kind of flower structure you get. This is the female clone, which is CV Karapaku. And you see the female inflorescence here. This is the male clone and this is the male inflorescence. It's a very strictly dioecious crops. And we have developed some of the hybrids. Even in hybrids, we see a lot of segregation for the sex. And we get very few male plants which are very viable and which are economically can be cultivated. So we are of the interest to see basic information, to develop a basic information to understand the ploidy variation among these clones, which can help us in designing a better hybridization program. And we have also tried to do extensive cytology work, but we found these difficulties. So this has a very high condensed cytoplasm and high number of chromosomes, as I was mentioning, anywhere between 52 to 119, 120, and very small size chromosomes, as is known in Piperaceae. And since it's a very smaller chromosomes, even if we get a very good preparation, we had a difficulty in counting the number of chromosomes, which is very important to finalize the ploidy status. So in addition to doing the cytology, we thought we can do flow cytometry studies. So we did use the facility present in the NCBS Bangalore. And this we have done based on whatever the published procedure by Puralai Latam, and with a little modifications because beetlevine needed a few modifications in that procedure. And we have used the root tip samples for the nuclear extraction, and we use this Partek high resolution kit type Partek, which is from Germany, according to the manufacturing instructions. And this is a procedure. We took the young root tip and then thoroughly washed, and it is put it and dried on a blotting paper, and the root tips are chopped with the blade, and then added the nuclei extraction buffer, which releases the nuclei. And this was filtered and added to the sample tube through a filter. And we have added the staining buffer, basically, which has come with the Partek kit. And the fluorescence was measured with the fluorescent flow cytometry. And this DNA fluorescence is taken in comparison with the already internal standard for the genome size as well as for the ploidy status. And basically, this is a simple brief procedure. And this calculation of 2C content was done using this formula, where we take the mean fluorescence of the sample with the mean fluorescence value of the standard. And using this formula, we calculated the total TcDNA of the content of the samples. So this is the graphs which show clearly This is the which one showing as a reference, which has a 9.09 picograms of nuclear content. And this is the male female clone, which is showing 1.46 picograms of 2cDNA content. And this is the female male clone, which is showing around 1.06 male content. And these are the different graphs which are showing for all the clones which we have taken in the study. And determination of ploidy, as I was mentioning, we have taken a reference sample of the beetlevine clone, which is Simroli-Bhavna, which is known to be tetraploid, as a reference. And based on that, we have calculated as from the flow cytometry, we got that information for the ploidy levels of the different clones. And you can see this is the female clone, which is a tetraploid. And this is a male clone, which is showing a triploid status. And this, after doing all the content analysis and the ploidy status, we can see that among the 10 female clones, we could see all kinds of ploidy status we found. A triploid, hexaploid, pentaploid, tetraploid. And the amount of DNA content is also, variation is very high from 0.84 picograms to, picograms to 1.80 picograms. And the mean content of genome size in the females is 1.32. When you come to males, we could see a majority of them fall into triploid category, but there are few clones out of those 10 clones. There are 2 clones which have shown the tetraploid, but the variation among the clones for the DNA content is quite narrow, that from 0.92 picograms to 1.20 picograms. And the mean content of 2cDNA content is also less in males. And this shows, for example, in our process, we get a very good seedlings from when we use Sirgamani 1 with any of these males. So now we see that because both are having the triploid status, probably we are getting a good hybrid population in these crosses. So this kind of information helps us in designing better hybridization programs. To conclude, the average genome size is high in females and less in males. And the variation among female clones is high, genomic size is high, and as well as we find the different ploidy status which are present in the female clones, all triploids, tetraploids, pentaploids, hexaploids. And in male clones, the variation for the content is less, right, from 0.92 picograms to 1.2 picograms. And we see a majority of the male clones fall into a triploid category. So though we found 1 or 2 male clones which are of tetraploid, so it shows that the different ploidy levels exist in the whole species alts together, and more so in among the female clones, where you find more variation for the content as well as the ploidy status. And in males, though there are published reports which say that they are mostly triploids, we find initially the reports say that the females are tetraploids and males are triploids, but our study clearly shows it is a complex group of polyploidy, and we find a lot of ploidy differences among females and among males. And this is the first kind of report what we are publishing with the 2C nuclear content and the ploidy status together. And of course, there is always a need for validating these results through a conventional cytological studies, which probably will be taking in further course. And this helps us mostly in understanding the gender discrimination. Along with the metabolite profiling data, we are going to see how these are different as a males and females. This information is important to us because there are certain areas where male clones are grown in an extensive area, and in most of the North Indian states, the female clones are grown. And this kind of information helps in basic understanding how gender discrimination is happening in this species. And also this information
Thank you, madam. Thank you.
for designing the breeding program. And I thank you for organizers for giving me this opportunity. Thank you very much.
Thank you, Dr. Himabindu, Principal Scientist for IHR, for giving a different ploidy levels available in India. With this, okay, thank you, Doctor. Now we will move on to Dr. A.K. Verma. 1 minute. Who is a scientist, ICR, CIH, Bechwal, Bikaner, Rajasthan, going to give a topic on performance studies on tomato brinjal, and chili germplasm for improvement under hot arid agro ecosystem. Dr. Verma, please.
Yeah, thank you, sir.
Yeah, please.
Good evening to everyone. I am Ajay Kumar Verma and will be presenting my oral presentation on performance studies on tomato, brinjal, and chili germplasm for improvement under hot arid agro ecosystem. So, as we all know that hot arid zone is characterized by very high temperature, erratic and low rainfall, drought, high temperatures will range from 0 to 48 degrees Celsius, high soil pH, many diseases and pests. Under such harsh climatic conditions, cultivation of vegetables under open field condition results in poor and low quality yield. Looking to those constraints, the performance studies on tomato, brinjal, and chili germplasm were taken out for improvement under hot arid agro ecosystem. As we all know that fruit set of brinjal, chili, and tomato is very temperature sensitive. If the temperature goes beyond the optimum temperature range, the fruit set and yield of brinjal, chili, and tomato is getting adversely affected. So the genotype of AHB03, that is CIH22 of brinjal, Mathania type selection 1 of chili, and AHSL1 of tomato, these three genotypes were evaluated with three environments during rainy and summer season to understand growth, fruiting, and quality and further improvement. And this is the general field view of the experiment under varying production situations. And this is the improvement of brinjal genotype under hot arid environment, that is AHB03, CIH22, that is thorny type, which I will describe later. And this is the growth and fruit yield parameters of brinjal genotypes under varying production situations. So the genotype has thorns on leaves as well as on fruit calyx, which is a desirable character of any genotype to be cultivated for high temperature conditions as well as drought conditions. And the thorn, which was in the range of 5 to 8 and 12 to 20 under varying production situations. Thus, the genotype has the ability to tolerate high temperature as there was continuous fruiting during hot summer months. And under these varying production situations, we could be able to achieve the fruit yield potential of 2.2 to 3 kg per plant, and the fruits were purple in color, which was retained throughout the fruiting period, which is very important under high temperature conditions. Thus, the genotype has the potential to be grown both during rainy and summer season with good quality fruit yield under hot arid conditions. Here, it was interesting to note that the two characters which are desirable for high temperature conditions, that is thorns on leaves and fruit calyx, as well as different flower, that is medium style, long style, short style, and pseudo short style, 80%, more than 80% of the flowers were of long styled in nature, and which was the reason for the high fruit set under high temperature conditions. And the pollen viability was 73 to 84% during summer and 89 to 95% during rainy season. And length of long style ranged from 9.29 to 9.3 mm during summer and 9.42 to 9.45 during rainy season. And this is the high fruit yield potential under high temperature conditions. Similarly, the performance studies on chili for improvement under hot arid agro system was also taken up. And there was a signal difference in the performance of genotype with respect to plant growth, flowering, and fruit set. The genotype had low fruit set during summer under all the three environments and hence needs further improvement. And this is the performance studies on tomato for improvement under hot arid agro ecosystem. And this is the growth and fruit yielding parameters of tomato under varying production situations. It has the high fruit yield potential of 1.59 to 3.68 kg per plant. And the ripe fruits were medium in size and reddish in color. It has the fruit yield potential under high temperature conditions. And in the next slide, this is the fruiting pattern of tomato genotype AHSL1 under high temperature conditions of. And there was good fruit set during the month of May and June when the temperature ranges to up to 48 degrees Celsius. So coming to the conclusion, pollen viability, pollen germination, fruit set per tress, tress per plant, fruits per plant, and in case of tomato, And in case of brinjal, ratio of long, medium, and short and pseudo-short style flower were the most important selection characters for fruit set and fruit yield under high temperature conditions. And thus the genotype of tomato and brinjal has the potential to be grown during both rainy and summer season with good quality yield and can be advanced further as a varieties for high temperature conditions of hot arid agro ecosystem. Thank you very much.
Thank you, Dr. Verma, for giving an insight of performance studies of tomato brinjal in hot climatic condition. One, my question is, what about the solacid development of brinjal in high temperature?
No, that we have not taken up, sir.
So that is the character you have to study. Without that, high temperature means solacid directly correlated.
Okay,
You have to take that. Okay.
Yeah, thank you, sir.
Thank you.
For
Thank
the
you. Thank you.
suggestion.
Now, I will. Okay. Now I will call upon Dr. M. Dandapani, who is Assistant Professor, Genetics and Plant Breeding in Department of Basic Engineering and Applied Sciences, Agriculture Engineering College and Research Institute, TNAU, Kumalur. Dr. Dandapani, please. Hello, am I audible? Dandapani? Hello?
Yes, sir, you are audible, sir.
Yeah, he is not replying. Dandapani.
He's sharing, sir, sharing his slides.
He is sharing. Hello?
He has to unmute. He has to unmute you.
Who, me?
Yeah, I unmuted.
Yeah, okay, now audible. Please.
Okay, sir. Okay, sir. So thank you for giving me the opportunity to present my talk on cold tolerance breeding in rice. Actually, low temperature during pre-booting stage, like reproductive stage, affect the normal physiological functioning of rice. So it affects the pollen fertility, anthesis, as well as fertilization, leading to sterility. It is also affecting the microspore development from the tetrad stage. So it needs reducing sugar for the energy to form microspore from the tetrad stage. So it will be arrested. The low temperature also affect the panicle emergence from the boot leaf. So only the portion of the panicle is emerged and the remaining panicle will be inside the boot leaf. So it will not be exposed to sunlight and there will not be any fertilization and seed formation. And commercial varieties are fine grain varieties, they are all sensitive to cold. And around 6 million hectare of land is 6 million hectare of land is being affected by cold globally, causing huge economic loss. And specifically, I am sharing my experience of Krishnagiri Dharmaburi district, where I worked before in Tamil Nadu. It belongs to the northwestern zone of Tamil Nadu, which is affected by low temperature induced cold stress for rice. The farmers are going for sowing of crop during September, October, where the land is receiving water or rainfall later stage. Usually, the optimum and normal season is during June first week, but the delay in monsoon or delay in rainfall, some region of the districts they go for September, October sowing. So to address the issue, I had chosen 16 rice lines, rice genotypes consist of popular varieties are being grown during June season, as well as some cold tolerant, moderate tolerant lines being grown during September, October season, and some popular donors and on local landers to fix the cold tolerance parameters, because this kind of work was not done before. Usually, even the identifying the cold sensitive symptom is a difficult one. Actually, it is being always confused with bacterial leaf blight or some physiological unknown reason they will say, and some sheath blight, sheath rot. Sometimes it's being confused with some insect damage, but it's not so. It is due to the genetic regulation of the trait. So I had chosen 16 genotypes, and I evaluated for both the season, normal season during June and October season, which is a cold season. So the main parameters I studied based on the ERI standard system of evaluation is pollen sterility, spikelet fertility, and panicle emergence. These are the three key parameters I studied for the cold tolerance and other yield and yield attributing parameters. By doing so, I could able to identify two extreme cold tolerant lines, Anna 4 and Karpunelli, which is a local land race based on the panicle emergence, spikelet fertility, and pollen fertility and yield. And AT39 and IR20 Co43, they were all moderate tolerant. And IR20 and AT39 are late sown variety. They are popularly grown in this region by the farmers. So these are the panicle emergence pattern. This Anna 4 is having complete emergence, and the popular commercial varieties BPT 5204, and in White Pony is Missouri actually, and in Bhavani, they are all very poor, having very poor panicle emergence, as well as they are completely sterile. There were no pollen formation at all. Very few seed set, very few viable pollen. Actually, I'll show the slides. These are the yield comparison between normal season as well as during cold season. The blue one is October sowing, and June one is normal sowing. You can see the drastic yield reduction in cold season. So these are the panicle emergence pattern of moderate tolerant lines. This ADT 38, IR 50, MDU 3, AT 39, Co43, MDU 4, and IR 20. IR 20 and AT 39, they are the moderate tolerant and popular late season varieties are being grown, but still they have not completely emerged. Kindly see this region; they are all enclosed, and whatever the spikelets are there, they are all sterile because they are not exposed to sun. Upon exposed to sun only, they will digest the pollen and they will pollinate. So these two lines are extreme tolerant lines. You can say resistant. This is Anna 4, complete panicle emergence with complete grain filling without any chaffy grains. Almost the fertilities are almost more than 90%. And this is the local landrace being grown in that region for late sown condition for several years, several years. I can say decades. But the area under this cultivation of this variety is, it's not there actually, it's complete extinct actually. I got this line from I collected this line from a merchant who was maintaining it for its cooking purpose. Some rice cake, and it is also used for idli making. For that special flavor purpose, special cooking purpose, that farmer was maintaining in a smaller area of 1 cent, 1 to 2 cents. So I got the seed from him. I could find out two types of lines were there. The one is on type. On types. On types are always correlated with the abiotic stress tolerance. So it is also it is also showing the cold tolerance pattern. So here comes the popular commercial varieties which are highly sensitive to cold. We can see all the chappy grains, poor panicle emergence. You see the Missouri, White Pony is Missouri, the famous variety, and BPT 5204. The spikelets are compact. They are not exposed to sunlight. They are all sterile, and there is no proper emergence. This is Vasundhara is a private variety. It is also famously grown. When it is grown under September condition, it's showing the sterility. And Payurunu is also a ruling variety. It's grown during June, but when it is taken up during September, you cannot see any grain. All are sterile. You can see the pollen studies. These are the moderate tolerant lines. AT 43, 39, IR 50. This extreme tolerant line Anna 4. The Vasundhara is the sensitive one. All the pollens are sterile pollens actually. They are lightly stained. Based on these parameters, I evolved segregating population from one class AT43 and PS2. From that, in F2 plant, I identified a cold tolerant line during November, December. I mean, the sowing was taken up during October, and the the tolerant line was identified during December under cold temperature, less 14 degrees Celsius. That line was self-put to F6, and then it was identified as PYR told as 0701 with fine grain quality with duration of 105 days. So I compared the performance of this PYR told as 071 with the popular late season varieties 80, 13 and IR20. It's having better yield of 4.5 tons under cold condition with complete panicle emergence. Here you can see the complete extraction of panicle from the boot leaf. Another speciality of this variety is the panicle is above the boot leaf, so it is completely exposed to sunlight. Also, this boot leaf is angular, like it's like a Japonica. It's like Japonica background, so you can have the 45-degree leaf angle, which is having higher photosynthetic efficiency. All the ear heads are above the boot leaf, so that they are all exposed to sunlight. So there will not be any non-dehiscence of anthers. So that's one kind of adaptive mechanism by this variety. The rice grains are fine quality, and 13.9 gram is the under 1,000 seed weight, and the fine grain quality. The cooking quality also good actually. Cooking quality is also good.
Dandapan, sir, you have to be little bit faster.
Okay, sir.
You have to be little bit faster.
Okay, sir. Thank you, sir.
Oh, is it over?
Hey, over, sir. Thank you very much, sir.
Okay, thank you for giving.
This project was funded by DST, so I'd like to thank DST for giving this
project.
Yeah, yes.
And
sir,
also very good studies you have made. Pollen fertility and other things in the post season is very less.
Thank you, sir. With the financial assistance from DST, I could develop that facility, sir.
Yeah, if you can develop tolerance varieties for cold season, it will be a great boon, particularly for Tamil Nadu and also for scented rice.
Sir, in Monday, sir. Monday it's happening now, sir. Actually,
in Monday it's happening, and scented rice also in the Monday. There some farmers are keeping the seed collections also more than
yes, sir. I can go for that exploration, sir. Definitely, sir.
Okay, thank you.
Thank you very much, sir. Thank you, sir. Thank you. Thank you, sir.
Now I call upon Dr. G.C. Manjunatha Gowda. Manjunatha Gowda, who is a scientist.
Yes, sir.
Yeah, 1 minute. Oh, God. Scientist from NRC on onion and garlic, is it not?
Yes, 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 sir. Yes, sir.
Yeah, please go ahead with your topic. That is marker-resisted selection of male child CMS8 cytoplasm and male fertility restorer locus in onions. And please be brief and adhere to the timings. Please go ahead.
Yes, sir. Yes, sir. Good evening to the chairman and co-chairman and delegates of the conference. I welcome you all for my presentation on marker selection of male sterile cytoplasm and male fertility restoration locus in onion. At the outset, I would like to bring your attention to the acreage in onion. India is the largest area in the world and second largest producer for onion. Whereas if you see the productivity, India...
Manjunath Gowda.
Madam.
Please share your slides.
It's not sharing?
No, it's not sharing.
Please move the window.
We are seeing your folder itself only in that.
Only folder, yeah.
Is it?
If it is not, we will go to the next presenter. Then we will come back to Manjunath Gowda. If there is a problem, shall I call Usharani? Okay, Manjunath Gowda, proceed. We are seeing your slides.
Okay, okay, sir.
Is there any technical problem?
Is it visible, sir?
Yes, sir, we can see your slides, but put on presentation mode.
Now is it visible, madam?
Yes, presentation mode
Visible.
is not on.
Is it in on presentation mode?
No, no, it's not. Now it's okay.
Okay, okay. Good evening to the chair and co-chair and delegates of the seminar, and I welcome you all for my presentation on marker stress selection on male sterile cytoplasm and male fertility restoration locus in onion. And at the outset, I would draw your attention to the production acreage. India is the first largest in area and second largest in production. Whereas if you see the productivity the India is comparatively lower than world average productivity compared to the other countries like USA, Egypt, Iran, and Turkey, because in India only grown onion acreage covered by open pollinated varieties, whereas other developed countries focused on using F1 hybrids in order to exploit heterosis for productivity in India, and we need to go for exploitation of male sterility. Otherwise, very difficult to compete with other countries like USA, Egypt, Iran, and Turkey. And the next question come to what is the significance, importance of male sterility? Why we need to go for male sterility system in the development of F1 hybrids in case of onion? And since onion bears very small flowers in the umbel, very difficult to emasculate, very tiny flowers. And more number of flowers per umbel. There are hundreds of flowers. When you do emasculation, during the process of emasculation, there is a possibility of submitting our pollen contamination to one flower to other flower. And this is the one of the complex thing in onion emasculation, and less number of seeds per cross, because in onion we can get only 4 to 6 seeds per cross, whereas in case of other solanaceous or cucurbitaceous seeds per cross, you can get more than hundreds of seeds per cross. So this is one of the limitation in onion breeding. And laborious crossing techniques, very difficult to do hybridization in case of onion by manually, and it's a very laborious. In this view, the male sterility came into the force for the exploitation of hybrid vigor in terms of competing with Western countries with respect to the productivity of Indian short day onion types. And in order to develop male sterile types, especially in case of short day onions, we made a survey in the onion growing districts, major onion growing districts of Karnataka, that is Bagalkot, Chitradurga, and Davangere, Chikmagalur, Chikballapura, Ballari. Nearly around 58 farmer growing onion lines were collected, populations were collected and examined for the bolting traits, especially for identification of male sterile lines in onion in our own short day types. And some of the population we could able to identify the male sterile phenotype. In the figure one depicted the umbel with flowers showing translucent yellowish anthers. We could not able to produce viable pollen grains visually. After confirming visual male sterility, these anthers are subjected for microscopic assay. And the next figure shows that these anthers are shriveled, shrinked, and these anthers do not have any pollen sacs inside the anthers. These are all empty anther sacs without any pollen grains. So this is confirmed that this is the menstrual phenotype. And further, we examined for palynology studies pollen stocarmine assay. for staining studies. We can observe that dead pollen cells adhere to the tissues of the anther. Whereas if you see the contrastingly, the fertile umbels, this is the umbel which having predominant anthers with protruded, predominantly visible anthers in case of umbel, and anther have a bulge with prominent development of anthers, combating with very good pollen sacs. And this is the very visually observing the viable pollen grains in case of fertile plant. And further, those whatever the maelstral lines were observed through morphologically, those lines were linked with marker trait by the validation of URF 725 marker, that is open reading frame segment marker developed by Kim 2009. Those marker has validated in hypothetically visual phenotypes and confirmed those lines were with CMS S cytoplasm. And further, rest of the populations were validated with for restoration of fertility and maintainer lines, and the marker PSAO marker were used. It was developed by Bang and et al. 2011. We confirmed that most of the lines are combination of dominant alleles, and the monomorph indicates the dominant alleles with restorer of fertility locus, and the heterozygous combination reveals that the combination of recessive as well as dominant alleles, so that these heterozygous alleles would be used for isolation of male sterile maintainer lines, and further these were used for the isolation of maintainer line. And frequently these heterozygous lines were crossed to the male sterile lines to maintain the male sterility, and the segregations were isolated as a maintainer. Finally, we could come up with 4 male sterile lines, MS3, MS5 and MS17 and MS25 along with their maintainer lines. And further purification of these lines were taken up by another PhD student in College of Horticulture, Bangalore, under the guidance of Professor Anjana Pasar. And finally, I thank the directors of ICR, DOGR, Pune for granting study leave, and UHS Bagalkot for facilitating the PhD research work at College of Horticulture, Bangalore. Thank you.
Oh, thank you, Dr. Manjunath Gowda, for restricting to the time. And you have given a very wonderful presentation on onion breeding. But one suggestion is while taking the onion bulb photographs, you should also keep the cross section of the onion bulbs.
Oh, surely, sir. Surely,
Yeah,
sir.
that is missing in your photograph.
Yes, sir.
You are an onion breeder, you should know this.
Yes.
Okay,
Yes.
thank you. Now, we will move on to the last presenter of this today's section, that is Dr. Usharani TR. Usharani TR, whether she is joined. Usharani TR, please share.
Sir, she is there. Madam is there. She has started.
Yeah. But I am not seeing the screen.
Hello, madam.
Tropical application of DS or RNA targeting multiple genes of cucumber mosaic virus, CME, in Cucumis sativus. Very interesting topics.
Am I audible?
Yeah, madam.
You are audible, but you have to share your screen.
Good evening, respected chairman, co-chairman, and all the delegates of this international conference on biotechnology and crop improvement. I first of all thank the organizers for giving me this opportunity to share my research findings on topical application of double-stranded RNA targeting multiple genes of cucumber mosaic virus in Cucumis sativus under the theme Advances in Horticulture and Field Crop Improvement. So coming to the introduction, cucumber, it's a widely cultivated plant of the gourd family Cucurbitaceae, and it produces the cucumbiform fruits that have been used as vegetables.
Madam, your slides are not displayed.
Slides are not seen.
Madam, keep on open your slides on the desktop or your laptop and go for this share screen option. From here, you need to select.
Share. Document.
Actually, the organizers has to collect previously. They should give. That becomes the easiest one. Lakshmi Devamma.
Yes,
Jai
sir.
Shri. Jai
Yes,
Shri
sir.
Ugla.
Yes, sir.
Navahari. Yeah.
Yes, sir. Sir, sir.
Next time it is for reference. That's all. No,
Madam,
no
any problem? Can you please send us?
I'm unable to share the slides, ma'am. Just a minute.
Just mail us, madam. We'll upload from here. Mail us that PPT, madam. We'll share. Yeah, madam. You are sharing now.
Yes. Yeah. Okay. Okay. Go ahead and approach. Our approach.
So our approach is RNAi. In fact, RNAi, the silencing approach, wherein the DSR double stranded RNA are being produced by the long dsRNA, the dicers. They are the RNase 3 kind of enzymes. These enzymes, they cleave the long double stranded RNA to the short siRNAs, so which will be incorporated into the RIS complex. There, the double stranded RNA gets cleaved as a single stranded guide RNA to do the site specific cleavage of the target DNA. So in our present study of this, the double stranded RNA is produced by in vitro method by using the 3 different genes of the cucumber mosaic virus. That is code protein, the 2A replicase gene, and the 2B a silencing suppressor gene. So what we are doing here is we are exogenously co-inoculating this dsRNA that is produced in vitro into the control the plants, and we are trying to see that whether it's going to help us in combating the virus. The objectives of the study were to develop the in vitro double-stranded RNA construct for multiple genes and to validate this in vitro synthesized dsRNA against cucumber mosaic virus infected plants of Cucumber sativus, then quantification of the viral load in the dsRNA treated plants by QRT-PCR. So why did we select the CP2A, 2B genes? So as we all know that the code protein is a protective coating on the virus. Then the 2A is the replication. It's involved in the replication of the viral RNA, and the 2B is a silencing suppressor. By silencing these important genes, we are likely to get the resistance to this virus. So our work begins with in vitro double-stranded DNA synthesis. Here, what we did was from the infected plants, we have isolated the RNA and we have performed the RT-PCR. We amplified the code protein, this 2A, 2B genes. So this actually through multiple alignment, we have selected the highly conserved regions of these genes and PCR amplified. After this, we have used overlap extension PCR to anneal all these genes together. So it is we have not individually used these genes. All these genes are being annealed together, and it is being cloned into the P blue script vector, and it was transformed into the DH5 alpha cells. So here we have first piece. That's the first PCR, and the second PCR is we tail this T7 RNA polymerase sequence to this. To this construct, that is the CP2A 2B gene at both the 5' and the 3' ends of DNA using the T7 polylinker primers. The second PCR product will be used as a template for synthesis of dsRNA using a megascript RNA kit as per the manufacturer's instruction. And the concentration of the dsRNA, that is double-stranded RNA, was estimated by spectrophotometer and was subjected to gel electrophoresis for confirmation. So this is the cloning of CMV genes. So the lines, the dark lines represent the genes, like the regions we have selected for making this construct and the PCR amplification. Then isolation and cloning of selected dsRNA. I have told that the conserved sequences was selected with the CP gene 164 base pair, with the 2A gene 125 base pairs, and with the 2B gene 82 base pairs. All these three genes were annealed together. So this schematic representation, you can see that the sense CP, sense 2A, 2B with an intron in between, and so that an antisense CP 2A, 2B is constructed. So this is the restriction digestion confirmation of our clones. And we tailed the T7 promoter sequence onto the 5' and the 3' end of these genes. So it was 364 base pairs, and after tailing, it was 410 base pairs. And this is a schematic diagram. So the first, the construct, we have the sense RNA construct. So in the first PCR, we amplify the portion of the gene, that is CP2A, 2B gene. And in the second PCR, tailing with the T7 promoter. Then after that, we are using the in vitro transcription. We produce the double stranded RNA for application into plants. So this is the components and the quantity of the constituents of the kit. And every 2 hours, we could quantify the dsRNA. It was around 1981 nanograms per microliter. So this is the dsRNA. And in order to validate this in vitro synthesized dsRNA against the cucumber mosaic virus, we co-inoculated the ratio of 4:1. And observation were made on 7th, 14th, and 21 days post inoculation. This was a one-time application we have done, and we characterized then later using the reverse transcription PCR. So in the mechanical co-inoculation, as you see in the table, so we consider them as the vaccinated plants, wherein we use the dsRNA along with the CMV inoculum. And the non-vaccinated plants are the inoculum alone with water instead of dsRNA. So this is being inoculated onto the seedlings of the cucumber. So we could see that on the 7th day, the A, B, and C are the non-vaccinated plants. In A, you can see that on the 7th day after the inoculum mechanical inoculation, we could see clear symptoms on the first the leaf that is formed. But in the D, E, F are the vaccinated plants. The D remained completely immune, and so B and C, so that is 14 and 21 days after the co-inoculation, that is post-inoculation. We have taken the photos, and so 7th day it was immune. Almost at the 14th day it started the symptoms, since it is one-time application. Then so these are the symptom expression percentage, also the symptom severity at different days post inoculation, 7 days, 14 days. So in the vaccinated plants, at the 7th day, there was no infection, no symptoms at all, and severity as well. However, started increasing by 14th day, and so for 21, probably we have to reapply and see that the effect is still there. So in order to molecular analyze this vaccinated and non-vaccinated plants, so we could do a RT-PCR, that is reverse transcription PCR. We isolated the RNA from the vaccinated and the non-vaccinated plants at 7th day and 14th day post-inoculation. In the 7th day post-inoculation, vaccinated plants did not show any band. However, in the 14th day, we could see some. We could see the bands of the virus, which is able to viral title is raising, and so we also have done this real-time PCR for quantification of the viral load. So here the recombinant CMV protein plasmid was diluted, and the standard curve was prepared to calculate the copy number per microliter of the sample. We have used the standard formula of concentration of plasmid DNA into Avogadro's number divided by molecular weight and into conversion factor and Avogadro's mass of 1 base pair DNA. So this is the amplification curve and the melting curve of our samples. So the curve, if it starts early, so that means that the viral titer is very high. So if whatever the curve is going beyond 30 cycles, then CT value is going beyond 30. That means that the viral titer is very low. So in that, so this is the plasmid that first we have to do a plasmid for standard curve analysis, and you can see that the threshold value, it is inversely proportional with the copy number of the recombinant gene in the plasmid. So this is a standard curve. We could get the R square value of 0.97, and I think so. With that the CMV with the water, that is the non-vaccinated plants, you can see the CT value is 16.37. So very early. So the titer is quite high. So CMV with water on 14th day, much. So the CT value is 13, much lower, means the titer is much higher. Viral titer is much higher. So dsRNA with CMV on 7th day, when dsRNA was there, you can see that the CT value has gone beyond 29. So this means that the titer, viral titer is going low in the vaccinated plants. Likewise in the 7th day, 14th day. So with this, what we conclude is this RNAi, it definitely works. And so unlike it's not that breeding or genetic engineering can't, but if at all resistance source is there, there is no issue with breeding unless it takes time. So yeah, with the dsRNA production, so we can use them as a spray. We can also express them in the bacterial systems and get more quantity of the dsRNA and take them for the spray. So this is what I conclude with the study, sir.
Thank you, Dr. Usharani here for giving a nice presentation on dsRNA targeting multiple genes in for fighting CMU. I think with this, we have come to an end of today's deliberation. That is second section. Anybody left? One Achal Kant. I think he is not going to present. With this, I thank the Usharani, and also I will have my final comments today in the second session. That is theme 2, advances in horticulture and field crop improvement. We had got 4 invited speakers, the IS category, and Mohan Rao, Dr. Dinakar Adiga, Dr. Vagish Babu, and there are 6 oral presentations given by Roshni Samarth, Dr. Hima Bindu, Dr. A.K. Verma, Dr. Dandapani, Dr. Dish Manjunath Gowda, Dr. Usharani, and all the topics selected by the organizers and submitted by the participants were very excellent. They are giving insight to the particular themes. For example, fiber genomics in the cotton. He has shown new things in that desired fiber quality, how it has to be obtained, and also accelerated breeding by tilling, a new topic, particularly in only in Karnataka. It's happening on papaya, and also this all the breeding for novel traits in cashew given by the Dinakar Adiga. Pre-breeding a chili pepper improvement also very nice with this, but only one of my concern is many of these biotechnology approaches have lacked the practical applicability. My request is yes, only academic interest, no practical applicability has to be seen. For example, chili, Gauri Bidnur and other things, he has crossed and made a very good selection and other things. I have to come to the farmers field, because in Karnataka, chili is a prominent crop, but the area is coming low, reducing like anything. And also all these breeding methods and other things expressed by the participants and oral presents are excellent. I compliment and congratulate all the invited speaker and also oral presentations. And finally, I thank the organizers of this wonderful international e-conference on advances and future outlook of biotechnology and commitment for sustainable productivity, which is needed for the hour. I thank the organizers and also the Dr. K. Mindresh, Vice Chancellor of Horticulture University, Bagalkot, and also Fakhruddin and his team of members for organizing such a wonderful event. Now, I request my co-chairperson, Dr. Ramesh, to say few words. Dr. Ramesh, please.
Sir, thank you, sir, for giving me opportunity to share my views. As you rightly commented, sir, there was a good mix of presentations covering major commercial crops slated for this presentation. And as per the oral presentation is concerned, there was a good mix there also. And only thing is, there was many of the presentations they lacked the giving good introduction to the topic. So maybe the presenters may improve in the later presentations. Overall, the good selection of the topics by the organizers. And with this, I thank the organizers for giving me this opportunity to quote. And if chairman permits, I have one question to Usharani.
Please, sir.
Usharani is online. Usharani is a good presentation.
Usharani?
Yes, sir.
Okay. Usharani, good presentation. Only thing is, what will be the cost of formulation of the RNA today, and what will be the cost of storability? Are there any issues, or are there any studies on this?
So nowadays they are trying to use the nanoparticles formulations to increase the stability of the life of this dsRNA. So a lot of work is going on in this line. So using nanoparticles and increasing the stability of the storage also. That is one thing, sir. And coming to its economics, so if it is used as using the bacterially expressed, like how we can express the proteins in bacterial systems, so it will definitely work out, sir. It will be it works out to be economical, sir.
Yeah.
This in vitro is just
yeah, that is most important, because research point of view, excellent results. From application point of view, they may become limitations, because if you see the COVID vaccine production results, so there have been 2 vaccine candidates which can be stored only in -75 and one more in -20. But one which is developed by Oxford University and India, AstraZeneca, that can be stored at 2.8 degrees centigrade. That is protein based, probably protein based. So which is more easier to store and then deliver the vaccine to mass of the people. In that direction, I asked this question. Okay. So you need to conduct more research on
yes,
that.
sir. We'll elaborate the research on that line.
Thank you, sir. Thank you, Chairman, sir, for giving me this opportunity.
Thank you, Dr. Ramesh, and also I thank the rapporteurs who are very important. I was not knowing who are there. Thank you, the organizers. Thank you, and all. Thank you.
Thank you, sir. Thank you, Baswaraj sir. And
Go to you,
sir.
Lakshmi Devamma.
Thank you, sir. Thank you all.
Thank you for your excellent management of such a lengthy and diverse session, sir. Thank you for your valuable time and your wonderful words and suggestions regarding this. So thank you very much, sir. I would also like to thank all the keynote speakers, as well as invited speakers, as well as oral presenters of today's session, of both the sessions. Thank you very much. And I request all the participants to log in by 9 o'clock tomorrow. Tomorrow we have 3 different themes with diverse topics and along with oral presentations. Thank you one and all.
Hello.
We will be ending the session.
Hello.
Thank you very much, sir.
Thank you.
Sir, thank you, sir. Ramesh, sir.
Lakshmi Devamma.
Sir.
Okay, phone. Please.
I'll send you the number, sir.
Lakshmi Devamma, congratulations for completing the day successfully.
Sir, thank you.
Okay.
Nice presentation, sir. Thank you.
Thanks, Amma. I would want to talk to Dandapani.
I'll send you the number, sir.
Yeah, Dandapani is online.
Oh, Lakshmi Deva ma, thank you.
Thank you, sir. Thank you very much, sir.
ಆಯ್ತಾಯ್ತು. Okay. ಇವನೇ
Sir, shall we end the session, sir?
ಆಯ್ತು. ಹೇಳ ನಾನು ನಿಮ್ಗೆ ಫೋನ್ ಮಾಡಿ ಹೇಳ.
Sir, ನಾ ಫೋನ್ ನಂಬರ್ ಕೊಡ್ತೀನಿ.
ನೀವು ಫೋನ್ ಮಾಡಿ ಆಮೇಲೆ.
Sir,
ನೀವು ಫೋನ್ ಮಾಡಿ ಆಮೇಲೆ.
I'd like to talk to Mohan Rao, sir. Lakshmi.
Dandapani, how are you?
Sir, I am fine, sir. How are you, sir? Happy to see you, sir. Happy to see you and Ramesh, sir. Yeah. It's like homecoming for me, sir.
Where are you now?
I am at Kumlur, sir. Trichy district, Kumlur.
Okay.
Sir, Fakhruddin sir is also there.
Yeah. It's like homecoming to see Fakhruddin, sir, Ramesh, sir, and you, sir.
Yeah, thank you so much.
Thank you so much, sir. It was a nice presentation, sir, on anthracnose.
Thank you so much. And good to see you after a very long time, actually.
Yes, sir. Almost like a